Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes.
Good day, everyone, and welcome. Thank you for joining us for Day 2 CEDAR track of the Regulatory Education for Industry Conference. For those of you who missed yesterday, this is the 12th year we are holding this conference, and we're very happy that you could join us today from whichever time zone you're in. My name is Lieutenant Commander Renu Lal, and I'm with the Small Business and Industry Assistance, or SBIA program, which operates within the Division of Drug Information within CEDAR's Center for Drug Evaluation and Research's Office of Communications. I'll serve as your host and moderator for today's sessions, along with Captain Ray Ford, who was your moderator yesterday. This 5-day conference will offer a total of 32.25 contact hours to physicians, pharmacists, and nurses. The CEs are available to all real-time attendees who self-identified as healthcare professionals during the Adobe Connect registration process and who attend via Adobe. Please note the emphasis on real-time and Adobe Connect. No healthcare professional CEs can be given for those who attend via YouTube or for post-event viewing. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only of a single supporting company. CE faculty or speakers are required to disclose to the attendees when products or procedures being discussed are off-label or unlabeled, not FDA approved, and any limitations on the information that is presented. We have a spectacular lineup of speakers today, and the planning committee, today's speakers, and CE consultation and accreditation team have nothing to disclose. Participants must attest to their attendance and complete the final activity evaluation via the CE portal, which is ceportal.fda.gov. For multi-day activities, participants must attest to their attendance and complete the faculty evaluation for each day. Final activity evaluations must be completed within 2 weeks after the activity, so the deadline for this 5-day conference is June 23rd. Apologies in advance, but we are unable to make any exceptions to that deadline. Pharmacists should note that failure to provide your correct NABP and date of birth information in the required format may result in the loss of credit for this activity. The NABP profile number should be the 6 or 7-digit profile number assigned by the CPE monitor, and your birth date should be in the MMDD format. For example, 0605, month, day, day. Do not provide your pharmacy license number. Please click the My Account tab and then navigate to Edit Contact Information to verify that your information is correct. Physicians and nurses may then view and print the statement of credit. Pharmacists should log into the CPE monitor 8 weeks after the last session of the activity to obtain their CE credit. Okay, now let's turn our attention to some logistics. As you know, this event is 100% remote in addition to being 100% virtual. Over these next 5 days, we have a total of 42 sessions and 46 speakers and panelists, not to mention our organizers, technical staff, all working from our own locations. So noting this, there may be some internet issues, but please bear with us. And if we encounter those issues, we'll try to get to them as quickly as possible. And speaking of tech issues, here is the link for our one-stop shop for answers to your technical questions, for downloading the files, accessing the survey link to obtain the certificate of attendance, and so much more. This is the same page that you used this morning for your online access to the event. So please bookmark that page. If you have any technical questions about this conference, please check there first, and you will most likely find the answer. We invite you to submit your questions to us so that we have a productive Q&A segment, question and answer segment, which is so important to these events. This is a learning opportunity, and we encourage you to take full advantage of the expertise that we offer here. So when you do submit your question, please identify that individual presenter by prefacing your question with the speaker's name as shown here. So for example, if I'm the one speaking, preface your question with @Renu. Some sessions can literally have over 100 questions, so please keep in mind that while we may not get to your individual questions, they are very helpful. They really provide insight to our faculty. Also, during each presentation, there will be a link to a speaker evaluation under the speaker's picture or information on the right side of the screen. We encourage you to take the time to complete the evaluation. Your feedback is very much appreciated. In addition to the speaker evaluation, we have our general event evaluation or survey, individual to each track. This final survey is the mechanism by which you will receive the certificate of attendance, which will support continuing education credits for RAPS, SOCRA, SQA, and ACRP. When an attendee completes the survey, they can download a certificate of attendance which may be used in support of those CEs. The certificate must be downloaded immediately after completing the survey. Otherwise, the opportunity is lost. Even if you do not want or need a certificate, please still take a few minutes to complete the survey. Your feedback will help guide the design of our future events. This is your opportunity to make us aware of what you need to know. We rely upon this feedback to help us with planning the agenda for next year's events. And again, the evaluation and certificate are only available for 2 weeks, which means that the deadline is June 23rd. Each track has its own survey, and a link will be posted on our one-stop shop in the afternoon of each day's event. As a reminder, all the sessions are being recorded. It will take us a week or so to process the recordings, but you will soon be able to access them from FDA's webpage, fda.gov/CEDAR-SBIA. Also, the direct link for this event is www.fda.gov/REdI 2023. In the interim, the YouTube link for each day's recording will be posted on the one-stop page at the end of each day. Note that the certificate of attendance and the healthcare provider CE submission are two separate processes. However, to simplify things, each night, SBIA will send one email to the attendees of that individual event that day. The email will have the information on both the CE claim codes and the survey link. So now I'd like to take a moment to call your attention to our upcoming SBIA events. On June 15th, we will host our Overview Clinical Pharmacology Considerations for Food Effect Studies webinar. In this webinar, FDA will discuss the importance of assessing the effect of food on drugs and reasons for issuing the final guidance. Then on June 20th, we will present our Decentralized Clinical Trials Draft Guidance webinar, where we will clarify the agency's recommendations on the conduct of these decentralized clinical trials. All of our events are listed on our calendar at www.fda.gov/CEDAR-SBIA. This next slide provides the QR codes for our social media avenues. You can scan them now with your phones and be taken to the appropriate platform. They will be advertised across the screen banner during the breaks if you miss it right now. Be sure to subscribe to our email updates so that you can stay informed not only of upcoming SBIA events, but also CEDAR regulatory actions, guidances, pilots, and initiatives. We also post that information to our SBIA LinkedIn page, so be sure to follow us there too. And now I'm sure you're ready for us to launch fully into today's agenda, and it looks like I'm the first speaker on deck. As a reminder, if you have any questions during the presentation, please type them into the chat pod and address them as @Renu, R-E-N-U.
The journey to develop and market a human drug can be long, complex, and filled with regulations. But FDA CEDAR Small Business and Industry Assistance, or SBIA, is here to help. SBIA provides education and answers to help all regulated pharmaceutical industries, not just small business, all over the world. What do we offer? Web resources for all your questions and training needs, the popular regulatory education for industry, or REdI conferences. Free events that offer direct interaction with FDA subject matter experts and provides RAC credits. We offer free webinars to share FDA's knowledge and expertise, featuring live question and answer sessions, video and audio archives, and virtual attendance from anywhere in the world. We publish the CEDAR SBIA Chronicles, a brief e-newsletter and podcast that highlight a specific and timely regulatory topic. And we offer in-depth CEDAR Learn, web-based learning tutorials. Stay current by signing up for our listserv or connect via LinkedIn. And best of all, everything is free.
I hope you all enjoyed that short animated video. My name is Lieutenant Commander Renu Lal, and I work with Brenda and the rest of the SBIA team. In this presentation, we're going to take a step away from those in-depth regulatory topics and discuss our CEDAR SBIA, or Small Business and Industry Assistance Program, the program that brought you this amazing conference this week, and how you can leverage SBIA resources to educate yourself and to find answers to your questions. In today's presentation, I will focus on how SBIA can assist you. So after listening to this presentation, you should be able to locate our SBIA webpage and identify the resources it provides, identify at least 3 services that SBIA offers to assist the pharmaceutical industry, and finally understand how to register for our SBIA events and also find recordings of the past events in the case that you missed them live. So within CEDAR, we have the Office of Communications, and under the Office of Communications there are 3 divisions, one of which is our Division of Drug Information, or DDI. DDI handles thousands of inquiries that come into CEDAR, and we work very closely with other offices to anticipate and respond to major initiatives. And Small Business and Industry Assistance, or SBIA, falls within the Division of Drug Information. SBIA recently revised our mission to better reflect our current actions and also to encompass our aspirations. Our mission is to provide industry stakeholders with immediate access to resources, education, and training, which allows for a more clearly informed and efficient developmental process and is aligned with CEDAR's goal of approving safe and effective human drugs and biopharmaceuticals. FDA's philosophy is that timely, interactive communication with sponsors during drug development is a core activity to help achieve our mission and to facilitate the conduct of efficient and effective drug development programs. And so ultimately the goal is to enhance public health by making new, safe, and effective drugs available to the American public in a timely manner. And we know that on the industry side, it is not always easy to navigate these complex regulatory processes. So then CEDAR SBIA often becomes the first stop for a pharmaceutical business trying to contact the agency. Our goal is to help these small pharmaceutical and large pharmaceutical businesses, all of industry really, navigate the wealth of information that FDA offers and to assist in understanding human drug product regulation. We want to ensure that industry stakeholders have immediate access to resources, education and training, and so forth, allowing for a more clearly informed and efficient development process with the goal of approving safe and effective human drugs and biopharmaceuticals. And our vision is really to become this premier educational source, to serve as the premier educational source on human drug and biopharmaceutical development and regulation. We want industry to come to us for the answers and for support. And we believe that with our real-time support for industry that you all can be better informed and in turn will have increased constructive interactions with FDA. So like I said, though our focus is on small business, our resources and outreach activities really extend to all of the regulated pharmaceutical industry and are not restricted to small business. Really for SBIA purposes, the term small business is defined as a business that has fewer than 500 employees, including employees of affiliates, but like I said, we are not restrictive. And SBIA is open to everyone. You don't have to qualify by the size of the business. We do help all of industry, but we do focus on helping small businesses since they may not have that expertise and the know-how of large companies. As you can see on this pie chart here, the majority of people that attend our events are from large pharma with more than 500 employees, but that means that about half of them are from small pharma. And this map here shows an overview of where our audience is located. So this map represents the reach of last year's regulatory best practices for global access to medicines conference. So you can see that we have a very global reach, since companies can benefit from pretty much all of our services online, and approximately 30% of our audience is outside the US. So I'm now going to review the various resources that we offer through SBIA, including direct communication services, our web pages, our training resources, and our news and updates. Let's take a look at some of SBIA's outreach services in detail. So we have a staff of over 35 healthcare professionals that answer direct inquiries from industry via phone and email. You call us, we will answer. You email us, we will respond. We have dedicated phone numbers and email addresses, as you can see here on the slide. Last year alone, we responded to over 6,000 inquiries. So we receive a wide variety of questions, and we respond pretty quickly, whether it's providing you with an answer or letting you know that we need to consult a different group within FDA, or provide you with information on whom you should be contacting, but we always get back to you in a timely manner. So, for instance, if a question is specific to an existing IND, the sponsor would really need to reach out to their FDA regulatory project manager. And if it's, a general question about guidances and so on, we can respond to that. And the service is available to anyone, like I mentioned, so please take advantage and contact us with your questions. A huge part of SBIA is our outreach to stakeholders via our Regulatory Education for Industry, or REdI, conferences. And though we're currently at the REdI annual conference, we use REdI to describe our topic-specific conferences too. So that's, so to speak, our trademark. Our audience for these events is small manufacturers of drug products who wish to learn and large, who wish to learn about how FDA approaches drug regulation. We typically have a hybrid format for all topic-specific conferences where people can attend in person or online. However, since the public health emergency, we have switched to 100% virtual. In 2022, we hosted 9 conferences and workshops and reached over 84,000 people in 130 countries via conferences, webinars, and recordings. And we continue to innovate and provide new and useful information for our stakeholders. Last year, we introduced a workshop on regulatory best practices for global access to medicines, including anti-tuberculosis medicines, where we collaborated with USAID, USP, and the World Health Organization, and reached low and middle income countries with top attendance from India, Pakistan, Nigeria, Ghana, and Kenya. We also last year partnered with FDA's National Center for Toxicological Research in the Nano Day Symposium we put on to address the development of products that contain nanomaterials in their formulation. And all of these conferences and workshops, I will get to this in a couple slides, but they're all, if you missed it, they're all posted online on YouTube. We also offer various types of continuing education credits depending on the conference, sometimes providing CEs to support RAPS, SOCRA, SQA, and ACRP, and sometimes providing CE for healthcare professionals. And most, if not all, like I said, of our conferences and workshops are recorded, and they are absolutely free. So in addition to our conferences and workshops, we offer free webinars, most of which also feature a question and answer or Q&A session with the FDA subject matter experts. Examples of recent webinars include an update on field alert reports and biological product deviation reports. We've had some on OTC Monograph, so OTC Monograph Drug User Fee Program, or OMUFA, fiscal year 2023 user fees and registrations. We just recently held that. We recently held a webinar on PDUFA VII scientific meetings. We've had a webinar on electronic systems, electronic records, and electronic signatures, statistical approaches to establishing bioequivalence, and many, many more. And like I said, these recordings are all available on our webpage and on YouTube. So we also have a newsletter, which we call the SBIA Chronicles. We publish this every couple months. They're short 2-page electronic newsletters and audio podcasts. They're accompanied by audio podcasts that we write and we record. We highlight different regulatory issues each time, and we usually interview a subject matter expert, depending on the topic. So some sample topics we've published on recently include pharmacodynamic biomarkers and biosimilars, cannabis and cannabis-derived compounds, adverse events associated with compounded drugs from outsourcing facilities, and continuous manufacturing. So here's a snapshot of our SBIA webpage, our landing page. If you've never visited, I highly recommend that you bookmark it, fda.gov/CEDAR SBIA. Our web pages are another means by which we reach out to industry stakeholders. This here is the SBIA landing page, and it really does contain very useful resources that you may have never even known existed. Up top here, we have a calendar of upcoming events, or SBIA conferences, workshops, and webinars with links to the registration. And this is just the first half of the page. And this is the middle of the page. So as you can see here, there are 4 graphics. Each are hyperlinked to different web pages. The first box is our regulatory references web page, which is essentially a database of regulatory information, which you can search, and I will show you that in detail. We then have our SBIA Learn online training repository. And this repository contains links to all of our SBIA events and also future events, and going back 3 years. It's not only the events, but it also has links to other training opportunities and our Chronicles newsletters, online courses, things like that. The third box is a link to SBIA on the LinkedIn platform, an invitation to connect. And then the fourth box, it links to the SBIA Learning Library on YouTube. So let's go through these subpages one by one. So when you click on this first regulatory references webpage, it brings you to this webpage right here. Like I said, it's a database of information for industry stakeholders, essentially a systematic compilation of FDA web pages, and very easy to search. So here you can find information on different topics related to drug development, from INDs to new drugs to generics, OTCs, information on submissions, labeling, manufacturing, quality, safety, and so on. Just so much more. And you can either use the topic drop-down bar to search for topics, or you can simply use the search bar, or a combination of the two. So here, I've just taken a screenshot showing you what the topic drop-down looks like. So if you find yourself lost looking for something on the FDA website, rather than turning to Google, come here first. That next graphic, the second graphic on the main SBIA landing page, was to the SBIA Learn online training repository, which is basically a database of training information for industry. So this is where you can find links to all the trainings I mentioned, both past and upcoming. Not only does it contain links to the web pages for the SBIA conferences, workshops, and webinars, which in turn contain links to the recordings and slides, but it also contains additional training and learning resources, as I mentioned, such as our SBIA newsletters and courses. And again, this database is searchable by keyword, or you can filter by topic as well. If you click on the topic caret, you'll find a drop-down menu where you can choose topics such as biosimilars, clinical trials and research, drug development, drug master files, drug quality, and so on. And again, I forgot to mention, you can see here I've included the QR codes for each of these pages, so make sure you bookmark them. Going back to the graphics on the SBIA landing page, that last graphic was for the SBIA YouTube Learning Library, which contains our most viewed recordings and contains links to the YouTube playlists. You can also browse our recordings directly from YouTube by visiting the Food and Drug Administration channel on YouTube, clicking on Playlists, and then clicking on the CEDAR Small Business and Industry Assistance playlist. You'll notice there's multiple organized by years. There's 2023, 2022, I think going back to 2017 or 2018. And the SBIA playlists are all easily identifiable with our trademark orange fiery stripe, and they contain the recordings of our SBIA conferences, workshops, and webinars, and which we try to make available within 2 weeks after each event. We post a lot of recordings and we get viewership from all over the globe. In 2021 alone, we posted 225 recordings of presentations with over 477,000 views. So these are very useful. The individual YouTube recordings are also found within the event pages in the SBIA Learn database. But this is another mechanism to browse the events and click directly on the recording links to view them. So we have multiple ways to get to this information. And although we have these recordings available, we still highly encourage you to attend real-time in person and take advantage of the live Q&A sessions to interact with FDA experts and get answers to your questions right on the spot. Real-time attendance to many of our webinars and conferences will also enable you to receive continuing education credits for sometimes for physicians, pharmacists, and nurses when offered, and a certificate of appreciation, which supports CEs for RAP, SOCRA, SQA, and ACRP, just depending on the event. And finally, the landing page also promotes our SBIA on LinkedIn, and it leads to our LinkedIn page for SBIA. We currently have over 27,000 followers, and we use LinkedIn to push information out to industry stakeholders. This could include information about CEDAR and FDA meetings, conferences, workshops, and webinars, Federal Register notices, new announcements, new initiatives. We also have these fun polling questions that we recently started for many of our events, and which I hope many of you have taken in anticipation of this conference. So we encourage you to participate. It's a great way to test your knowledge and kind of anticipate and figure out what's in store for you during the conference, and to get your questions answered there. In addition to LinkedIn, we also disseminate information to industry via our email listserv or distribution list. We have over 143,000 subscribers, and we issue information daily regarding new regulations, FDA announcements and initiatives, webinars, conferences, and more. So you can find a link to sign up for the emails right from our landing page, which was fda.gov/CEDAR SBIA. And if you scroll all the way to the bottom of the page, there's a bar where you can type in your email address and just click subscribe. So this brings us to challenge question number 1. In which of the SBIA resources can you find a database of searchable FDA web pages relating to drug development? A, regulatory references. B, SBIA Learn online training repository. C, in the calendar of upcoming events. Or D, on the SBIA Learning Library on YouTube. I'll give you a minute to answer that. So if you chose answer A, this is correct by clicking on the box titled Regulatory References on fda.gov/CEDAR SBIA. You will find a searchable database of regulatory information, which includes a great deal of information on drug development. Note that the SBIA Learn online training repository contains links to recordings of webinars, conferences and workshops rather than to web pages about specific information about a topic. And challenge question number 2, which of the following statements is not true? A, you can stay connected with the latest regulatory information and offerings by subscribing to the SBIA listserv and following SBIA on LinkedIn. B, industry stakeholders may call or email SBIA directly. C, SBIA services are only available to companies with less than 500 employees, including affiliates. Or D, SBIA offers many free conferences, webinars, and workshops on various regulatory topics. So which of these statements is not true? And the incorrect answer, is C. So SBIA services are available to all industry stakeholders, regardless of company size. So I hope that I have enlightened you today with information about the SBIA program and how to leverage SBIA as a regulatory resource. All of our programs are geared towards training and educating industry across the entire spectrum of drug development. So here I leave you with some action items. Email or call SBIA with your regulatory questions. We respond Monday through Friday, 8 a.m. to 4:30 p.m. Eastern Time. Here's our contact information. Bookmark these web pages, fda.gov/CEDAR SBIA and fda.gov/CEDAR SBIA Learn. And you'll get access to all of this great information that we provide. Browse our SBIA playlists on FDA's YouTube channel. And finally, follow us on LinkedIn and subscribe to our listserv to stay in the know and to stay connected. We're really doing all that we can to help industry, to help you, and to achieve our vision to serve as the premier educational source on human drug and biopharmaceutical development and regulation. So I thank you for your attention today, and I'm available to answer any questions that you may have.
Okay, so we will now launch into our Q&A session. So please enter your questions into the Q&A chat pod if you have not already, and we'll answer as many questions as time allows. It looks like we have a few questions that have been submitted. Our first question: should SBIA be the first point of contact for questions that we can't find the answers for online? For example, would you refer us to the appropriate resource or a person to contact? So great question. Yes, if you cannot find the answer to your question, please feel free to contact us, contact SBIA via email or phone, and we will either answer your question or consult the appropriate group within FDA, or we will point you in the right direction. So if you don't know where to go, contact us and we will help you. Hopefully we can answer your question, but if not, we'll get it from the correct subject matter expert within FDA, or we'll let you know where to go for answers. Okay, we have a couple more questions. Question 2, have you considered providing your conferences and webinars in different time zones to make it easier to attend for people in different areas of the world? So we do recognize that a lot of you are not in the US, are not in Eastern Time, and we appreciate that you are dialing in from wherever in the world you are. And we have considered, timing our conferences at different times to accommodate everybody, but as you can imagine, there's so many different time zones, so it's very difficult to accommodate everyone. We have people attending from India, China, Europe, Africa, different time zones in the US, and from all over the world. So we need to consider all that. We also need to consider the availability of our FDA faculty. So it's easiest to stick to Eastern Time. There are some exceptions, though. For example, there was one pharmaceutical quality extended webinar that we did a few years ago. We called it Midnight Madness, and that was specifically for industry in India and China, where it was the middle of the night here in the US. And we were able to provide that extended webinar in their time zone. But I don't think we'll be able to repeat such a time zone accommodation. It's very difficult coordinating all the speakers here in a different time zone. But we do record the events. We post them to YouTube, so you can always watch them later. You won't be able to receive the continuing education credits or submit the survey to obtain the certificate of attendance, but you will be able to get the information via those recordings. Okay, I have a question. Can we ask an IND NDA specific question? So if you can contact SBIA with your questions and we'll direct you. If it's something that's specific to an existing application, you would need to contact your project manager for that, because there's just so many nuances. We can provide you with general information and guidances, but if it's specific to an existing application, your review team is the one you should be communicating with. Let me see. Okay. Another question. When and where would the recordings for the REdI conference be posted? So for the REdI conference or for any other conferences that we offer, we usually have the recordings up within 2 weeks and in this case, it would be the 2023 playlist of FDA's YouTube channel. So if you visit fda.gov/CEDAR SBIA Learn and click on the REdI conference, it will lead you to the YouTube recordings once posted. And we will also send out an email message for those who register. We'll send out an email message through our distribution list to let people know that the recordings are available. But it's 2 weeks for them, about 2 weeks, sometimes sooner, for the recordings to be posted on YouTube. You can also go to YouTube, go to the FDA channel, and then click on playlists, and you'll see all the SBIA playlists there. Okay, do you work with NIH to help companies in very early stages of drug development looking for grants? So in the past, we have worked closely with NIH. They used to have this annual conference and offered live consultation at these conferences from many different government entities, and we were one of them. So we used to go and participate in this live consultation. And we still work with NIH to help them in a variety of different capacities, but not via that conference. And we're, of course, available via phone or email to answer questions. Okay. Let me see. Does SBIA work with the Office of New Drugs to review applications? We do not work with the Office of New Drugs to review applications, but we often consult them when we receive questions from industry. So we can contact them. We can contact the reviewers or whoever it is in the division if there's certain questions that we need to consult out. And we also, as you can tell from all of our speakers and panelists, we work very closely with the Office of New Drug staff to put on conferences and webinars and all of our events, and to put out our different courses and newsletters. We're always working with them and to determine what it is that we can provide to industry for training and education. So they may tell us what it is that their division needs to, what messages they need to put out to industry, and we help provide that outreach. When will the slides be posted to our website for this conference? So probably within a month. So the recording should be up within 2 weeks, but the slides probably a month, give or take. Okay, we do a couple more questions. Can we find the clinical trial results of a certain drug in regulatory references? So we get this question quite a bit, people looking for the results of clinical trials. So FDA does not publish the clinical trial results. If an application has been approved, you can find some of the information in the review documents posted in FDA's Drugs at FDA database, which is right on our website. You can look the drug up by the name or the application number, click on review documents, and you'll see FDA's review of the clinical trials. But if a clinical trial is unpublished, you're not going to find it on FDA's website. Okay. Although SBIA is under CDER, do you give equal attention to CBER products and topics? So we do sometimes include CBER topics or topics that apply to both CDER and CBER, but our main focus is CDER. On occasion, an SBIA webinar will address cross-center guidances and topics, and will include faculty from all the relevant centers, including CBER and CDRH, if applicable. And this conference, as you know, is cross-center with all three centers, but we do try to work with them. I think that's all the questions we have for now. Let me just take one more look. Yeah, I think I've covered it all. So thank you for your great questions. I appreciate them. We can now move on to our next two speakers, Dr. LaShawn Schnupp and Dr. Lolita Sterrett. Our first speaker, Dr. LaShawn Schnupp, is a Senior Regulatory Health Project Manager and STAR Program Manager in the Program Development, Implementation, and Management staff within the Office of New Drugs, Office of Program Operations. She will be providing an overview of FDA Split Real-Time Application Review, or STAR, pilot program. Our second speaker, Dr. Lolita Sterrett, will present on use-related risk analysis and human factor protocol reviews, what to submit for an efficient review. Dr. Sterrett is Associate Director for Human Factors in the Division of Medication Error Prevention and Analysis 2, within the Office of Medication Error Prevention and Risk Management in CDER's Office of Surveillance and Epidemiology. For a complete biography on each presenter today, please refer to our speaker biographies located in the same area on our website as the conference agenda. Now, please join me to welcome our speakers.
Thank you for that introduction. I appreciate it, and it's great to be here. Hello, everyone. My name is LaShawn Schnupp, and I'm the CDER Program Manager in the Office of Program Operations and part of the Program Development, Implementation, and Management staff. And today, I'll be discussing the Split Real-Time Application Review, or STAR, pilot program, which started in October of 2022. So this slide provides the learning objectives for today's presentation for STAR, and we'll discuss the background of STAR, including PDUFA VII, and then I'll describe the STAR pilot program, including why this program was created, what it is, and its goals. I'll also describe the components of the overall STAR program. I'll outline CDER's review of STAR applications, and also share with you the STAR website. Then we'll go over a quick review of what we learned, as well as the program assessment for STAR. And then lastly, we'll review options for resources. So now we'll go over some of the background for STAR. And so under the Prescription Drug User Fee Act, or PDUFA VII, the STAR pilot program was introduced. And this program became available to applicants in October of 2022. So as you can see here, this is just a snapshot of the letter that can be found online and on the FDA website. And as you can see, STAR is listed under Section D of the letter. So now I'll be going over the STAR program in more detail in these next slides. So you may be asking why we created the STAR program. Well, during negotiations, industry raised a common problem that they faced, which is that typically all of their data sets and other application materials for a supplement are ready a couple of months in advance of 3 components of their applications, which are namely the clinical study report and then 2 of their integrated summaries for effectiveness and safety. And then in instances where the product is intended to treat an unmet medical need, that sometimes can represent a delay for patients getting access to these treatments. And so in order to try and address that problem, we created the STAR program. So what is the STAR program? Well, the STAR program is a pilot program for qualified priority efficacy supplements to be submitted in 2 well-defined parts. And I'll talk to you in just a minute about what those 2 well-defined parts are, but really the intent was to enable an earlier review and action in order to overcome the problem statement that I mentioned in the previous slide. So the overall goal is to shorten the time from the date of complete supplement submission to the action date in order to allow earlier patient access to therapies that address an unmet medical need. So that can be kind of confusing, and so I want to show you this graphic. Hopefully it'll help tell the story of the process I was describing earlier. So this slide compares STAR to a traditional application, and I'm going to go into more detail with subsequent slides. And so on this graphic, though, you'll notice that the blue bar, which is over here, this is the priority review bar for a typical efficacy supplement. And above that blue bar, you can see the red bar indicating the STAR review. So that's right over here. But you'll notice that it's shifted to the left. And you may have also noticed the other key components within the STAR overview timeline that are different from a normal supplement review. So, for instance, you may have noticed on this graphic that a STAR entry request meeting will need to be submitted in order for FDA to determine whether it's accepted into the program. So here is a STAR entry request. You can also see here that we receive the supplement as a split submission in 2 parts. So this is part 1 submission and part 2 submission. So once we receive part 1 and part 2, then we have a target action date at least 1 month in advance of the PDUFA goal date. So over here we have the STAR target action date, and this is the PDUFA goal date. And we'll go into more detail with subsequent slides, but as you can see here, FDA will begin application review upon receipt of part 1 of that submission. And part 1 really contains the entire application, everything we need to start the review, and the only things that are missing are those 3 components, which are the clinical study report and the 2 integrated summaries for safety and effectiveness, which I mentioned a moment ago. And those are often delayed on industry side as they go through levels of clearance. So part 2 then is those additional 3 components, again, specifically the clinical study report and the 2 integrated summaries. When we get the part 2 submission, that will complete the supplement, and that's when the PDUFA clock starts, which is shown here. And as noted in this graphic, you can see that FDA begins the application review with part 1, which again is right over here. By the time we receive those last 3 components, it should hopefully enable FDA to take an earlier action when timing it by the PDUFA clock. So it's kind of a win-win situation where FDA still gets the full time to review, but then at the end of the day, we're hoping that we can get these treatments for unmet medical needs out to patients earlier. So the first component is a STAR entry request that gets submitted to the FDA and is reviewed and held either as a standalone TCON, where we simply just speak with the applicant about whether their supplement meets the criteria to get into the STAR program. Or if they wanted to discuss content and format of their anticipated supplement, then the applicant would submit a Type B pre-submission meeting, just like they would now, and we would talk about STAR within the context of that Type B meeting, along with their other questions such as what would be submitted to the application. And please also note that if the STAR entry request is submitted as a standalone TCON, then there are no meeting minutes. But a formal letter is sent to document the decision. So we encourage an applicant to use option 2 only if questions related to what would be submitted in the application, such as Part 1 versus Part 2, will be asked. And in terms of the content of the request itself for the STAR entry request, the sponsors will submit the top-line results from their pivotal trials, and they'll also submit their proposed labeling, and then also an explanation of how the supplemental application meets the STAR criteria. And so please note that the acceptance into STAR does not guarantee an application approval. The application must still meet the applicable standard for an approval. So I mentioned that there is criteria for getting into the STAR program, and we'll be going over the 4 criteria. And as you look at the slide, you'll probably notice that the first 2 criteria look somewhat familiar, and that was intentional. So the first criteria states that clinical evidence from adequate and well-controlled investigations indicate that the drug may demonstrate substantial improvement on a clinically relevant endpoint over available therapies. And the second criteria states that the application is for a drug intended to treat a serious condition with an unmet medical need. So these are the same 2 criteria that we use in the Breakthrough Therapy Designation program. So the caveat, though, for STAR is that the application does not have to actually receive a formal Breakthrough Therapy Designation. And for other products and other centers, they won't actually have to have received other designations, such as the Regenerative Medicine Advanced Therapy or RMAT designation, but they do have to meet the same standards that's outlined here within criteria 1 and criteria 2. And so the last 2 criteria 3 and 4, they're really about timing and some aspects of the complexity of the supplement. So number 3 just states that the supplement can't have any component to it that would require a longer review time. And an example here would be a risk evaluation and mitigation strategy, or a REMS, because it adds a complexity to an application. And then the last component on here, criteria number 4, it just points out that if there is a need to inspect a foreign manufacturing site, a supplement would also not qualify just due to the logistics of scheduling those inspections and getting them back. However, please note here that domestic site inspections may be allowed if it doesn't affect the expedited time frame. Also, please note here that STAR is limited to priority efficacy supplements, so a supplement must really qualify to be a priority review in order to get into the STAR program. Okay, so assuming an application gets into the STAR program, the applicant would submit their submission in 2 parts, as I mentioned earlier, and Part 1 here is the entire application, everything needed to review, with the exception of those 3 components that take a little bit longer on the sponsor side, which are namely the clinical study report, the integrated summary of effectiveness, and the integrated summary of safety. And again, the review data begins at the receipt of Part 1. However, the PDUFA clock doesn't start until Part 2. So please also note that you may also contact the RPM for any questions, including questions regarding eCTD submissions. And in addition to the items mentioned in the earlier slide, we have some items here that we wanted to call to your attention that we've also asked to be included in Part 1 as well, because these items are often submitted as part of the clinical study report. However, they are items that are ready earlier and that can be submitted with Part 1. So Part 1 should also include the tables, figures, and listings, as well as protocols and amendments for pivotal trials, the statistical analysis for pivotal trials, and the sponsor's high-level assessment summary of safety and efficacy results, as well as the death summaries. So, if FDA identifies substantive missing information, reviews will stop, and a revocation of STAR status letter may be issued. And please note that applicants who submit an incomplete Part 1, they can still resubmit a complete supplement at a later time, but it just will no longer be reviewed under the STAR program. So for Part 2, please note that it should be received no more than 3 months later and should include the remaining components of the supplement as listed here: the CSR, or Clinical Study Report, the ISE, and the ISS. And as mentioned earlier, the PDUFA clock starts upon receipt of the Part 2 submission. And again, it should be received no later than 3 months after the Part 1 submission. For Part 2, if the FDA identifies substantive missing information, then the reviews will stop, and a revocation of STAR status letter may be issued, and at that point, the application is treated like any other complete application and receive a refuse to file or an RTF. So, to summarize, for a STAR review, the review begins with the receipt of the Part 1 submission. The PDUFA clock starts when FDA receives the Part 2 submission, and then FDA will target an expedited review, which means an action at least 1 month before the PDUFA goal date. And so here we have a snapshot of the FDA STAR website, which launched again in October of 2022. It's another resource that goes into the overview, the eligibility criteria, any process considerations, as well as any additional clarifications. So please feel free to visit this website if you're looking for information for STAR. It is a great resource. So going forward, we'll be conducting an assessment of the STAR program, and the interim assessment will be conducted by the end of fiscal year 2025. And a public workshop will be conducted by the end of quarter 2 in fiscal year of 2026. And then please note here that the outputs from the assessment and workshop will be published in a publicly available report. So we have a few challenge questions to go over. And here is my first challenge question for all of you. When does FDA begin their review of a STAR application? Is it A, once the STAR program acceptance letter is issued? Or B, upon receipt of the STAR entry request? Or is it C, upon receipt of the STAR Part 1 submission? Or D, upon receipt of STAR Part 2 submission? Take a moment to see which answer you think is correct. And hopefully all of you selected answer C. The FDA will begin their review of a STAR application upon receipt of the STAR Part 1 submission. And again, Part 1 does contain the entire application except for the clinical study report, the integrated summary of effectiveness, as well as the integrated summary of safety. So for challenge question number 2, the question for all of you is, when does the PDUFA clock start for a STAR application? Is it A, once the STAR program acceptance letter is issued? Or B, upon receipt of the STAR entry request? Or is it C, upon receipt of the STAR Part 1 submission? Or D, upon receipt of the STAR Part 2 submission? And again, take a moment to see which answer you think is correct. And hopefully all of you selected answer D. The PDUFA clock starts for a STAR application upon receipt of the STAR Part 2 submission. So for challenge question number 3, the question is, when is the integrated summary of safety submitted? Is it A, with the STAR entry request, or B, with the STAR Part 1 submission, or is it C, with the Part 2 submission, or D, 120 days after the complete supplement? And again, take a moment to see which answer you think is correct. And the answer here is C. The integrated summary of safety is submitted with the Part 2 submission. So these are some additional resources, which includes the link to the PDUFA commitment letter, as well as the FDA's STAR website, which I encourage you to visit for more information on the STAR program. And so to summarize, STAR's goal is to shorten the time from the date of complete submission to the action date to allow earlier patient access to therapies that address an unmet medical need. The application is submitted in 2 parts to enable earlier review and action. And also, STAR is only limited to priority efficacy supplements that meet a specific criteria. So again, this is just an overview of the STAR program. We went over all of the different parts. We now have gone through the process, including the STAR entry request. We discussed steps to get into the program. We discussed the application review and the submission Part 1 and Part 2, and the STAR target action date. So this is just a quick visual again of what we discussed. And if you have any questions, please feel free to reach out. Again, my name is LaShawn Schnupp, and I'm the CEDAR Program Manager for the STAR Pilot Program. And as a closing thought, please remember to include all components required for each of the 2-part submissions to utilize the benefits of the STAR Pilot Program. And that concludes my presentation. I'll be back shortly to answer any questions. Thank you, everyone.
Hello, my name is Dr. Lolita Surratt, and I'm from the Division of Medication Error Prevention and Analysis within OSC as part of CEDAR and FDA. And today I'm going to talk to you about the use-related risk analysis and human factors protocol reviews and what to submit for an efficient review. First, let's talk about our learning objectives. Define the objective of a use-related risk analysis. Define the objective of a human factors validation study protocol. Describe the biosimilar user fee amendments PDUFA 3, prescription drug user fee amendments PDUFA 7 commitments related to human factors submissions. Describe common agency-initiated information requests for HF submissions. Identify tips which may result in a more efficient agency review of URA and human factors validation study protocol submissions. And lastly, discuss the importance of the early identification of human factor data needs for medical product development. A little bit about the division I work for, DMEPA. DMEPA resides within the super office called the Office of Surveillance and Epidemiology. OSC monitors and evaluates the safety profiles of drugs available to American consumers using a variety of tools and disciplines throughout the life cycle of drugs. OSC has 4 core functions: pharmacovigilance, pharmacoepidemiology, medication error prevention and analysis, and risk management. CEDAR is further divided into 2 offices and 5 divisions. Under OMEPRIM is where DMEPA lies, and DMEPA is highlighted here in green. Now let's talk a little bit about DMEPA's mission. Our mission is to increase the safe use of drug products by minimizing use error that is related to naming, labeling, packaging, or design of drug products. To achieve this mission, DMEPA is involved in all of the following: conducting a safety assessment of proprietary names for drug name confusion that may lead to wrong drug errors, and we serve as signatory for these name reviews. We are included in the review of labels, labeling, and packaging, and product design to promote safe use of medications. We review post-marketing error reports to identify safety signals and take action if needed. And we participate in the development of guidance for industry and FDA staff, work groups, and advisory committee meetings. Lastly, we review human factor studies and data to optimize product design and minimize the risk for use error that may lead to medication error. But who looks at medication errors? Safety evaluators. Within DMEPA, which was created in 1999, we're composed of healthcare professionals, scientists, and engineers with very professional backgrounds. We're further divided by therapeutic categories into DMEPA 1 and DMEPA 2. We lead CDER's review pertaining to medication error prevention and analysis and human factors for drug and therapeutic biologics. So we just said that DMEPA leads CDER in the review of medication error and the review of human factors evaluations for drugs and therapeutic biologics. But how do we do that? A key component of the evaluation of medication errors for combination products or complex drug products is to evaluate the submission for the need for human factors data. Keep in mind that we do not limit our focus on combination products. There may be human factors considerations or certain instances where we have complex instructions or novel packaging of drug products that are not considered combination drug products, but still their complexity may arise to a level of needing additional data to ensure safe use. Some of you may be familiar with our practices that we use to apply best practices to decrease vulnerability to medication error in labeling and naming of products. But how do we evaluate medication errors from a human factors perspective? It's important to note that we do not work alone. We consult with our experts within the agency to help us with those evaluations. So let's walk through the process of how DMEPA engages other offices within the agency to perform human factors evaluations. So after receiving a submission for review, DMEPA will determine if that submission is such that we need to consult other offices. At that time, we may issue an inter-center consult to a team such as CDRH and their human factors team when input is needed for certain device-heavy products. For example, we consult the CDRH human factors team for things like external infusion pumps or an on-body infuser, or maybe a software-driven medication dispenser or software-driven device. In those instances, we rely heavily on CDRH colleagues to provide their expertise in the device-heavy component of the proposed product. We use their conclusions about the device to help determine what and how those may weigh in on medication errors in the intended use of the proposed product. Depending on the intended user of the product, DMEPA may also consult the patient labeling team for review of the IFU to ensure that the language is patient-friendly. Let's say a particular combination product's IFU contains specific instructions for the lay user to administer the product in a home setting. In this instance, we consult PLT and work with them in the review of the IFU or quick reference guide to gather their input on how to best provide clear instruction for the lay person. After we get those consults back, we'll refer to them within our review and also use their input to help us determine what the data is needed for human factors considerations. Now, there are several different types of human factors, and let's define the types of human factor submissions that we'll discuss today. First, use-related risk analysis. The use-related risk analysis is a risk management tool that supports the entire human factors engineering process and should be utilized as part of an overall risk management framework. The URA informs the human factors validation study design, testing, and evaluation of a medical product. A use-related risk analysis is important to help identify use-related hazards associated with the combination product, as well as to characterize risks so they can be mitigated or eliminated through improved product user interface design. As a reminder, conducting a use-related risk analysis is an iterative process. As part of the product development and testing of the product prior to agency review, there may be times during the product development when additional use risks or mitigations are identified. This could be identified as part of formative work or information gathered as part of clinical trials, but as a result, there may be additional changes to the user interface required to address use risk or to test mitigations. In these cases, it is a good practice to document and continuously update the URA to reflect that these iterations for a comprehensive record of use risk and how those risks are mitigated. Essentially, the URA does help characterize use-related risks, and it's the risks that we're trying to characterize and understand in order to then consider if further risk mitigation strategies are needed. Now that we've identified the risks in our use-related risk analysis, how do we use that use-related risk analysis to determine or even design human factor data needs for the proposed product? First, based on the findings of the URA, human factors data has been determined that it may be needed. So, the risk analysis may have identified use risks that need further mitigations in this case. If it is unclear if that risk mitigation proposed will address the risk, you may need to provide data from a human factors validation study to support that your mitigations are sufficient. The risk analysis can inform the design of your protocol. As part of your human factors protocol design, the risk analysis should be used to ensure that you've addressed all of the steps in the use of your product and include any use scenarios to study and address any possible use risks. The submission of a human factors validation study protocol for agency review is not required, but it is encouraged to ensure you that your methodology is appropriate to achieve the study's objective. By getting input from the FDA early on your protocol, that may help decrease pain points later on in your development process, especially after an application has been submitted. So, for example, you did not have any prior agency feedback on your protocol or your product development plans before you submitted your application for your NDA or BLA. You run the risk of the agency deeming that you need to submit human factors data as part of your marketing application, or if you've already completed your study without agency feedback on the protocol, we may then deem that your methodology is flawed and not accept the results. As a result, we may require a new study from the application, new study to support the application that has already been submitted. So these things will definitely eat into your timeline. So you've determined and we've agreed that data from a human factors data validation study is needed to support your marketing application. We reviewed your protocol, which I like to repeat and reiterate. A protocol review is optional but encouraged. And after these steps have been completed, the next step is to actually run the study. And what is the goal of a human factors validation study? A study which is conducted to demonstrate that the final finished combination product user interface can be used by the intended user without serious use errors or problems for the product's intended uses and under the expected use conditions. The study should demonstrate that use-related hazards for the final finished product has been eliminated or that the mitigation for residual risk are acceptable. These are the types of things we look for in the study. Of note, we generally find it feasible to conduct these studies under simulated use conditions, but there are rare occasions where we would need to see that study in actual use. When talking about study results, making errors is not always a bad thing because it is likely that use errors will occur in every study. Our focus, though, will be to learn from those errors to ensure that we are finding what mitigations are required and if we find the residual risk is acceptable. These are things that can be discussed in the Human Factors Validation Study Report. The study participants are representative of the intended users and study conditions, and the expected use conditions is one key component that we would like to see in your study. In the past slides, we talked about human factors and we talked about drug products, and we mentioned combination drug products. But what is a combination drug product? The formal definition can be found in 21 CFR 3.2, which reads, therapeutic and diagnostic products which combine more than one, either a drug, device, or biologic product. A combination product can combine drugs, devices, or biologics. They can be physically combined, for example, drug inside of an autoinjector, or chemically combined, such as a pill which is embedded with a sensor. Combination products can be co-packaged in a kit. For example, a drug vial, which is co-packaged with a syringe for administration or measurement. Or it can be separate cross-labeled products. For example, a drug label, which indicates to use the drug with a specific device, like a nebulized drug, which is to be used with a 510K cleared nebulizer. And here are some examples of combination drug products. IV infusion bags, which are pre-filled syringes, pen injectors or auto-injectors, pharmaceutical aerosol delivery devices or inhalation products, transdermal delivery systems or patches, drug infusion devices, kits containing drug and administration devices. And because of the combination products may include drugs, biologics, devices, then you can imagine there are different regulations that we must adhere to. This is where it becomes important that we reach out to our agency experts in different areas, such as devices or biologics, to ensure that we provide accurate information about human factors needs without causing conflict. This is truly a group effort. As we discuss combination products, it's important to note that there are instances when we may ask a company to apply human factors considerations for products which are not combination. For example, birth control pills in a blister with a novel packaging design or a unique shape with blister labeling, which is unique enough that there could be a medication error concern of confusion for the intended user to determine where in that packaging to start and where in that blister to stop. If taken out of sequence or just taken incorrectly, this use error could result in pregnancy. So, in this case, we may ask the company to provide and evaluate the risk in the use of this product and this product design to be considered. It may stop there, or depending on what the risks are that have been identified, it may go further to the point where proposed mitigations are not enough, and we need to see data from a simulated use human factors validation study. This is just an example, but it definitely could happen with novel packaging or novel unique design. In addition, we need to consider a new user group. New user groups could be an instance where a standalone vial presentation, and when I say standalone, I mean it is not co-packaged with syringes. It's just a vial itself. So that standalone vial is proposed to a new patient or caregiver user group who traditionally has not had exposure to withdrawing, preparing, or administering a dose from a vial presentation. Perhaps this group is accustomed to using an auto-injector or pre-filled syringe, something where it's already measured up and they just need to administer it. So in that case, we're comfortable with the administration steps, and we feel that those users will be aware of that. But for the task of preparing the vial, measuring a dose from the vial, those things will be new to this user group. In this case, we may not have enough data to assure us that the user will be able to perform these tasks as intended. we would advise the company to provide additional data to justify that this user would be able to understand and perform these new use tasks as intended. Additional data doesn't always mean study data. It could mean literature search, anything to justify your position that these intended users will be able to use this product as intended. What about complex instructions? This is another instance where it may not always be apparent that users will be able to prepare, measure, and administer the product as intended. This could be a unique product where, again, a standalone vial could be stored at room temperature, but once you combine that vial, which has lyophilized powder, once you combine that vial with the diluent, the vial must then be placed on a chiller before administration to activate the drug product. And once chilled, you have a limited amount of time to draw out the dose before it's no longer stable. A use error in any one of those steps could potentially lead to patient harm. So you see, these particular human factors considerations should be considered in other products, not just combination products, because they may have a use risk that causes a concern for a vulnerability to use error based on the design of the user interface or the design of the product characteristics. In these cases, it may not be so clear to the sponsor what human factor needs are required. This is why it's important to reach out to the agency early to help in that determination. You may be thinking, why is human factors data even a consideration? Let me walk you through the regulatory history for devices and briefly explain how we got here and where FDA's regulatory authority to consider human factor studies stem from. We can start with a device regulation 21 CFR 820.30, which is a regulation which in general requires each company to use quality control methods to establish and maintain procedures to control the design of the device in order to ensure that specified design requirements are met. The goal here is really to ensure that safe and effective use by implementing quality controls for the proposed device. For combination products, we may not always ask for data, but we always remind companies that according to this particular CFR, you have the obligation to ensure good manufacturing quality control methods have been used in the development of your product. Also, a drug regulation comes from the Food, Drug, and Cosmetic Act, specifically the Kefauver-Harris Amendment to the 1938 Food, Drug, and Cosmetic Act, which is requirement to show that a drug is effective. Now, if we put the device regulation together for good quality, along with the amendment to the Food, Drug, and Cosmetic Act for efficacy, we can further say that based on this information, human factor studies may be needed to be submitted for agency review to demonstrate minimization of elimination of use-related hazards and medication errors. The key words are may be needed. So we don't always ask for human factor studies data to be submitted to support the application. However, we do ask that you use tools to evaluate use risks and to subsequently determine if any additional human factor data is needed to be submitted for agency review to justify safety and efficacy of any proposed product. And these tools help in making that determination. You may also be thinking, when do I need to begin my considerations for human factors considerations? At what phase in your development? This slide shows drug development process and where human factors and DMEPA involvement fit in. So DMEPA usually begins within the IND, once the IND is filed, but sometimes it can be as early as pre-IND phase. Human factors work including product design, preliminary analysis, formative work, and validation testing can begin during pre-IND and can continue through phase 4, which is post-marketing. Updates to the use-related risk analysis are made continually throughout the drug development process as risk is continually assessed and reassessed. For example, if a company finds errors during the formative study and subsequently the design is modified for the product, then the URA needs to be updated to reflect that modification. That revision needs to be assessed for risk. The same can apply for human factors validation study. If the sponsor identifies any failures or risks that could lead to patient harm, that particular failure needs to be assessed and mitigations need to be proposed and implemented. Those need to be updated as part of the URA. Keeping in mind that after each revision to the user interface or product design, the sponsor also needs to determine whether or not, after updating the risk analysis, does that mitigation require an additional validation study, or if the revisions are such that it doesn't impact usability, there's no additional studies needed. That justification will be added to the human factors validation study report for our alignment. So continual updates to the risk analysis are key. Continually evaluating risk is important. So when we talk about user interface changes, please understand that the user interface is not only limited to the device. Anything that the intended user interacts with is the user device, its user interface. So that includes all points of interaction between the product and the users, including things like a display, a control, packaging, product labels, instructions for use, etc. Thus, knowing this definition of user interface helps us to understand why the term user interface is not limited to only combination drug products. It could be relevant for all drug products. Now that I've gone over some background information on human factors and some key terms, let's talk about the latest BsUFA and PDUFA commitments as it relates to human factors. The first commitment I'd like to talk about pertains to performance goals for human factors protocols submitted for biologics. So beginning in fiscal year 2023, which is October 1st, coming up 2022, we agree to review and provide the sponsor with written comments for 90% of filed human factors validation protocol submissions within 60 days of receipt of that protocol submission for biologics. What this means is that in addition to the already human factors validation study protocols that we currently review for drugs, we will now add the review of those biologic human factors protocols. It is likely that the number of human factors protocol submissions to the agency may increase with this agreement to reach 90% performance goal for all those protocols submitted to the agency. So as you can imagine, we really have our work cut out for us here with these protocol submissions. Another commitment which is related to human factors is about URA submissions. In summary, we have agreed to a staged implementation of the review for use-related risk analysis in PDUFA VII and bioinformatics III. We agreed to review and notify sponsors of agreement or non-agreement with comments within 60 days of receipt for 50% of filed submissions beginning in fiscal year 2024. After that, we'll agree to 70% of filed submissions within 60 days will be reviewed. And then lastly, in fiscal year 26-27, we agreed to notify sponsors within 60 days of receipt for 90% of filed human factors use-related risk analysis submissions. So in the past, although we have provided a review and written recommendations for use-related risk analysis submissions, we've not had a commitment for the review timelines of URR submission by a certain time frame. With this implementation of PDUFA VII and bioinformatics III, along with the goals I just talked about, the number of URRA submissions to the agency we expect to increase as of October 1st. So the last commitment to discuss here today relates to a new guidance. With the increase in URRA submissions, we would like to ensure that the submissions are comprehensive and complete. So by the end of fiscal year 2024, FDA will publish a new draft guidance for review staff and industry describing considerations related to drug device, biologic device, and drug products on the topics, such as guidance that will convey FDA's current thinking regarding how a URA, along with other information, can be used to inform when the results from an HF validation study may need to be submitted to a marketing application. This guidance will also provide a comprehensive, systematic, and stepwise approach with examples, when applicable, to illustrate how to make this determination. This is important because if you recall in my last slide, I said that we have never had any commitment as far as an enforceable review clock as far as URA submissions are concerned. So we were in the past able to provide a thorough and detailed review of URAs that were submitted to the agency in a reasonable time frame as possible. However, with the new implementation of URA goal dates, we are required to meet the review of URA in 60 days as of October 1st. So we have had some time and experience with these reviews over the past few years, so we are familiar with the review process. However, to ensure that we are providing a good and comprehensive yet timely service in the review of these URA submissions, we need to be able to provide the tools and guidance to industry, enable for you guys to be able to follow for a complete submission. And this guidance will eliminate a lot of back and forth on what we need to perform our review. And just to note, this guidance is currently under penmanship, but it's not quite yet published. Okay, so now we've gone over some background materials and also discussed some new commitments. In light of these new commitments, how can we cut back on the amount of time we spend gathering necessary information or lacking information to perform an efficient agency review? I'll go over a few times when we could not complete our review or the review of human factors data was delayed due to a lack of clarity or missing information in the human factors submission. I'll also give you some tips on how to avoid these issues and lead to a more efficient and timely review. Oftentimes, when we're in a position where we cannot perform a comprehensive review at the agency level, it's because of confusion or uncertainty regarding human factors data needs. The company who submitted the submission may not be aware of or may be unsure of what the human factors requirements are. So how does one find out what human factor data needs are required? We have guidances which are available, and references are included at the end of this presentation to refer you to those guidances. But the first step would be to refer to the guidance which the agency has developed to help outline the process. Although we aim to write our guidance as basic as possible, sometimes there may be situations which are unique or novel, and those basic guidances may not provide exactly what a company needs to formulate their submission. This may lead to more questions, not specifically addressed in the guidance because they're more custom questions. Remember, a guidance is a suggested guide, which should cover the most routine situations. It may not always meet your needs as the only resource if you have any unique or novel circumstances. In that situation, early interaction with the agency to ensure that you've prepared for and consider if any human factor data needs exists for your proposed product. This will help and lead to an early determination that your product is a combination product or has been identified as a product which requires additional studies based on novel or unique design. In this case, it's determined that data from a human factors validation study is needed to support your application. We have performed a review of the protocol for the company. We provided written comments, and now the company is back and they submitted a response to previous human factors advice, but there are not enough details provided or maybe no questions submitted to help us determine what exactly is needed in this submission. Thus, we're left to figure out what is needed. What are we reviewing? To avoid this delay, we have to ask questions. We have to send an IR. Do you need clarity on recommendations previously made? Did you implement all of our recommendations? Did you disagree with any of our recommendations? And because of this, we need to wait for your response. And it's important to provide clarity on the intent of your submission is the key takeaway for this slide. If we don't know what actions you want us to perform, we may spend time combing through the submission, trying to figure out exactly what is our task at hand. We'll then need to send an IR asking these clarifying questions that I just read below in this slide. And after we have these answers, then and only then are we able to proceed with our review. However, the back and forth with the IRs will eat away at your timeline. So what can you do to preserve this timeline? We think that the use of the cover letter is a good idea. Use the cover letter to give us a quick summary of what you need from the agency. Communicate your intent that way. For example, you may say, we have aligned with most of the agency's recommendations and seek clarification on recommendations 4 and 7 as part of the cover letter. And then this is just a suggestion, but it helps us keep everything in one spot. But to help clarify, we'd like you to format your submission of those questions using the same table presentation that the agency initially sent, just adding an additional column with your concerns over to the right. Again, this is not a requirement, but using this particular format helps us understand what was the identified concern, what was the agency recommendation, and what does the sponsor have a concern with in response to that recommendation. A third example of an issue we've encountered here in the human factors submission is we have received a revised human factors protocol, but we don't know why that revised protocol was submitted. What is the intent again? There may be instances when you want the agency to provide another review of your revised human factors protocol. This could be when we previously reviewed the protocol and provided recommendations, but the revisions that were implemented were significant enough that it warrants a new and complete review of the protocol once again. This could be a case where the user groups have changed or the device design has changed significantly enough that it required additional tasks, or even the company who initially proposed the product has changed hands and they want to go in a different direction. These things could warrant a totally new review. Alternatively, there may be instances where the intent is just to submit your revised protocol for documentation's sake. So, do you align with all our recommendations that were given, but you're just submitting a revised protocol for the record? We've seen this in certain instances where we've already performed a review of your protocol, we provided recommendations, and we got that second submission without detail. We're not sure if you want another review or if you want just to document for record. So both of these situations would result in an IR being sent out to clarify the intent of the submission. Again, those IRs will take time to be once cleared from the agency, then sent from the agency, then we have to wait a response. It could take a week or more to get this particular issue rectified. So what can be done? Again, we suggest clarity in the cover letter. It's a very quick way to let us know that you are simply submitting the revised protocol for the record, or you truly do request an additional review of the revised protocol. So here's a few two example languages that we've seen in the cover letter. It may say, we have aligned with the agency's recommendations and are submitting the final human factors protocol for the record. Or we may have had something similar to, we've determined that our user groups are no longer representative. This revised human factors protocol provides a new methodology as well as new user groups. And this is just one point of clarification I'd like to make. I want to make sure it's clear that adding a summary sentence or two to the cover letter does not mean we do not need the details in the submission. We still need that detailed information about the revision that you made to your protocol in order to make our determination on the acceptability of the new components of your submission. So providing those comprehensive details within the submissions are still warranted. Along the same lines as the previous slide, this is in situation number 4, when we may get a revised protocol submitted for agency review, and there's a quick summary in the cover letter that tells us exactly what's changed and that the changes were significant enough to warrant another review. Our first thought is, well, what are those changes that we need to look at? So, for example, what revisions were made to the methodology? What are the revisions to the moderator script, if any? Are there any changes to the IFU? And do any of these changes require updates to the URA? These are 4 questions that may impact our previous review and also may impact our forthcoming recommendations. So these questions do not have to be answered in the cover letter, but they need to be considered and included within your submission. It would be beneficial if we knew what those changes are before and after, so that we can provide a more comprehensive review. One helpful approach to provide with your submission in this case is when you have submitted a revised protocol for review, provide us with a side-by-side or a red line version to identify those changes. This way we can easily pinpoint the revisions and then make a quicker determination on what has changed and how that change may impact our previous determinations. This is in addition to a clean version of your human factors protocol within your submission. So this approach may be helpful to ensure that none of the new or revised information impacts our previous recommendations. The annotation in your submission of what has changed will lead to a more efficient review, combined with a high-level summary in your cover letter that, yes, we do need a review for this particular reason. So in example number 5, there are times when we have human factors protocol submissions and we find that the URA is incomplete or missing. If there's no URA, then that's problematic because as I spoke earlier, I stated that the URA is the backbone for the human factors protocol submission. Sometimes we get a high-level URA, or maybe only a few select critical tasks have been evaluated. But without a comprehensive URA of all steps in use of the product, we will not know the task criticality, or even if the use scenarios that are selected are needed to be included in the protocol. In these instances, we'll be forced to send an IR asking for a detailed list of exactly what's needed. We cannot move forward without this particular missing information because, like I said before, the URA is very critical to us evaluating the methodology of the protocol. So in the letter that you receive from us, we'll include language, something similar to, until we have this URA information submitted to us, we will be unable to proceed with our review. So what do you do to prevent this from happening? Well, guidances are available. We do have one guidance, the content of a complete submission for threshold analysis and human factor submissions to drug and biologic applications, which is still in draft, but it does offer some guidance for what to expect to be included in a human factors protocol submission. And because the risk analysis is the background of the protocol submission, it's very important that that risk analysis is very detailed and it has evaluated all steps in the use of the product, providing things like task criticality, proposed mitigations, things that you see here at the table as table headers. If we do not have all this information in the use-related risk analysis, we'll send that detailed IR out again. The guidance is there to help you to avoid us with the IR and the back and forth. So human factors protocol submissions, issue number 6, and the user interface. These are examples where when we will send a letter to you asking for missing information about things like description of intended product uses, use environments, intended dosing, training. These are things we need in order to make sure that all steps in the use of the product have been considered. We need graphical depictions of labels and labeling, things that help us to see what the intended user will see and how they will interact, and how we need to have user in scenarios built around them. If there's no IFU, we need that IFU in order to go through the steps and use of the product. We don't have an IFU, then we don't know if all the risks have been involved because we don't know how the product is intended to be used. So we need all of these user interface pieces in order for us to go forward with our review. So what can we do? In this case, again, refer to that same guidance, contents of a complete submission for threshold analysis and human factor submission for drug and biologic applications. And without this detailed information, I think I made it clear that we cannot move forward, and it limits our ability to opine on the acceptability of your protocol methodology or if all the risks have been considered. So additional information of what we consider a complete submission is included in that draft guidance, and I encourage you to use it as a resource. Challenge question number 1. When determining if your proposed product may need human factors data, you should include a narrative of your independent determination once you submit your NDA or BLA. Reach out to discuss with the agency as early as possible before you submit your NDA or BLA. Leave the box on the 356H form unchecked, and someone will reach out to you to follow up. Or the FDA does not allow the approval of combination products. And the correct answer would be B. Reach out to us as early as possible before you submit your NDA. I think I've noted several times here that early interaction is key. Challenge question number 2. Which of the following statement is true? Only combination products require submission of human factors validation data to support safe use. If you are approved for an accelerated approval, like fast track, your proposed product is exempt from the submission of human factors data. It is safe to say that a product proposed in a vial presentation for self-administration will never require human factors data. Or D, a use-related risk analysis is important to help identify use-related hazards associated with combination products, as well as to characterize risks so they can be mitigated or eliminated through improved product interface design. So the answer is D. We need to know, the use-related risk analysis is the backbone. So we need to make sure we have that in order to determine any type of risks in the use of the product and how that would impact your product interface design. So in summary, I'd like to thank you all for your attention. But in summary, here we have learned about or heard about the implementation of PDUFA VII and PDUFA III and the number of protocol submissions to agency, which we think may increase. And to ensure the timely and efficient review, it is imperative that each human factors submission is a complete submission. And we recommend you come to us early to seek advice on your human factors development plan and to determine if we see any indication to encourage you submit. We encourage you to submit your human factors validation study protocol for agency review. So based on this information and the concerns, we want to ensure that your submissions that you send us in are in good shape and for provide an efficient review. In closing, my hope is that you're able to use these tips in such a manner that they will contribute to the efficient review of your proposed product. So we'd like you to consider your call to action is to review the information presented today and your future submissions to help to ensure a complete and timely review. This is a slide of resources. And now I'd like to open up for questions.
Thank you both for those excellent presentations. We will now begin our Q&A panel session for these two presentations. And we'll also be joined by J. Paul Phillips, Director of the Office of Program Operations in CDER's Office of New Drugs. And if you have not yet had a chance to enter your questions into the Q&A chat pod, please do so now. We will answer as many questions as time allows. So we do have some questions that have come in. Our first question is for Dr. Schnupp. Dr. Schnupp, what is the difference between STAR and RTOR?
Good question. So STAR is a PDUFA program open to development programs from any therapeutic area, whereas RTOR or the Real-Time Oncology Review is not a PDUFA program, and it's only available for oncology development programs. And if you have more questions about RTOR, oncology product sponsors should contact the RPM in the Office of Oncologic Diseases, or OOD, their division seeing their application. Thank you.
Thank you, Dr. Schnupp. Next question. If an ISE/ISS is not needed for a supplemental NDA per agreement with the review division, is the STAR program then not appropriate?
This is another good question. So in that case, benefits of the timeline for the program would not be realized since there couldn't be a Part 2 submission. So it would not be a good fit for the STAR program if you do not plan to have an ISS or an ISE. Thank you.
Thank you, Dr. Schnupp. Let's do one more question for you. What if you failed to submit Part 2 within 3 months of the Part 1 submission? Would the STAR submission then be canceled?
Good question. Thank you. Yes, the application would be removed from the STAR program. Thank you.
Thank you, Dr. Schnupp. Let's move on to Dr. Starrett. So your first question is, how would sponsors know when to submit a use-related risk analysis for a product? Not all products would require a URA, correct?
Yes, thank you for that question. It's a great question, and it is one that we get quite often. It is correct that a risk analysis will measure risks of the use for your proposed product, but not all products require a use-related risk analysis. One of the clear-cut ways is to know when you need to submit a risk analysis is considering when you have a combination drug product. For those, a use-related risk analysis should always be developed to examine what the risks are in the use of that product to make sure that you've addressed all the potential errors in the use of that combination drug product. If your risk analysis doesn't identify any new or unique risks in the use of that product, then we would still need to see your risk analysis along with the justification that you have determined that no new data is needed because there's no new or unique risks that have been identified in the use of your product. So that is kind of a clear-cut way. Everyone is pretty clear on combination drug products. But when it gets tricky is when you don't have a combination drug product. This could be yes or no. You may or may not need to submit a use-related risk analysis. And when you don't have a combination drug product, you still may have risk in use of your product. So we still need to see a risk analysis in cases of something like novel packaging, or if there's a new identified user group for an already established product, or if there are complex instructions that are needed in order to safely administer or measure or prepare your proposed product. So this gets to be tricky because these are subjective opinions of what is novel packaging. what is complex instructions. So in that case, we like for you to reach out to us early to, just to get a peek at what you're proposing. And in the times when we have determined that your particular product could be prone to use error or could be confused and lead to medication error leading to patient harm, this is when we would advise you at that time that it would be a good idea to provide or complete a comprehensive use-related risk analysis to determine that no risk would be presented to the intended users of your product. So that was a long way to say, I guess it depends, but you do not need to always submit a use-related risk analysis. There are times when it's clear-cut and there are times when it's not. But in any event, reaching out early to the agency to have a discussion on your product development would be beneficial. Thank you.
Thank you, Dr. Starrett, for that very informative response. We do have another question on use-related risk analysis. So when a sponsor submits a human factors validation protocol for FDA review, do they also need to submit the use-related risk analysis? Do you review that as well? And when should this protocol be submitted for FDA review?
Thank you for your question. That's a great question. So we have a draft guidance actually, that is titled Contents of a Complete Submission. And within that guidance, it provides details of what you should include with each type of submission. Now, we don't go into details on how to evaluate the human factors submissions there in this guidance, but we do give you kind of like a checklist of what to include with each particular type of human factors submission. So for a human factors protocol, yes, we do need a use-related risk analysis to be submitted as part of a human factors protocol submission. As mentioned earlier, we know that a use-related risk analysis is considered to be the backbone of your human factors development process, because it examines the risks and help us to identify what particular use scenarios need to be evaluated in your protocol. If there are any use steps that are identified as problematic, then those use steps could be focused on as critical in your evaluation and your human factors validation study. So yes, we do need the use-related risk analysis. We will review it. As part of our protocol review, one of the key steps is to review the use-related risk analysis in combination with the instructions for use to make sure that every step that is in your IFU is also included in your use-related risk analysis. And once those steps have been determined, that all steps have been evaluated and considered, then and only then can we give the comprehensive review that your human factors protocol methodology as far as the steps is complete. And I think the last question is, when do you need to submit your human factors protocol? So just to reiterate, the submission of a human factors protocol is not required, but it is recommended. And we do recommend that you send us your protocol before you do your study. So before you run your study. And we ask that the human factors protocol be submitted as part of the IND phase. Just keeping in mind that we need a minimum of 60 days to provide a written response for the review of your protocol. So you need to factor that into your product development plan. But like I mentioned, it's not a requirement. But if you don't submit your human factors protocol for review prior to running your study, that is a business decision that you can make for your company. But just know that once you've completed your study and you've submitted your formal marketing application, we will look at your results and we'll evaluate your results to see if they are indicative of ensuring safe use for the intended users in the use environments, etc. But if the methodology that you use as part of your study is something that we cannot align with, we may ask that you provide additional data And one good way to make sure that we're in alignment with your methodology is to have us do a review of your human factors protocol before you actually run your study. So I think I answered all of those questions. So thank you so much.
Thank you, Dr. Surratt. So Dr. Schnupp, we have a couple more questions for you. The first question is, does the STAR pilot apply to oncology products, or should oncology applicants only pursue RTOR?
Good question. So both STAR and RTOR, they are available to oncology products. Sponsors may discuss their preference with the RPM of the relevant division for their particular product. Thank you.
Okay, your next question. Is STAR available only to supplemental applications?
Yes, so currently STAR is only available for certain efficacy supplements. And as I noted during the presentation, assessments will be conducted to consider the feasibility of expansion in the future. Thank you.
Thank you, Dr. Schnupp. Let's see, another question for you, asking how long will the pilot plan run for the STAR program?
That's another good question. Thank you. It is premature to discuss since we will be conducting assessments and workshops in order to evaluate STAR, and we have not yet received feedback and results from those efforts, which will help inform us of any decisions about next steps. Thank you.
Thank you. And one more question for you. How does STAR compare to BTD if the criteria for clinical evidence are similar?
Again, that's also a very good question. So breakthrough therapy designation, or BTD, it primarily provides benefits during the development or IND phase, whereas STAR is more intended to provide a benefit during the early review of the supplemental marketing application. Thank you.
Thank you, Dr. Schnupp. Let's move back to Dr. Surratt for a few more questions. Dr. Surratt, can you please provide a typical sample size and duration for a human factors study validation?
Hi, yes, thank you for your question. So a typical sample size, it will depend on your number of distinct users, how many different types of distinct users you have. So we usually require, or we suggest and recommend 15 distinct users per user group. So that final number of sample size will change depending on if your product is intended to be used in healthcare providers only, or if it's to be used in patients, caregivers, and then there may even be subsets of patients who are injection naive or injection experienced. We even sometimes have a distinct user group who are distinct because of their age. So the final sample size number is something that's going to be unique to your specific product, but I guess in the intended use of the product as well. But what we do know is based on references that we use here at FDA, it's been published that a sample size of 15 is sufficient to find a minimum of 90% and an average of 97% of all use problems. So that's going to be your starting number. It's going to be 15 per distinct user group. Now, the second question about the duration. The duration of your study is also going to depend on how many steps are required to use your product. So if you have a fairly small or short instructions for use with, just a few steps, then of course your study will be shorter. But if you have a complex product, which may have an upwards of 10, 20 steps in order to administer your product, that's going to drive the duration as well. Just want to reiterate that an agency review of your draft protocol before you complete your study will help us determine if you've identified the correct sample size and the correct intended user, number of distinct users, so that we can say that your methods are acceptable. Thank you.
Thank you. Let's see, one more question for you. Is submission of product samples required in order for FDA to evaluate the human factors validation protocol? If yes, how many samples will FDA allow the sponsor to set up the product for FDA reviewers in the same way that the sponsor sets it up for users?
Thank you for your question. That's a great question. So samples are not required for the review of a human factors protocol. We do realize that by the time we receive a submission to review a human factors protocol, it is fairly early on in the product development process. We do expect that within the human factors validation study, you are evaluating the product that you intend to market as close as possible to the intent to market product. But we do also realize that sometimes you may not have a physical sample for whatever reasons. We have had issues with people getting us samples due to mailing or transportation types of concerns. But if we don't have a physical sample, we would like to have a graphical depiction at least, so that we could look at a real picture of what was used in the study and to see all sides of the labeling, the carton, the container, the device itself, if it contains the device. So it's not a requirement that you submit samples. It is ideal, and we would love to have samples. But if not, we would work with you to get alternate means of us evaluating your samples. So I think that was it. Thank you so much.
Thank you. Let's turn it back over to Dr. Schnupp. Dr. Schnupp, if the drug product and packaging are conducted, if DP and packaging are conducted in the US, but API is manufactured at ex-US, would this disqualify from participating in the STAR pilot program? Would this disqualify from participating in the STAR pilot program?
Another great question. Thank you. Due to the timelines for scheduling a foreign inspection, they do not align with the intent and timelines for the STAR program. So supplemental applications that require an inspection of a foreign manufacturing site do not qualify for STAR. And therefore, whether a supplement with an active pharmaceutical ingredient or API manufactured at an ex-US site qualified depends on whether or not review of the supplement would require inspection of the ex-US site. If FDA determined that an inspection is required at the foreign site, then the supplement would not qualify for STAR. So thank you for that question.
Thanks. And one more for you. How long will the pilot program or the pilot plan run for the STAR program?
Again, it's premature to discuss since we will be conducting assessments and workshops to evaluate STAR, and we haven't yet received feedback from the results from those efforts, which will help us in terms of informing us of any decisions about next steps. So thank you.
Okay, Dr. Sturett, we have a few more questions for you. The first one, what is the agency's timeline for review of clarifying questions for a response to a previous human factors advice?
Great question. Okay, so the agency timeline for review of clarifying questions for previously provided advice Well, because we've already provided a response in writing in advance, we should be very familiar with the protocol, and we won't need much time to get back to the sponsor on their clarifying question. Usually we can get that back fairly quickly, considering the clarifying question is submitted very soon after our initial correspondence. That would be, helpful. So it's kind of still fresh in our minds. But if there may be times when the clarifying question is very complex, or if that question may require us to reach out to our counterparts, our colleagues in the FDA for additional input for the experts in their field, and we don't have a established timeline per se for getting back on those clarifying questions. So I guess in this case, the answer would be it depends on how much time we need to put into considering the response. However, we are aware that these questions are necessary to be answered to move forward with the sponsor study. So we keep that in mind and make sure that our goal is to get back to the sponsor as soon as possible, so as not to hold up their development process for their human factors timeline. Thank you.
Thank you. Another question for you. If I am submitting a user-related risk analysis, must I also submit comparative analyses?
Thank you for that question. Okay, so this is also going to be an answer that's going to be, it depends. Well, let's start with, as I stated in the presentation, the risk analysis we know is going to be used as the backbone for submission of the human factors to determine if any human factors data is needed. Because, it's going to determine what risk is needed in the use of the product, and also determine if any additional mitigations are identified that are needed for risk controls. So when considering the submission of a comparative analysis, it's going to be in conjunction with the use-related risk analysis. The risk analysis may be used, the comparative analysis could be used as a supportive documentation to the risk analysis. And when I say that, I mean that in your risk analysis, if you determined any level of risks, you can also provide a comparative analysis to identify a comparator that's similar or same, and go through an exercise where you will do a comparison with the labels, the labels, and the tasks to determine if there are any already or similar products on the market that are already approved in that presentation, and use that comparative analysis as kind of a tool or a reference item to say that, hey, this product is already marketed. It's very same or similar as the one that I have. This controls are already put in place. So I should not have to provide any additional data. So we'll ask the sponsor to use the comparative analysis just to justify that their product is whatever is different about their product, or whatever risk has been identified for their product in the risk analysis. We'll use the comparative analysis as a tool to justify that there's no need for submission of additional risk because of what's already been proven in the market. So if the sponsor will determine that, there's no need for additional data based on the risk analysis, and they submit a comparative analysis to further validate that conclusion, then we will provide, we'll ask that they submit that along with the justification so that we could look at both the risk analysis and the comparative analysis before we make a determination that we agree no additional data is needed. So it becomes a tool for, supportive documentation that no additional study is needed. Thank you.
Thank you. I think we might be able to squeeze in one more question for you. I am not sure that my human factors protocol methodology is correct. Is there an option to request a meeting and to ask for questions on my protocol design?
Oh, sorry. Yes, thank you for that question. If you're not sure that your protocol methodology is correct, we would like for you to, first of all, just submit your protocol. A meeting to have us review your protocol is not always the most efficient way to look and see if we can help you with your methodology, just because of the time constraints and the amount of comprehensive study that we need, comprehensive review that we need to provide. So what we'd like you to do is submit that protocol to the agency for review. And then what we'll do is ask that you also provide those same questions that you have for us that you wanted to submit as a meeting. Provide those questions with your protocol submission. And then we'll do a comprehensive review of your protocol and your questions and get back to you within 60 days in a written format so that you'll be able to move forward with your product development. Thank you.
Thank you. Thank you to the presenters and to the audience for asking the questions, the presenters for answering, and for the great presentations. That's all the time we have for questions right now. And so let's take a short break and we will reconvene at 11 o'clock Eastern Time. Thank you. Welcome back from the break. I hope you were able to stretch and get some coffee or tea. We will have two additional presentations in this next set before lunch break. Our next presentation is on the modernization of clinical trials through digital health technologies, decentralized clinical trials, and point-of-care trials. Presenting is Ms. Beth Konkoski, Health Science Policy Analyst, Clinical Methodologies in CDER's Office of Medical Policy. After this, we will hear about PDUFA VII, real-world evidence from Captain Kimberly Smith, who works in real-world evidence analytics in CDER's Office of Medical Policy. Please join me in welcoming our speakers.
Hello, my name is Beth Konkoski. I am a Health Science Policy Analyst in CDER's Office of Medical Policy. Today, I will be discussing the modernization of clinical trials through digital health technologies, decentralized clinical trials, and point-of-care trials. All of us are privileged to be participating in a technologic revolution. Technology dominates almost everything we do and has opened all sorts of new opportunities. Information technology knows no geographic limits. Data can be instantaneously transmitted from anywhere in the world. Looking at clinical trials, there are many opportunities to embrace these advances. It's obvious that many of traditional processes in clinical trials can be modernized. Decentralizing activities in clinical trials is not new. Patient diaries, interactive voice response systems, and telephone follow-up have been used in clinical trials for years. COVID-19 changed how we conduct clinical trials. Not having patients travel to trial sites allowed many ongoing trials to continue during the public health emergency. During this time, FDA published a guidance on the conduct of clinical trials during the public health emergency. FDA recently published a draft guidance on decentralized clinical trials for drugs, biological products, and devices. A decentralized clinical trial is a trial where some or all trial-related activities occur at locations other than traditional clinical trial sites. Decentralized clinical trials might involve the use of telehealth visits, digital health technologies, direct distribution of product to participants, use of electronic informed consent, use of home health visits to trial participants, or the use of local healthcare providers and facilities. Why are regulators interested in decentralized clinical trials? To improve patient accessibility, to allow patients with rare diseases or patients with mobility or cognitive challenges to participate, to enhance the diversity of patient populations, to enhance patient convenience, to facilitate trial efficiencies, and to reduce the spread of contagious diseases. One of the first things to consider when conducting a DCT is the design of the DCT itself. We see trial activities potentially occurring at locations such as participants' personal physician's offices or their home. So when designing the DCT and determining the assessments to be performed, consideration should be given as to whether an assessment should produce similar results regardless of whether it is conducted remotely or at a trial site. For example, if a participant is performing home spirometry, the result of this assessment may differ than if it was performed under the direct supervision of an investigator at a trial site. Further, assessments performed by local healthcare providers as part of routine clinical practice, for example, evaluation of a participant's symptoms, may be more variable and less precise than assessments conducted by trial personnel. This factor also should be considered when designing a DCT. Another consideration for study design as it relates to DCTs is the statistical analysis to be performed. In non-inferiority trials, if the effect size of an active control drug has only been determined in a traditional site-based clinical trial, this may differ in a DCT setting. In addition, as mentioned, the variability and precision of the data obtained in a DCT may differ from that of a traditional site-based clinical trial. This would not affect the validity of a finding of superiority, but it could affect the validity of a finding of non-inferiority. In terms of inspections related to clinical trials, while many locations used in a DCT may be remote, there should be a physical location where all clinical trial-related records for participants under the investigator's care are accessible and where trial personnel can be interviewed. This location will need to be included on Form FDA 1572 for drug trials and in the IDE application for device trials. When conducting a decentralized clinical trial, you may consider remote trial visits. These allow the investigator to oversee trial-related activities at other locations. There may be challenges associated with remote visits, including local regulations on telemedicine, inability to conduct physical examinations. Use of photography may be helpful but also has limitations. Investigators may not have the same relationship with trial participants with the absence of in-person visits. If there are complex drug administration procedures, it could be troublesome. Some investigational products may require close medical supervision in case of adverse effects. DCTs may also allow for home visits. This novel approach extends the physical reach of the trial and enhances convenience for trial participants. This may allow participants that have mobility challenges to still participate in a trial. Either dedicated trial personnel or contracted fee-for-service staff may go to the patient's home or other location of preference to collect trial-related data. Now, there are also mobile trial units that bring a lab to a more rural area to enhance convenience for trial participants. Just like in a traditional site-based clinical trial, in a DCT, the investigator is responsible for the conduct of the trial, and the individual is delegated to perform trial-related activities. A key difference for DCTs is the extent to which the investigator uses telehealth, trial personnel working remotely, local healthcare providers, and/or digital health technologies in the conduct of the trial. Decentralized features may necessitate additional training, coordination, and standard operating procedures to ensure consistent implementation. DCTs may allow for the direct distribution of investigational product to trial participants. In all trials, the investigator must control release of the product to trial participants. Local state laws differ on direct distribution to patients. They may require locally licensed healthcare professionals, such as a pharmacist, to distribute the investigational product. Sponsors must consider the packing, handling, and shipping of product. Another factor that should be described in the protocol is how investigators will track and document that trial participants or legally authorized representatives receive investigational products. Additionally, the protocol should describe the procedures that investigators or participants should use to return or dispose of unused investigational products and how this return or disposal will be documented. Local healthcare providers, such as doctors or nurses, may be used by sponsors or investigators to perform certain trial-related activities. This may be done, for example, on a fee-for-service basis. Again, such local HCPs would not be considered trial personnel. Please note, though, there are certain stipulations regarding the trial-related services local healthcare providers should provide. These services should not differ from those that they are qualified to perform in clinical practice, and also should not require a detailed knowledge of the protocol or the investigational product. These services might include, for example, obtaining vital signs or performing a physical exam on a trial participant. If there are activities required by the study protocol that are unique to the research or do require a detailed knowledge of the protocol or the investigational product, these activities should be performed by qualified trial personnel who have been appropriately trained. The idea of allowing local healthcare providers to perform certain trial-related activities that fulfill the criteria just mentioned is that the local healthcare provider would simply be performing tasks no different than what they may perform in the course of their usual practice and which do not require specialized training to perform. Such local healthcare providers are already licensed and regulated in terms of providing such services, and their expertise could be relied upon to support the clinical trial. In terms of delegation of trial-related activities, when permitted by the trial protocol, investigators can delegate trial-related activities to local healthcare providers in order to allow the conduct of activities that require in-person interactions with trial participants. These activities might include a physical exam, drawing blood, taking blood pressure. These activities may occur at a local healthcare facility or at a participant's home. One critical consideration regarding delegation of such trial-related activities to local healthcare providers is the potential for variability in the approach across different practices for certain activities. These might include what data are documented when collecting vital signs, how a physical exam is performed, or how evaluation of adverse events is conducted. In order to help reduce variability in such activities, quality control measures, including regular review by investigators of participant data entered by local healthcare providers, should be performed. This will help in the assessment of the consistency and completeness of the required procedures. The type and scope of quality control measures should be tailored to the criticality of the data and the complexity of procedures done by the local healthcare provider. In addition to delegation of in-person activities to local healthcare providers, investigators may utilize technologies such as video conferencing to allow for direct oversight of trial activities delegated to trial personnel. These might include activities such as fitting of wearable sensors, photographing skin lesions, or possibly conducting physical exams. Investigators conducting drug trials as either traditional or DCDs must complete Form 1572, the Statement of the Investigator. This form includes information such as the name and address of the investigator and the research facility, the name and address of clinical laboratory facilities to be used in the study, and the names of any sub-investigators. In terms of determining when trial personnel should be included as sub-investigators on Form 1572, if they are contributing directly and significantly to the trial data, they should be included as sub-investigators on Form 1572. For further information on completing Form 1572, please see the frequently asked questions on the Statement of the Investigator. Local healthcare providers should not be listed on Form 1572 as sub-investigators. Local healthcare providers should, however, be included in a task log, which we will discuss next. To prepare and maintain adequate case histories, investigators must maintain a task log of local healthcare providers who perform trial-related activities. The task log should include the name and affiliation of the local healthcare provider, a description of their role and assigned tasks, the date these local healthcare providers are added to the log, as well as the locations where these activities are conducted. The task log should be dated and signed by the investigator when it is initially created and updated when a new local healthcare provider is added to the log. The task log should be available to FDA during inspections. Electronic informed consent is fairly common in all clinical trials these days. The regulatory requirements for obtaining informed consent and IRB review do not differ for DCTs. Electronic informed consent allows patients to review and sign documents at their convenience at home. Investigators may provide videos and other graphics to make the process informative and more easily understood. FDA published a guidance on electronic informed consent in 2016. If you would like to learn more about the DCT guidance, please join us on June 20th for a webinar on the guidance. The DCT guidance is a draft guidance. Please submit comments to the docket by August 1st. To test to see what you have learned about investigator responsibilities in a decentralized clinical trial, here is a challenge question. All local healthcare providers participating in a clinical trial would need to be listed on a Form FDA 1572, the Statement of the Investigator. Is this true or false? And the correct answer is false. These could be listed on a task log. Now let's discuss how new technologies have enabled new capabilities in clinical trials. We all recognize the profound impact that new technologies have on our ability to gather clinical data. Computers that formerly occupied entire buildings are now condensed into our cell phones with extraordinary capacities for data storage and analysis. Digital health technologies can transmit data from patients wherever they are, as well as provide opportunities to quantify clinical features more precisely. A digital health technology is a system that uses computing platforms, connectivity, software, and/or sensors for healthcare and related uses. Some common examples include portable sensors such as activity trackers or mobile apps. Digital health technologies allow access to more frequent or continuous data. They have the ability to detect sporadic events that might not be caught in a clinic, such as seizures, arrhythmias, or falls. They can collect patient-reported outcomes, such as ecological momentary assessments. They allow for collection of data in the real-world environment and to record objective functionality. Here are some examples of digital health technologies. Some of them are sensor-based, such as continuous glucose monitors, ECG monitors, or actigraphy. Some DHTs are mobile applications run on smartphones, such as patient-reported outcomes or coordination tests for neurological disorders. FDA published a draft guidance on digital health technologies for remote data acquisition in clinical investigations in December 2021. FDA has been reviewing these docket comments and is working towards finalizing the guidance. Some DHTs meet the definition of a medical device, others do not. This definition is defined in the Food, Drug, and Cosmetic Acts. I have determined my DHT is a device. Now, do I need pre-market clearance or approval to use the device in a clinical investigation for another medical product? Devices intended only for use in clinical investigations are typically exempt from many requirements applicable to devices, including pre-market clearance or approval, as long as the investigation complies with applicable requirements under 21 CFR Part 812, also known as the IDE regulations. When is an investigational device exemption application to FDA required? For a DHT that is a non-significant risk device, an IDE application to FDA is generally not required. If my DHT is a significant risk device, when all information required in an IDE application is also contained in an IND, FDA generally does not expect sponsors to submit a separate IDE for these clinical investigations. If my DHT is a device used in accordance with its authorized intended use, an IDE application to FDA is generally not required. Here's another challenge question. All digital health technologies need to be cleared or approved to be used in a clinical trial. True or false? False. As we just discussed, not all devices need to be cleared or approved to be used in a clinical trial. When it's time to select a digital health technology for your clinical investigation, it's important to start with some basic fundamental questions of, is the digital health technology comfortable? Is the participant going to put it on and wear it all day? Is it easy to use? Does the battery last as long as possible? What's the process to sync the data to ensure that the sponsor receives all of the data they need for their protocol? Is it a bring your own approach where a participant is using their own digital health technology? After selection of the DHT, it's time to think about verification and validation of the DHT measurement. This is a technology question to evaluate the DHT. How accurate and precise is the DHT in measuring the targeted feature, such as steps or sleep? Does the algorithm used to interpret the raw signal reliably represent the clinical characteristic or event we're trying to measure? Are the data recorded by the DHT and trial participants the same as the data we would report if we were looking directly at the trial participant? Justification of the endpoint is a clinical question about the feature being measured, regardless of the technology used to make that measurement. Is the endpoint a clinically meaningful measurement of drug effects? Usually comparison with an existing benchmark of performance, such as the UPDRS or other patient-reported outcome or the 6-minute walk distance, is a good way to assess this. Collaboration from many different stakeholders, such as patients, caregivers, physicians, disease experts, and regulators, can help to assess if an endpoint is a clinically meaningful measure. Developing a novel endpoint based on a DHT-derived data can be challenging. First, you have to decide what you're measuring, such as steps. Then you need to define the window of observation and the response that will be seen in each patient. There are many other aspects that should be considered when running a trial using DHTs. There should be trial participant and personnel training. Technical support should be available for their participants and personnel. In addition, there must be a plan to address updates to DHT hardware and software. It is fairly common for security updates to be rolled out regularly, and sponsors must ensure these updates do not impact the integrity of data collected for the trial. 21 CFR Part 11 outlines electronic record protection and retention requirements. When first designing a trial based on DHT data, sponsors should discuss with relevant review divisions the type of data recorded from each participant to be submitted to the FDA for review. The data output of the DHT and its associated metadata should be transmitted from the DHT to a durable electronic data repository. The source data is generally considered the data housed in the durable electronic data repository and not the data stored on individual DHTs. Additional information is available in the newly published draft guidance on Part 11, Electronic Systems, Electronic Records, and Electronic Signatures, Questions and Answers. DHTs may allow us to modernize the performance of trials and improve trial efficiencies. They allow for measurement in challenging populations such as neonates or patients with dementia. They may allow measurement of rare events which are difficult to capture, such as arrhythmias, seizures, and apneic spells. They allow for the measurement of new clinical characteristics such as stamina, gait stability, and tremor. They also provide a more convenient or precise way to measure existing features such as sleep, exercise, or blood pressure. Here's another challenge question. Justification of an endpoint as clinically meaningful depends on the type of DHT used in a clinical investigation. True or false? This is false. Justification of an endpoint is independent of the DHT used. Trials in the clinical practice setting. The opportunity to use existing clinical infrastructure, particularly when supported by interoperable data system, has become an area of increasing interest. The recovery trial in the UK for COVID-19 reportedly recruited 40,000 COVID patients through the National Health System in the UK within 6 weeks. They were able to show the mortality benefits of steroids in treating patients hospitalized with COVID. Practice settings allow engagement of large numbers of patients in short periods of time. They reflect the effectiveness of treatment in real-world environments and the accessibility of clinical trials to patients who wouldn't normally participate. FDA is committed to modernize clinical trials, incorporate technological and scientific advances, and potentially address the enormous costs and burden of drug development. There's interest from Congress, industry, and others in the community to enhance the efficiencies in drug development. There's interest of new stakeholders, engineers, DHT manufacturers to support clinical trials, and patients are seeking more convenient ways to participate in research. Congress also passed several pieces of legislation in the last year to modernize clinical trials. They passed the Prescription Drug User Fee Act, and this includes a specific section on enhancing the use of digital health technologies to support drug development and review. And at the end of last year, they passed the Food and Drug Omnibus Reform Act, which includes specific sections on decentralized clinical studies and modernizing clinical trials, and both of these have guidance requirements associated with them. The PDUFA VII DHT commitments include developing a framework, establishing a DHT steering committee, convening public meetings, identifying and funding demonstration projects, developing guidance, expanding review capabilities, and enhancing IT capabilities to review DHT-generated data and submissions. FDA published the framework for the use of digital health technologies in drug and biological product development in March of this year. FDA published a draft guidance on Part 11 earlier this year. This is Electronic Systems, Electronic Records, and Electronic Signatures in Clinical Investigations, Questions and Answers. FDA held the first of its public workshops in March of this year on understanding priorities for the use of digital health technologies to support clinical trials for drug development and review. CDER has a new webpage, DHTs for Drug Development. This webpage will highlight all of the work we are doing to meet our PDUFA commitments. One PDUFA commitment is to track submissions containing DHT-derived data. To do this, we recently updated Forms 1571 and 356H. Next time you complete these forms, please be sure to check the box yes if your submission contains digital health technology data. Modern clinical trials provide opportunities to improve trial efficiencies, convenience for patients, access to diverse participants and participants with rare diseases, to broaden how we conduct clinical trials, to collaborate with many different stakeholders, and to facilitate drug development. Engaging with the FDA. If you have plans to conduct a DCT or use a DHT in a clinical investigation, please reach out early in the development phase. If you have a specific drug you are developing, contact the appropriate therapeutic review division and request a representative from the DHT steering committee participate in any discussions. If you are considering a DHT or DCT not associated with a specific drug development program and would like to discuss general feasibility, please reach out to DHTs for Drug Development at fda.hhs.gov. Thank you. Now, Captain Kim Smith will discuss real-world evidence. At the conclusion of her presentation, we will both be available to answer any questions. Thank you.
My name is Kim Smith. I'm with the Real-World Evidence Analytics team in CDER's Office of Medical Policy, and I'm happy to be here with you today to discuss the real-world evidence provisions under PDUFA VII. The objectives of today's talk are to describe the FDA's real-world evidence program for drugs and biologics, to understand FDA's approach to evaluating real-world evidence for effectiveness, and to discuss FDA initiatives related to real-world evidence, including new commitments under PDUFA VII. So to start with a little bit of background on the U.S. effectiveness standard, it's grounded in law with the Food and Drug Cosmetic Act of 1962, which states that a drug's effectiveness must be established by substantial evidence consisting of adequate and well-controlled investigations. That's generally been interpreted as calling for 2 adequate and well-controlled trials. With the Food and Drug Modernization Act of 1997, substantial evidence could also be met by a single trial plus confirmatory evidence, which gives some flexibility in the evidence needed to support effectiveness claims. This is further laid out in regulation with 21 CFR 314.126 describing the characteristics of adequate and well-controlled studies to support claims of effectiveness for new drugs, and in guidance with a draft guidance published in 2019 on demonstrating substantial evidence of effectiveness for human drugs and biological products. So this is the background on which we're building the real-world evidence program for drugs and biological products, because our effectiveness standard has not changed. So fast forward to a time when the healthcare system is generating large volumes of clinical care data that could potentially be harnessed to inform our regulatory decision making. Now, this is nothing new. FDA has a long history of using real-world data to monitor and evaluate the safety of approved drugs in the post-marketing setting. Once we get out of the clinical trial setting, we have a lot more patients taking the drugs for a lot longer time. We can sometimes detect safety signals that you wouldn't ordinarily see in a clinical trial setting or a study setting. And so we have always used these types of data to inform our understanding of the safety profile of drugs. We have also used real-world data and real-world evidence to support product effectiveness, but at a really more limited basis. It's often that our decisions have been based on a more traditional clinical trial paradigm. That said, we now have a lot more data than we used to, and also some advances in the analyses of real-world data. And so this is leading to increased interest in the potential use of such data to support regulatory decisions in areas where we typically hadn't focused in the past. So in 2016, Congress passed the 21st Century Cures Act, which was designed to accelerate medical product development and bring new innovations faster and more efficiently to patients. It included some provisions related to real-world evidence. And in response, the FDA established a program to evaluate the use of real-world evidence to support new indications for a drug or to satisfy post-approval study requirements. The agency issued a draft framework in 2018, which described sources of real-world evidence, as well as opportunities and challenges in the use of real-world evidence for regulatory decision-making. The agency, as follow-up to issuance of the framework, issued multiple draft guidances between 2021 and 2022. And underlying all of this, as I said a couple slides back, our effectiveness standard remains unchanged. So the task before us is really to understand when can real-world data and real-world evidence actually meet our effectiveness standard and be used to support a new indication for a drug or to meet post-approval study requirements. So here's just a screenshot of the cover of FDA's framework for real-world evidence, and there is a link down below if you're interested in learning more. The framework applies to the Center for Drugs, CDER, the Center for Biologics, CBER, as well as the Oncology Center of Excellence, OCE. CDRH, of note, has their own real-world evidence program just because their regulatory framework for devices differs somewhat from that of drugs and biologics. The framework provides a multifaceted program to implement real-world evidence, including internal processes within the agency, engagement of external stakeholders, the development of guidances, as well as demonstration projects. The framework is grounded in two key definitions, and I'll use these terms a lot in this presentation, so just to be clear from the outset on what I'm referring to. Real-world data are data relating to patient health status or the delivery of healthcare that are routinely collected from a variety of sources. Real-world evidence is clinical evidence regarding the usage and potential benefits or risks of a medical product derived from analyses of real-world data. To continue with terminology a bit more, we have interventional studies, which is what we've traditionally called clinical trials. In interventional studies, patients are assigned to the treatment or the intervention by a study protocol. This is in contrast with non-interventional studies, often called observational studies, in which patients receive the treatment of interest during routine medical care. It's a treatment that was prescribed by their medical provider. This is not a dichotomy necessarily, so studies can have components of both. So for example, you can have an externally controlled trial where the interventional treatment arm was determined by a study protocol, for instance, a single arm trial, and the outcomes in those patients are compared with patients who received a different treatment through routine medical care. So the control arm is a non-interventional or observational group. There is a spectrum of reliance on RWD in studies. It's not necessarily all or none. So on the left in this table, you have the traditional randomized trial where you may use real-world data in the planning of the trial, for instance, to assess feasibility, determine appropriate enrollment criteria, maybe select trial sites, but all of the other data from the trial are collected through trial sources, central labs and electronic case report forms and so on. On the far right, the opposite end of the spectrum would be a completely non-interventional or observational study. So this is where patients are getting routine medical care with treatments prescribed by their medical provider, and data is collected as part of routine care. Whatever labs the provider is ordering is being collected in electronic health records or maybe claims data. There is an area in the middle, though, where there may be components of a traditional randomized trial, but some of the data for the trial are collected through routine care practices. So maybe you're randomizing a patient to treatment, but then following that patient through data collected routinely and gleaned from the medical record. Or, as I mentioned on the last slide, an externally controlled trial where there are components of both an interventional and a non-interventional treatment groups. So the framework also lays out how the agency assesses real-world evidence for regulatory decision-making. And the three buckets in terms of key considerations are the real-world data. Are they fit for use? Is the study design appropriate? And then is the study conducted in accord with regulatory requirements? We'll go into each of these in a little more detail in subsequent slides. So to start with data, so whether the real-world data are fit for use, there are really two major components of that. Reliability, meaning are the data accurate? Are they complete? Are they traceable? Can we link them back to an actual measure with an actual patient so that we can have confidence that the data that we have are real data? And then relevance. Are the data that you have relevant to the question of interest? So do you have the data elements you need to answer the question? And are the patients representative of patients who will use the drug? And do you have sufficient numbers of those patients to draw conclusions from the data? We have several draft guidances that have published that deal with data considerations. So key sources of real-world data are electronic health records and medical claims. There is a guidance that was published in September of 2021 on assessing EHRs and medical claims data to support regulatory decision-making. Registries are another common source of real-world data. So we have published a guidance in November 2021 assessing registries to support regulatory decision-making for drugs and biological products. And then finally, not specifically related to a data source, but how those data are handled and submitted to the agency, because there are requirements for adherence to certain data standards. And so there's another guidance that was published in October 2021 that addresses data standards for drugs and biological product submissions that contain real-world data. So won't go into detail on all of these, but readily accessible on the web if you're interested in what we say about each of these topics. Moving on to another key consideration when evaluating real-world data and real-world evidence for regulatory decision-making, and that's the study design. So we need to have confidence that the trial or the study design can provide adequate scientific evidence to help answer the regulatory questions. So can the study as designed meet requirements for the intent of the study? If it's for an effectiveness claim, would it meet the requirements for the U.S. effectiveness standard? And so we do have one guidance that has been published in this space to date, and that is on considerations for the design and conduct of externally controlled trials for drugs and biological products that was published in February of 2023. And as noted on CDER's 2023 guidance agenda, we have two additional guidances that are in development. So one is on considerations regarding non-interventional studies for drugs and biological products, and another on using clinical practice data and randomized controlled trials. So stay tuned for both of those guidances. And then finally, in terms of the three key considerations, here are the regulatory considerations. So does the study conduct meet FDA regulatory requirements to support a decision, a regulatory decision? And so here, key is protection of human subjects. So in situations where you may need to get informed consent for the study and to protect data from human subjects. Being transparent about the study design and the study conduct, pre-specifying how the study is going to be conducted and analyzed. Access to data by FDA, so patient-level data to allow us to explore the data, understand the nature of the data, and conduct any additional analyses we need to understand the findings of the study. And then finally, study monitoring, so ensuring that the study is being conducted in accordance with the protocol and to meet the requirements from a regulatory perspective. So there is a draft guidance on all of these considerations. It's called the Considerations for the Use of Real-World Data and Real-World Evidence to Support Regulatory Decision-Making, and that one was published in December of 2021. A couple excerpts from the regulatory considerations guidance I think are important to highlight. So the first is that regardless of a study's interventional or non-interventional design, the evidence submitted by a sponsor in a marketing application to support safety or effectiveness must satisfy the applicable legal standards for the application to be approved or licensed. And there are a lot of considerations with non-interventional studies that differ somewhat from interventional studies. And so it's really important in this space for sponsors to engage with the agency in the early stages of designing a non-interventional study that's intended to support a marketing application. So shifting gears a bit from the 21st Century Cures provisions to PDUFA VII, which will cover fiscal years 2023 to 2027. We have 4 real-world evidence provisions under PDUFA VII. At the top, FDA will establish an advancing real-world evidence program, will report aggregate data on an annual basis describing RWE submissions to CDER and CBER. The third is we'll convene a public workshop or meeting to discuss RWE that meets regulatory requirements. And then finally, we will use lessons learned in this process to update existing or to generate new guidance documents in the RWE space. So to start with the top, we were directed to establish a real-world evidence program to identify approaches for RWE that meet regulatory requirements. The program is also intended to develop agency processes that promote consistent decision-making, and finally to increase awareness of real-world evidence characteristics that support regulatory decisions. So the Advancing Real-World Evidence Program, on the left you'll see a screenshot from our website, and the link is below in case you'd like to get additional information. This is a program that was announced on October 20th, 2022. It allows for up to 4 meetings with sponsors and the agency to discuss the use of real-world evidence in medical product development. It is an optional program. All of the usual opportunities to meet with the agency through established meeting pathways remain available, but this is a new opportunity to have some of these more focused discussions on the use of RWE. So the goals of the program are to identify approaches for generating RWE that meet regulatory requirements, to develop agency processes related to the review of real-world evidence proposals, and to promote awareness of real-world evidence that can support regulatory decisions. As you'll see, one of the components of the program is a disclosure agreement between the sponsors and the agency that allow the agency to discuss the work under this program in a public forum to promote that awareness. So to be eligible for the program, sponsors must have an IND or a pre-IND for a specific product and indication. This is not intended to discuss sort of more general considerations related to real-world evidence and real-world data. The real-world evidence proposed must be intended to meet regulatory requirements in support of either labeling for effectiveness, such as new indications, populations, or dosing informations, or be intended to meet post-approval study requirements. And there needs to be agreement on the information that the agency can publicly disclose related to the proposal and the discussions. So the submission deadlines are twice per year, March 31st and September 30th, and that continues through the March 31st, 2027 deadline at the end of PDUFA VII. The initial request is a 12-page summary of the proposal. If you're interested in what that should contain, all of it is on the public website. For each cycle, FDA will review all proposals received in the preceding 6-month cycle and will accept 1 to 2 initial requests in the first 2 fiscal years, so FY 23 and 24, and 1 to 4 requests in FY 25 to 27. That decision will be based on, first and foremost, the scientific merits of the proposal, but also, given that this is a program intended to encourage learning and exploration about potential uses of real-world evidence, it will also take into account diversity of data sources, study designs, methods, as well as regulatory indications. Sponsors will be notified whether their initial meeting request of the program is accepted within 45 days of the deadline. So here's just a visual representation of that cycle. So on the left, you see that the sponsor submits the initial meeting request on day 0, which is either March 31st or September 30th. The agency has 45 days to evaluate the requests, and by day 45, we'll notify all sponsors of whether they're accepted or they're not accepted. And the acceptance is pending reaching agreement on those disclosures. And then by day 75, we'll have the first Advancing RWE program meeting. Sponsors can take information gained from the discussion and can determine whether follow-up meetings would be of benefit, and if so, at their discretion, they can request up to 3 additional meetings through the program, and if granted, those meetings will occur within 45 days of the follow-up meeting request. So as I mentioned, once we notify a sponsor that they've been tentatively selected to participate, we need to reach agreement on disclosures in terms of what design elements the FDA may present publicly as case studies. And once we have a signed agreement letter, we move forward with that initial program meeting. So the second provision under PDUFA VII related to real-world evidence was that by June 30th of 2024, the FDA is to report aggregate data on an annual basis describing submissions to CDER and CBER, including data sources, study designs, and types of regulatory requests. So more to come on that starting in 2024, but at this time, I just want to call your attention to a final guidance published in September of 2022 on submitting documents with real-world data, real-world evidence to the agency, which requests that when you submit these documents, that you include in your cover letter certain information that will allow us to identify that the submission includes RWD, RWE. So in the guidance at the link at the bottom, you'll see an example table that goes through different categories, the purpose of using the real-world data, real-world evidence, the study designs that you're proposing to employ, as well as the real-world data sources that you're proposing to use with your submission. The third provision under PDUFA VII is that by December 31st, 2025, the agency will convene a public workshop or meeting to discuss case studies. And that'll be focused on how to generate real-world evidence to meet regulatory requirements. So more to come in a couple of years on that. And then finally, I mentioned several guidances that we've published and more to come. The FDA will use the lessons learned from the Advancing Real-World Evidence Program and the other work under PDUFA VII to update existing guidances, to generate new guidances as needed, and that work will occur by December 31st of 2026. So to summarize, there's a lot of work being done in this space. The FDA's Real-World Evidence Program is just advancing as was described in the 2018 framework. Real-world evidence presents some new challenges with regards to data, to study design, as well as to regulatory conduct of these studies. To try to move this field forward and to explain our thinking in certain areas, FDA has issued multiple guidances intended to inform the use of real-world evidence for regulatory decision-making. And we have several new initiatives underway that aim to identify and promote awareness of real-world evidence-based approaches that meet regulatory requirements with several new provisions under PDUFA VII. So moving on to our first challenge question for this talk. The 21st Century Cures Act changed the effectiveness standard for new drug approvals to allow for approvals to be based on real-world evidence. So is this true or false? Just think about your answer to this question. So this one is false. Hopefully you got that one correct. The 21st Century Cures Act did not change the effectiveness standard. It just directed the agency to develop a real-world evidence program and begin to explore how real-world data and real-world evidence could be used to support regulatory decision-making. Challenge question number 2. Which of the following statements is not true? A, real-world evidence can arise from both interventional and non-interventional studies. The use of real-world data to understand trial feasibility is considered real-world evidence. Determining whether data are fit for use includes consideration of reliability and relevance. Or it is important to engage FDA early when considering use of a non-interventional study to support a marketing application. Which of these is not correct? All right, hopefully you got B. The use of real-world data to understand trial feasibility is considered real-world evidence. Sometimes we hear it described in this way, but really the types of explorations that are done to understand trial design we don't consider to be real-world evidence. That's sort of traditional uses of real-world data, but real-world evidence is really understanding the benefits and risks of products through analyses of real-world data. So if you're interested in more information on any of the topics I discussed, there were links, I think, throughout the presentation to the various guidances, the framework, publications. So that is a good place to start. We also have an email address for questions that's shown here on the screen. And feel free to reach out if you have specific questions. We can help to direct you further. So thank you very much for your time, and we will be available soon to answer some questions. Thank you.
Thank you both for those very informative presentations. We will roll right into our panel session. So for the audience, if you have not had a chance to enter your questions into the Q&A chat pod, this is your chance to get your questions answered, so please do so now. We will try to answer as many questions as time allows. So it looks like we have some questions that have already come in. For Ms. Concott, we'll do yours first. A few questions for you. The first question is, have applications been accepted into the Advancing Real-World Evidence program? What advice could be provided to support a successful application?
Yes, and this is Kim Smith. I'll take this one. Thank you for the question. So we did just pass our first deadline on March 31st for submissions to the program for initial meetings, and we have completed our evaluation process and have selected initial programs to participate. More to come on that once we've had the discussions and have some learnings to share with the group more broadly. In terms of advice for a successful application, I think start with the externally facing program website, which goes through in detail the information we would like to see in the initial meeting proposal, and make sure you adhere to the format and content guidelines, as well as sharing the information requested in as clear and succinct a manner as possible. I think that is an excellent starting point. It allows us to carefully evaluate each proposal and determine which to select for each cycle. Thank you.
Thank you, Captain Smith. Since we have you, let's give you one more question. Would the use of real-world data, such as an external control arm, clinical trial, be used only for critical or rare conditions? Are there limitations on the types of indications being studied?
Yes, thank you for the question. So we don't have sort of specified limitations on situations where we would be willing to accept real-world evidence to support a regulatory decision. So I think at this point, it's really on a case-by-case basis through early discussions with the agency. Things that we do consider are situations where there is unmet need, where it may not be feasible or practical to conduct a different type of a study. I think we all recognize that there can be some limitations in the use of real-world data and real-world evidence. And so generally, we're looking for situations where the uncertainty that may result could be minimized, and also it would be reasonable to accept the level of uncertainty that may result from the use of a real-world evidence study to support the decision. So sometimes that is in situations where it is a severe rare disease and it's challenging to conduct other types of studies, but we are certainly open to proposals in other areas and evaluate them on a case-by-case basis based on the criteria that I shared earlier. Thank you.
Thank you, Captain Smith. Another question for you. Why would a sponsor pursue a meeting through the Advancing Real-World Evidence program rather than through another meeting pathway?
Yes, thank you. So I think the advantage of the Advancing Real World Evidence program is this really is a focused pathway to discuss proposals involving real world evidence. So, we have a team that's been convened to evaluate these proposals and to discuss these proposals with sponsors that really has expertise from across the agency in the use of real world data and real world evidence for regulatory decision making. So we know that there are issues that we need to work through. Some aspects of the use of these types of data and studies differ from our more traditional trial paradigms. And so this is really an opportunity to discuss in the context of a specific development program many of those issues with the right people in the room. It also allows for some iterative interactions on a more quick time frame than some of the other meeting pathways would allow, because the program allows for up to 4 meetings on the proposal, with follow-up meetings being held within 45 days of the request. So I think the hope is that the program will allow for, number one, an all-hands-on-deck approach with all the right people in the room to address the issues raised by a proposal, and also to be able to do so in an iterative fashion through multiple interactions over a relatively short period of time. Thank you.
Thank you, Captain Smith. So now let's move on to Ms. Kankowski. So your first question, do the decentralized locations need to be listed on the 1572, or is it only the main site?
Thank you for that question. I think that's the golden question when it comes to a 1572 and decentralized clinical trials. So the guidance discusses that for inspectional purposes, there should be a physical location where all clinical trial related records for participants under the investigator's care are accessible and where trial personnel can be interviewed. This location should be listed on the Form 1572 or for IDE applications, they must be included in the IDE application. And I think some of the other questions that we're going to get into now talk a little bit more about how you make that distinction in a decentralized trial.
Thank you. Another question for you. Are there examples of decentralized clinical trials where an investigational drug, a drug that has not been approved for any indication, is shipped directly to participants' homes?
So the specifics of what has or has not been approved in an IND are generally confidential in nature, so I can't discuss that specifically, but I can talk a little bit to, in the guidance, it talks a lot about more of the risk-based approach of how to decide if a DCT is appropriate for whatever drug you're studying. So some of those lower risk drugs that a patient can administer on their own and is not likely to have a serious adverse effect would be appropriate for shipping directly to a patient's home. Again, that's discussed a little more detail in the draft DCT guidance that is out right now.
Thank you. Let's take one more question for you. We have one here. Would local healthcare providers need to be listed on the 1572 as sub-investigators?
Thank you. So no, local healthcare providers can be listed on what we are considering the task log that is kept by the investigator, and that is just a running list of any local healthcare providers, their name, licensing, affiliation, where they're located, and then it would be a running list of the activities and the dates and times that those local healthcare providers conduct as part of the clinical investigation.
Thank you. Let's move back to Captain Smith with a few more questions. Captain Smith, when is it appropriate to use real-world evidence to support a regulatory decision?
Yes, thank you. I think it's important to note that RWE can involve various study designs. So I think we're often thinking of non-interventional or purely observational studies, but this can also involve single-arm trials with comparator arms from real-world data. It can also involve randomization, where you're randomizing patients to an intervention of interest, but then you're collecting their outcomes using real-world data. So, for instance, you're following electronic health records or medical claims data to assess what happens to the patients following their randomization to a particular treatment. So, I think it's important to understand that real-world evidence is a fairly broad construct. Regardless of the type of study design, however, as I mentioned, our substantial evidence standard remains the same, and so we really need to think through situations where we can minimize the uncertainty involved, and we can have a clear sense that the drug is producing the effect that we're approving it to produce, if you will. So I think it's again on a case-by-case basis, but really understanding the data sources, the study design, as well as the regulatory conduct, so that we can have confidence that the results of the study warrant the regulatory decision that we're making. Thank you.
Thanks, Captain Smith. We have another question that came in for you. Is the agency looking for particular types of study proposals for the Advancing Real-World Evidence Program?
Thank you. No, not necessarily. So we are looking for proposals that are linked to a specific drug for a specific indication. So not a proposal, for instance, just to address some general concept in the real-world evidence space. We really want to be talking about a specific idea that would support a new indication or meet a post-approval study requirement for a drug. And beyond that, we're going to be looking, as I had mentioned, at the scientific merits of the proposal. Are the proposed data sources likely to be appropriate? Is the study design likely to be adequate to answer the question? And then is the sponsor going to be able to conduct the study in a way that would meet our regulatory requirements? Initially, we have you can cast a fairly broad net. Over time, we're going to be looking for ideas that may be more novel, because we do want to have a breadth of experiences through the program in terms of different indications, different types of methods, data sources, etc. So no, beyond a specific drug and a specific indication, where we don't have additional criteria at this time that would narrow the types of submissions that we're looking for. Thank you.
Thank you. Moving back to Ms. Konkoski. There need to be an agency review of protocol-specific definitions of healthcare professional activities to verify that the healthcare professional is the appropriate designation versus sub-investigator.
Thank you. I'll start with the broader picture of making that distinction of the sub-investigator versus a local healthcare provider. And there is a guidance that has been out there for some time on, it's called the Information Sheet Guidance for Sponsors, Clinical Investigators, and IRBs Frequently Asked Questions, Statement of the Investigator, Form FDA 1572, and this was updated in May of 2021. And this really talks about the distinction between the investigator and the sub-investigator. And now that we've come out with the decentralized clinical trials guidance, we break that down a little farther. Section 3D2 is the investigator roles in that guidance. And there it talks about how local healthcare professionals are contracted to provide trial-related services that are part of routine clinical practice. For example, performing physical examinations, reading radiographs, obtaining vital signs, and where a detailed knowledge of the protocol, investigational product, and the investigator brochure is not necessary. These should not be listed on the 1572 as sub-investigators. And so this is always a challenging distinction of where do you draw the line. And so this is where your question is perfect of, yes, of course, we would love to have this discussion with you. Come talk to the agency early when you're planning your decentralized clinical trial. Tell us what your proposal is and why. but we really are hoping that we can start to rely more on some of these local healthcare providers, but that oversight is still the responsibility of the investigator for the trial. So we're happy to have that discussion with you early on as you are designing your DCT. Thank you.
Thank you. Another question for you. What are the most significant barriers to DCT adoption?
So as I was just alluding to, that balance between the investigator and the sub-investigator and the local healthcare provider, we have certainly heard from many sponsors and investigators that it's been a challenge to figure out that role, and also particularly what an investigator is actually comfortable with in delegating and not overseeing day to day, because on a piece of paper they are still legally responsible. So that is definitely one area we've heard a lot of concerns. Also from the planning and logistics aspect of a DCT, as one of the questions was about shipping investigational products. All of those things, there need to be significant planning to successfully carry that out for a large-scale clinical trial. There has to be a lot of training for the trial personnel. Sometimes contract study nurses are used. Those still need to be trained on what the responsibilities are associated with the trial. There needs to be the training with the participants. If a participant is using a digital health technology, they have to be trained on how to ensure all of that data is appropriately captured for the trial. And so all of these things all have costs associated with them, so that can increase some of the upfront costs with running a decentralized clinical trial. Thank you.
Thank you. Let's do one more question for you. If a patient's digital health technology is used, what consideration should be given to source data being available for monitoring or future auditing by the FDA?
Thank you. So the Digital Health Technologies Guidance has a section on record retention and retention, and also there is now the new Part 11 Electronic Systems Guidance that came out earlier this year, and both of those speak to source data and the digital health technology. We recognize that it is not realistic for the source data to be stored on the digital health technology, so we are considering, once that data is transferred from the digital health technology to the sponsor's data repository, that data repository that the sponsor has responsibility for is considered the source data, and so that is what would be inspected if there were a FDA inspection. Thank you.
Okay, let's move back to Captain Smith. Captain Smith, here's a question for you. To generate real-world data to evaluate the drug for a particular indication, should the drug be approved through regular pathways to approve its safety and effectiveness?
Yes, thank you. So I assume the question is asking, for a sponsor to use real-world evidence to support a new indication, should the product already have been approved using more traditional study designs, such as a randomized trial, and have some existing safety and effectiveness data. So should this be for drugs that are already marketed in the US? I think again, there are no absolutes in this space, but in general, for products who have already been marketed in the US and there may be some off-label use for different indications, that's a situation where it may make sense to consider the use of real-world evidence, because you will have existing US data, and so less questions about whether data, for instance, from other countries is applicable to the US population and practice of medicine. And potentially you would have a breadth of experience on which to base determination of efficacy and safety for a new indication. So that would be an area where it may make sense to pursue these types of discussions moving forward. But again, this is a fairly new area, and there's growing interest with all of the data that are now out there. So we are open to discussions of other types of proposals depending on the disease area and the study design and the available data sources. Thank you.
Thanks. We have another question for you. How do the PDUFA VII real-world evidence initiatives relate to the broader real-world evidence efforts already underway?
Yes, thank you for the question. So I really view the PDUFA VII commitments and initiatives as a progression. And so it's a way for us to begin to engage more in specific areas where we know, based on the guidances we published, that there are a lot of questions. And we need to learn through interactions and through case studies how to move this area forward. And so through the Advancing Real World Evidence program, we'll gain experience with specific sponsors and specific proposals. That information and those lessons learned, we'll be able to discuss publicly with others who are interested in this space through the disclosure agreements and future discussions of case studies publicly. And then as we learn more and gain experience in this area, we can feed that back into our guidances with either revised or new guidances, as well as public meetings in the future. So I think all of this is just moving us towards gaining experience, learning, and then using those learnings to feed back into the guidance that we're providing in the future. Thank you.
Thanks. And one more question for you. What happens after sponsors participate in the Advancing Real World?
Yes, thank you. So, as I mentioned, based on one of the other earlier questions, the up to 4 meetings through the Advancing Real World Evidence program are really intended to, provide sponsors with iterative feedback on proposals and try to work through any issues and challenges that may be presented by the use of real-world evidence to support an indication or to meet a post-approval study requirement. And at the end of those interactions, the goal is really to have a generally agreed upon plan. And then, the sponsor, as with any other development program plan, will continue on with the usual pathways for interactions with the review division and with the agency. So maybe that the next step is to submit a formal protocol to the review division for any additional feedback before implementing the study, or if needed, to engage in additional discussions of the proposal. So, this is a program that allows for a large group of folks with expertise to come together with the review divisions and provide input on issues. But if at any point the sponsor is ready to go back to the usual processes, those are always available. Thank you.
Thank you, Captain Smith. It looks like we have a few more minutes, so we will go back to Ms. Konkoski. This is kind of a longer question, but when using is currently on the market with 510(k) clearance as validated measures for evaluating a drug on trial, and later a recall or a device alert occurs for the device used for the investigation, is the suggestion to perform a risk assessment and provide a rationalization for accepting the outcome, then list the limitation or potential variation in the validation device as part of the submission?
Thank you. This is a fairly specific question, but I think it's a best practice for anything that comes up unanticipated in your clinical trial is come talk to the agency. If your method of collecting data in your trial is recalled or there's an alert for it, you're putting your entire study at jeopardy. So I would certainly recommend approaching the review division as soon as you find out this information. Your approach sounds very reasonable of performing a risk assessment and then addressing how those changes or risks are being addressed in your clinical investigation. But really, I can't encourage you enough for many of these different aspects of modernizing clinical trials. Come talk to the agency early and we'll talk through it with you, and that way we can both ensure the validity of the data that you're collecting through your study. Thank you.
Thanks. I think we have time for maybe one more question. For a digital if it were considered significant risk, is there a guidance or regulation that points to allowing the information required for an IDE to be placed in an IND? Additionally, where would the documentation be placed in the IND? Does this apply to all review divisions?
Thank you. That is something that I did address in my presentation earlier this morning, that yes, we do foresee that most significant risk devices can be reviewed as part of the IND. This also is addressed in the digital health technology guidance that is out in draft right now. That is something we did receive many docket comments on, so we are revising that section to make it a little more robust and making it a little clearer for the different buckets of device regulation and digital health technologies. I would encourage you to include that information within your IND. This is where the cover letter is very important, to include in your cover letter that your IND includes a digital health technology. It's considered significant risk. The device aspects of that are that would normally be included in the IDE are actually included in the IND, and just outline in your cover letter table of contents where all of that documentation is actually included. But you are right, the transparency and explaining that up front will really go a long way to facilitate the review process of both centers. Thank you.
Okay, well, I think that's all the time we have for questions right now. Thank you for all those great questions and the for those excellent responses and the presentations. So let us now, we will break and we will return at, is it, let me check, 12:45 from lunch. So
Good afternoon, everyone. This is Brenda once again. I hope that you're enjoying the conference thus far. And for those of you who have the opportunity, I know we're in various time zones. If you have the opportunity, please participate in our poll here today, as I believe it's on our screen right now. So please, the question is, how many SBIA conferences you have attended? This poll is really important to us as we
Welcome back, everyone. I hope you enjoyed your lunch break. To get us started, we do have a few housekeeping notes and reminders. So just as a reminder, this activity is eligible for an attendance certificate, which supports continuing education by SOCRA, RAPS, SQA, and ACRP. Please refer to our website, www.fda.gov/CEDAR SBIA for more details. You will be able to obtain the attendance certificate only upon completion of the survey, which will remain open for 2 weeks. Remember, that's June 23rd. Remember to download the certificate immediately upon completing the survey. It cannot be retrieved after the fact. And please let us know your comments and feedback via the survey. This will guide the design of our future events. As another reminder, and separate from the certificate of attendance, this 5-day event has been approved for 32.25 contact hours of continuing education for physicians, pharmacists, and nurses. Attendees have 14 days from the last day of the activity to log in, complete the required CE evaluations, and attest to attendance to claim credit. So be sure again to complete the CE session evaluation on or before June 23rd. And again, the email that we send to registered attendees at the end of each day's event will have information on both the CE claim code and the survey link. Moving on to our presentations, we have 3 presenters in this block. Our first presentation is on PDUFA VII post-marketing requirements, commitments, pre-approval and post-approval. And our presenter is Ms. Kathleen Weil. She serves as Senior Science Policy Analyst, PMC Program Manager on the Safety, Policy, Research, and Initiatives team, or SPIRIT, within CEDAR's Office of New Drugs. Our next presenter, from our next presenter, we will hear how CEDAR is accelerating rare disease cures and the PDUFA VII Rare Disease Endpoint Advancement Pilot. Presenting will be Dr. Carrie Jolie, Associate Director for Rare Diseases for the Rare Diseases team within the Division of Rare Diseases and Medical Genetics in the Office of Rare Diseases, Pediatrics, Urology, and Reproductive Medicine. And our final presentation in this session is on PDUFA VII Chemistry, Manufacturing, Assessment Updates, and will be presented by Dr. Paresma Patel, Director of the Division of New Drug API in CEDAR's Office of New Drug Products, Office of Pharmaceutical Quality. Please welcome our presenters.
Hello, my name is Kathleen Weil. I'm the Senior Science Policy Analyst and PMR PMC Program Manager in the Office of New Drugs at CEDAR. I appreciate being invited to talk to you about the new PDUFA VII PMR commitments today. The focus of my talk will be on the following: the 2 new PMR commitments under PDUFA VII. I'll also review the 4 authorities for PMRs. We'll discuss how the agency will communicate anticipated PMRs during pre-approval, and how the agency will review PMR release requests. So let's get started. In this slide, I'm going to define PMRs. They are essentially studies or trials that can be required under one of the 4 authorities listed here: Accelerated Approval, Animal Efficacy Rule, Pediatric Research Equity Act, which is PREA, and 50503 PMRs. The definition of studies or trials for both of those definitions can be found in the October 2019 guidance titled 50503 of the Federal Food, Drug, and Cosmetic Act, and that's listed below with the website address in the footnote on this slide. 50503 PMRs are related to safety and reduced effectiveness. As you may recall, FDA's authorities under 5050 were expanded in the Food and Drug Administration's Amendments Act, FDA, of 2007 and the Support Act of 2018. The Support Act stands for Substance Use Disorder Prevention that promotes opioid recovery and treatment for patients and communities. The first PMR commitment I'll review is related to communicating anticipated PMRs. Per the footnote below, anticipated PMRs are those studies or trials based on data received with the original application submission. This is a very important definition. This commitment addresses inconsistencies in the unpredictability in communicating anticipated PMRs, and it recognized the need for a standardized process. And in response to this observation, we created new internal timelines and processes for communicating these anticipated PMRs. Please note that this new commitment applies to only new molecular entity NDA applications and original BLA applications. To highlight the new timelines, anticipated PMRs will be communicated no later than 8 weeks before the PDUFA goal date for standard applications, no later than 6 weeks before the PDUFA goal date for priority applications. And please note that if major safety issues are identified in data that is received after the original application submission, then the timelines on this slide will not apply to communicating that safety PMR. We may refer to this as an unanticipated PMR. As far as PDUFA metrics, we will try to meet this goal 60% of the time in 2023, 70% of the time in 2024, and 80% of the time 2025 through 2027. After you receive the letter communicating anticipated PMRs, we would like you to encourage you to continue those discussions with the FDA about anticipated PMRs and PMCs. As you recall, post-marketing PMCs are post-marketing commitments, which are studies or trials agreed upon in writing between the FDA and the applicant. We may include those in our anticipated communication letter to maintain consistency within our PMR-PMC program and to communicate any of those anticipated PMCs we are aware of at the time. For secondly, we'd like you to consider study or trial design. We prefer that you include only one study or one trial in each PMR or PMC. This facilitates our ability to track and monitor the progress of these studies and trials which brings us to the third point. Please start to prepare a list of those milestones. There should be at least one milestone in the schedule. The schedule may include those milestones listed here, such as a draft protocol submission, a final protocol submission, a study trial completion date, and a final report submission. Make sure your milestone dates are reasonable and they include time for the FDA to review, so for instance, the protocol. For longer studies and trials, we may want you to consider interim reporting submission milestones so that we can keep track of the progress of that study and trial over the years. As mentioned before, for some PMRs, only one milestone may be needed in the schedule. For instance, with animal efficacy PMRs, the study schedule may include only the final protocol submission milestone. Now let's discuss the second commitment, which is for post-approval or existing PMRs. This commitment is related to requests, as I said, to release existing PMRs. It was created to address the lack of a standardized process or timelines for reviewing the release requests related to existing PMRs. In response, the agency established several new standardized processes for reviewing these requests. As you can see, we created several processes and timelines related to the review of an applicant's release request. Going down the pathway to the right from the purple box at the top, when we receive a request to release a PMR, if a request is received and the agency needs additional information to make a release decision, we have 45 days to issue that request for additional information. Following the pathway down to the right, once we receive the additional information, we have 60 days to review the original and additional information and issue a release, not release decision letter. Starting at the top again and going down the pathway to the left, if we receive a release request and we don't need additional information, we have 60 days to review the request and issue a decision letter. This slide is a continuation of the PMR post-approval commitment regarding release requests. It specifically addresses requests for reconsideration. If an applicant receives a letter that denies the release of a PMR, what we call a not released letter, the applicant can submit a request for reconsideration of that decision. The request is reviewed by the division and senior leadership. And senior leadership will be addressed in several different committees, one of several different committees, either the MPPRC, MPCC, or PERC. It depends on which committee it goes to based on the information provided in the request and the type of the PMR. For example, all PREA PMRs will be reviewed by PERC, the Pediatric Review Committee. The agency has 45 days in this whole process to review the request, take it to senior leadership, and respond with a released or not released decision. So what are the next steps for the applicant to consider? Please ensure that a release request includes a complete justification and additional information or supporting information you think the agency will need. For a request for reconsideration, you should include the information submitted with the original request, any other supporting information you think will be helpful for the agency to make its decision. Additionally, applicants can help the FDA easily identify their submissions by clearly labeling the release requests with a PMR-PMC set number and including specific headings on the cover page related to the type of submission. So for instance, with original release requests, please clearly identify that with a PMR-PMC set number. If you're responding to additional information, please clearly identify that as additional information for the PMR-PMC release request, again with the PMR-PMC set number. And for requests for reconsideration to release the PMR-PMC. Please make sure that you submit only one release request per submission. This is our preference because it assists us with our internal processes and assigning a goal to that submission. Now for our first challenge question. During the pre-approval process, when should you review official communication about anticipated PMRs? Is it immediately after the mid-cycle meeting? Immediately after the late cycle meeting? Receive it by 8 weeks for a standard and 6 weeks for a priority before the PDUFA goal date for the application? Or in the approval letter? The answer is at 8 weeks or 6 weeks. You should receive this communication at least by that time. So now for our second challenge question. FDA is now required to provide a response to a release request for a PMR within how many days of receipt? 30 days? 60 days? 90 days? There's no requirement. The answer is B, 60 days. Some key takeaways from today's meeting include the following. We discussed 2 new PMR commitments under PDUFA VII. We also discussed the new process for communicating anticipated PMRs during pre-approval and when the applicant can expect to receive a letter communicating anticipated PMRs. Those 8 weeks before PDUFA goal date for standard applications and 6 weeks before the PDUFA goal date for priority applications. We also discussed new processes and timelines for reviewing release requests and when an applicant can expect the agency to respond. For release requests, we should respond within 60 days. And regarding requests for reconsideration, and including the review of the request and the additional information by the division and the senior leadership, our response should be within 45 days of receiving that request. I also provided some next steps for applicants related to both new PMR commitments that we'd like you to remember. Thank you for listening to this presentation today. I welcome any questions you may have.
Hello, my name is Dr. Carrie Jill Lee. I am the Associate Director for Rare Diseases and the lead for the Rare Diseases team. Our mission is to facilitate, support, and accelerate the development of drug and biologic products for the benefit of patients with rare disorders. We are located in CEDAR, the Center for Drug Evaluation and Research. And I'm very happy to be speaking with you today on how CEDAR is accelerating rare disease cures, as well as updates on the PDUFA VII Rare Disease Endpoint Advancement Pilot Program. This is my disclosure slide. I have a number of learning objectives for you today. The first is to discuss rare disease and orphan product approval trends in the Center for Drug Evaluation and Research. The second is to discuss challenges and considerations in rare disease drug development. And the third is to share updates on CEDAR's Accelerating Rare Disease Cures ARC program and on CEDAR's Prescription Drug User Fee Act 7 Rare Disease Commitment, the Rare Disease Endpoint Advancement Pilot Program. And this latter is a joint venture between CEDAR and CBER, the Center for Biologics Evaluation and Research. When looking at the progress we've made in rare diseases between the years of 2015 and 2022, what you can see is that we really have made progress. CEDAR has approved 180 novel drugs for rare diseases in this time period, and that is exactly half of the drugs we have approved in total. The FDA has approved over 550 unique drugs and biologics for over 1,100 rare disease indications since the passage of the Orphan Drug Act in 1983. However, we are very, very cognizant of the fact that 30 million Americans live with a rare disease, and the vast majority of these do not have approved treatments. And therefore, we understand and are committed to the fact that there is still significant work to be done, despite the fact that we have made significant progress. This next figure shows the proportion of CEDAR novel drug approvals that are orphan over a time period starting in 2010 through 2022. Specifically, this figure shows the number of novel drug approvals over this time in columns for each year that are divided into the number of orphan novel approvals in green and non-orphan drug approvals in blue. The purple line above indicates the percentage of orphan drug approvals of all approvals in a specific year. And so what you can see is that since 2010, the number of orphan approvals has risen dramatically for CEDAR. And in addition, the percentage of all approvals that are orphan in each year has increased. This continued in 2022, where we approved 20 orphan drug approvals in comparison to 17 non-orphan approvals, and this is 54%. The next slide I wanted to show because it's a visual representation of CEDAR's use of expedited development programs. These are intended to facilitate and expedite development and review of new drugs to address unmet medical need in the treatment of serious or life-threatening conditions, which includes many rare disorders. You'll note that in dark blue, we have the non-orphan percentages for each expedited program pathway or program. And in red, we have the orphan programs, the percentage of orphan programs that utilized these. And I think overwhelmingly the takeaway here is that the orphan drug programs really make use of these programs that are intended to expedite development to a higher degree than those that are non-orphan. The expedited programs come with such benefits as increased engagement with the FDA, the ability to utilize rolling review, or in the case of the accelerated approval program pathway, the potential approval of an application based on a reasonably likely surrogate. So what have we learned from all of the programs that we review in CEDAR? And really what we've learned is that we face common challenges in rare disease drug development. We know that our natural history is poorly understood, which is incredibly important when it comes to planning and designing a trial that can demonstrate benefit. We know that diseases are progressive, serious, life-limiting, and often lack adequate approved therapies, so there's urgent need here. We also know that many of these conditions have a pediatric onset, and so there's an additional layer of challenges in drug development in pediatric populations. We know that there's very small populations, so this often restricts study design options. We know that there, within very small populations, can still be phenotypic and genotypic diversity within a disorder. This makes it very challenging when you go to select the patient population specifically in which you need to study the potential effect of a therapy. We know that development programs often lack a solid translational background, and this can be critical to several elements that are just really important in rare disease drug development, such as the strength of confirmatory evidence. When you're thinking of utilizing a pathway towards demonstrating substantial evidence for effectiveness, that is one adequate and well-controlled trial plus confirmatory evidence. Translational background is also critically important to the selection of a surrogate that you believe represents or is reasonably likely to represent a direct clinical benefit. Drug development tools can be lacking, so outcome measures and biomarkers. And also the lack of precedent, including clinically meaningful endpoints. These are often, first novel studies in patient populations that don't have a therapy. So the selection of an endpoint is novel, and this can be very challenging in drug development for many rare diseases. There are also common considerations for the environment of rare disease drug development. So there's many smaller companies that have less regulatory experience. There are active patient stakeholder groups who are looking to navigate and participate in rare disease drug development. There is a dedicated academic community that may have limited knowledge of regulatory requirements, so that's not what they're thinking of when they are engaging or embarking on academic exploration or aspects of clinical trial development. So from the CEDAR perspective, where we receive drug applications to review for the establishment of substantial evidence of effectiveness and safety, it's our duty to engage with our stakeholders to enhance their understanding of the regulatory process and to gain their alignment and support. So how do we do this? Well, we do this through our Accelerating Rare Disease Cures program. And so with this program, we came together approximately a year ago with a vision of speeding and increasing the development of effective and safe treatment options to address the unmet needs of patients with rare diseases, with a mission to really drive scientific and regulatory innovation and engagement to accelerate the availability of treatments for patients with rare diseases. This is a schematic of what CEDAR's Accelerating Rare Disease Cures program looks like. The CEDAR ARC program serves as an umbrella. It is governed by the senior leadership from the Office of New Drugs, the Office of the Center Director, and the Office of Translational Science. And the reason these three offices have come together is because they are really the center of evaluating and dealing with rare disease drug applications on a day-by-day basis as part of their everyday work. And therefore, they are best poised to inform and align on policy development and share information about rare disease drug development to best address the challenges that I enumerated on previous slides. The program is managed by the Rare Diseases team. We serve as the Program Management Office to execute CDER's ARC program. And as I mentioned in my introduction, we are PDUFA mandated to advance the development of drugs for rare diseases for the center. And in serving this role for CDER's ARC program, this really enables us to fulfill our mission. Now, although CDER's ARC program is governed and managed by the offices and team that I just discussed, we work very closely across CDER with other offices and divisions to rely on their expertise for various issues regarding rare disease drug development. For example, we work closely with the Office of Medical Policy, where real-world data evaluation and real-world evidence policy come out of. We work very closely with the Office of Surveillance and Epidemiology when needed for real-world data, as well as post-marketing issues related to rare disease drug development. We might work with the Office of Strategic Programs when it comes to decision-making and evaluation for benefit risk. And then other CDER offices and divisions are pulled in depending on the issues that we are addressing. Under CDER's ARC program umbrella, we also work very closely with other FDA offices and programs because tackling the challenges in rare diseases truly requires a collaborative approach and a community. And so CDER's ARC program works very closely with the Center for Biologics Evaluation and Research on many of our issues, as well as the Center for Devices and Radiological Health. The Oncology Center of Excellence is another partner when it comes to rare disease drug development and evaluation. And within the Office of Commissioner, we work very closely with certain offices, specifically the Office of Orphan Product Development, the Office of Pediatric Therapeutics, or the Patient Affairs staff as needed. I encourage you all to visit our CDER ARC website and email us at cderarcprogram@fda.hhs.gov. Year one for the ARC program has been all about engagement. So I'm going to highlight just a few of the conferences that we developed, planned, participated in, because our goal in this engagement was not only to share our regulatory perspective on innovation and how to address challenges in rare diseases for topics that were very important to stakeholders, both externally and internally, but also to keep these incredible programs on a shared website for stakeholders in perpetuity, so that when we come together and share innovative learnings and discuss challenging problems, anyone can come to our website and then view these conferences and the topics that they addressed moving forward in order that it's really beneficial to as many people as possible. So the first conference that happened around the time of our close to the time of the inauguration of the ARC program was an FDA NIH conference on regulatory fitness in rare disease clinical trials. And this was a really broad overview conference of challenges that brought together all the different disciplines and how they approach these challenges in rare disease drug development. The conference was done between the Rare Diseases team and the National Center for Advancing Translational Sciences at the NIH, and the focus was really on academic investigators and those who were looking on how to bridge that gap between academic investigation and the regulatory aspects of a drug development program. Later that month, there was an FDA Duke Margolis virtual public workshop that centered around the importance of translational science in drug development, so surrogate endpoints, biomarkers, and more. That was put on between the Office of Drug Evaluation and Science within the Office of New Drugs, as well as the Office of Clinical Pharmacology. The focus there was on translational science and its use in the development of surrogate endpoints. Two more conferences that have just recently taken place. One was the FDA CDER Johns Hopkins Center of Excellence in Regulatory Science and Innovation CIRSE workshop on addressing challenges in the design and analysis of rare disease clinical trials, so considerations and tools. The CDER's Rare Diseases team worked very closely with our Office of Biostatistics as well as Johns Hopkins University to put on a wonderful 2-half-day conference on how to collect high quality and fit for purpose data for rare disease clinical trials, the use of real world data to inform rare disease drug development, the design and analysis of methodologies for use in rare disease clinical trials. And that was a really wonderful conference that if you haven't seen, we know that many of these topics are of the utmost importance to our rare disease stakeholders, which was the impetus for putting on the conference. But again, it is available in perpetuity on the ARC website. Also, there was an FDA CDER and University of Maryland Center of Excellence in Regulatory Science CIRSE workshop, creating a roadmap to quantitative systems pharmacology, informed rare disease drug development. And as one can imagine, this was primarily the Office of Clinical Pharmacology within CDER that led and planned this conference in conjunction with the University of Maryland. And so this is another important one that will live on in perpetuity on the ARC website. Patient engagement is equally important, as patients are the foundation and the reason for developing therapies, and their input is critical. CDER staff during our first year of ARC engaged in no less than 23 rare disease patient listening sessions with patient groups. Now, patient listening sessions are held by the Office of Commissioner's Patient Affairs staff or CDER's Professional Affairs and Stakeholder Engagement, and you can go to that website to learn more about those. CDER staff also engaged in 19 patient-focused drug development meetings. And the patient-focused drug development meetings are held by the patient-focused drug development staff within the Office of Center Director in CDER, and those are critically important as well. We also wanted to increase our communication and transparency about rare disease activities. We've heard from people it's hard to know what's going on across CDER when it comes to rare diseases. So one of the things we did with ARC was launch a quarterly newsletter, which you should really go and subscribe to hear more about these conferences, other conferences, public-private partnerships, and other rare disease news that's highlighted going on across the center. We've also recently added a rare disease filter to FDA TRACK. So the FDA TRACK has a Center for Drug Evaluation and Research Drugs and Biologics tracker. And what we are now currently doing is adding a rare disease filter to the information that is provided on FDA TRACK, so that the public really has an understanding of how rare disease approvals are progressing. And finally, one of the most important things that we've sort of engaged on at CDER ARC during this year of engagement is the Learning and Education to Advance and Empower Rare Disease Drug Developers, the LEADER 3D project. So what is LEADER 3D? This is where CDER really sought input from stakeholders who design or conduct rare disease drug development programs. The purpose behind this is to identify knowledge gaps for stakeholders about the regulatory process of rare disease drug development, and then using the information that we receive for us to be able to create or expand educational resources for stakeholders. Through this engagement, we're hoping to better understand exactly what the challenges are in bringing rare disease drug products to market. In identifying these knowledge gaps and producing educational materials, what we were really focusing on was what types of non-clinical and clinical pharmacology considerations are stakeholders struggling with, what clinical trial design and interpretation issues are stakeholders struggling with, and what regulatory considerations for rare disease drug development are stakeholders struggling with. In parallel with this LEADER 3D effort, CDER is additionally working with the National Organization for Rare Disorders, or NORD, to develop an advanced drug development education series for patients and patient groups. And you can read more about this at the link that's provided. Next, I want to turn to some updates in a program that is not directly run by ARC, but we expect to take a lot of the learnings under the umbrella of ARC and translate those to stakeholders internally and externally. And it's the PDUFA VII pilot program, the Rare Disease Endpoint Advancement pilot program. The impetus behind this program is that it provides a mechanism for sponsors to collaborate with the FDA throughout the efficacy endpoint development process. So as I mentioned before, this is a challenge in rare disease. These are often novel endpoints. And a lot of time is spent on the challenges of trial design, which are many in rare disease drug development. And what we really felt was important and what stakeholders felt was important in constructing PDUFA VII was spending time to focus on that development of a novel endpoint that we're confident can demonstrate whether or not a therapy meets that bar of substantial evidence of effectiveness. So in order for a proposal to be submitted to this REdI program, which as I said affords increased interaction with the Center for Drug Evaluation and Research as well as the Center for Biologics Evaluation and Research, this is a joint program. The associated development program should be active and address a rare disease with an active IND or pre-IND for the rare disease. There are some caveats for that. If there is a natural history study, for example, which is intended to look at an endpoint for a future study, then that might be able to be submitted as well. The proposed endpoint is a novel efficacy endpoint intended to establish substantial evidence of effectiveness for a rare disease treatment. And what we mean by novel in this setting is that an endpoint could be considered novel if it's never been used before to support a drug approval, or if it's been substantially modified from previous use to support a drug approval. And really, when we're looking at selection for this program, preference is going to be given to proposals that have the potential to impact drug development more broadly. So one that utilizes a novel approach to develop an efficacy endpoint or an endpoint that could be potentially relevant to other diseases. Preference will also be given to accepting proposals that reflect, as the program grows, a range of different types of endpoints. This is a pilot program, so there's a limited number of qualified proposals that are going to be selected. In fiscal year 23, there's a maximum of 1. The program opens July 1st. In fiscal year 24 through 27, FDA will accept up to 1 proposal per quarter with a maximum of 3 proposals per year. For transparency, because it's very important that we translate learnings from these programs more broadly to rare disease stakeholders, FDA will conduct up to 3 public workshops by the end of fiscal year 2027 to discuss various topics related to endpoint development. Also, novel endpoints developed throughout REdI may be presented by us in guidance document or on our public-facing website or at the aforementioned public workshops. And this may occur prior to the FDA's approval for the drug studied in a trial. Sponsors will complete the REdI pilot program once they have completed the maximum of 4 REdI meetings and do not have additional endpoint-focused questions or issues to discuss with us. Advice provided during these meetings does not constitute a regulatory decision and is considered non-binding. Completion of this meeting this meeting process doesn't guarantee approval. It's to help you develop an efficacy endpoint that is novel for a rare disease. After completing these meetings, however, the sponsor can certainly request additional input through any. Updates on REdI include the fact that we now have a website, which I encourage everyone to visit. There are frequently asked questions on this website, so if you have questions about the program, check there first. They may already be answered. You should also be able to see proposal elements. You should see meeting package elements. You should see disclosure elements. And also links to endpoint development resources for the different and various types of endpoints that you may be constructing. I also wanted to take the moment to put in a plug for a virtual public workshop that is taking place tomorrow and the 8th. So it's a 2-day workshop. It is jointly hosted by Center for Drug Evaluation and Research, the Center for Biologics Evaluation and Research, and Duke Margolis Center for Health Policy. There's a link to register if you haven't heard of this workshop already. And if you have questions, you can always email us at rdmeetings. So the conclusions from this talk is that in recent years, over 50% of CDER's novel drug approvals were for rare diseases. CDER's ARC program will help CDER work more effectively under our rare disease umbrella with all of our rare disease drug development partners. And CDER's ARC program really does look forward to sharing learnings from other rare disease programs, such as the PDUFA VII Rare Disease Endpoint Advancement Pilot Program. I have a few challenge questions for the group. Looking at CDER novel drug approvals from 2015 to 2022, approximately what percentage were for use in non-rare diseases versus rare orphan designation projects? And the answers are here. The correct answer, I'm giving people just a moment to input their answers, is 50%. And our challenge question 2 is that the REdI program will provide the opportunity for sponsors to engage with the FDA on what aspect of drug development? Model-informed drug development, complex innovative design, rare disease endpoint advancement, or all of the above? It's a tricky question, but the rare disease endpoint advancement is the correct answer. We have additional PDUFA programs for model-informed drug development and complex innovative design. Thank you so much. I have really enjoyed speaking with you all today, and I look forward to talking with you all in the question and answer session.
Good afternoon. My name is Pareshma Patel, and today I'll be presenting on chemistry, manufacturing, and controls assessment with a focus on expedited clinical programs, and I'll provide some recent updates under PDUFA VII. I'm currently a division director within the Office of New Drug Products under the Office of Pharmaceutical Quality. By the end of today's presentation, you should be able to describe some PDUFA VII product quality enhancements, name some common regulatory strategies to address CMC challenges under expedited programs, and understand the CMC development and readiness pilot, generally the application process, eligibility, and selection criteria. The outline of today's presentation is shown here. I'll start with a brief overview of product quality. Under PDUFA VII, there are a few enhancements that are directly related to CMC assessment activities. And today I'll be highlighting the enhancements related to facilitating CMC readiness for products with accelerated clinical development timelines. I'll specifically focus on two areas. One is a recently published map that published at the end of last year entitled Quality Assessment for Products and Expedited Programs. And the other is a CMC development and readiness pilot that I'll briefly highlight from the CDER perspective. I'll end with a brief summary and provide some resources. I'd like to give you a brief overview of the vision of the Office of Pharmaceutical Quality, which really describes the end goal when moving from clinical development to commercialization. We believe that a quality product of any kind consistently meets the expectations of the user, and drugs are really no different. Patients expect safe and effective medicine with every dose that they take. And pharmaceutical quality is assuring that every dose is safe and effective, free of contamination and defects. And this is really what gives patients the confidence in their next dose of medicine. I'll start off by highlighting a recently published map titled Quality Assessment for Products and Expedited Programs. This map really outlines OPQ's current thinking on how to work with sponsors on CMC development for products with accelerated clinical timelines, specifically products that have been granted breakthrough or fast-track designation, or products that are part of the CMC readiness pilot, which I'll discuss toward the end of today's presentation. This map may also be considered for products that are mission critical and examples of products that may address public health emergencies such as COVID-19 where there are no available treatments. it may also include products meant to address drug shortages or have orphan drug designation. This map provides examples of regulatory flexibilities that may be considered to address CMC challenges or to expedite CMC development where it's warranted. And the emphasis generally are two things. One is early engagement between sponsors and FDA when products have an expedited clinical development timeline, and the use of a science and risk-based approach in making regulatory decisions and applying flexibilities as they are warranted. And of course, the end goal here is really the anticipated clinical benefit for earlier patient access to drugs. The Office of New Drugs, which is where our clinical divisions reside, typically identifies products that qualify for an expedited program designation. And this is really based on the information that a product has demonstrated the potential to diagnose, treat, or prevent a serious disease or condition for an unmet medical need. Once that designation has been granted, the OPQ assessment team, focused on product quality, plays a significant role in communicating early with sponsors. The team actively participates in industry meetings, including providing forward-thinking and detailed advice for CMC-related issues in a variety of meetings, including Type B, Type C, and Type D meetings, typically focused in CMC-focused meetings. The team works to determine the status and progress of CMC development and the proposed plans for commercial manufacturing. We also review the strategy to ensure that commercial facilities are ready for manufacturing and are GMP compliant. We work to identify opportunities to expedite CMC development using a benefit-risk framework as well. And we identify potential regulatory flexibilities that may be warranted in the program. The team also considers the data and information that should be submitted to support a marketing application and the timing of that data. So for example, there may be data that should be submitted at the time of NDA filing, for example, and other data that may be acceptable to be submitted during the NDA review cycle or even post-approval. The assessment team generally aims to engage early with sponsors and establishes a communication plan for meetings and other interactions to agree to regulatory approaches by the time a marketing application is actually submitted. I won't spend too much time on this subject, but I would like to briefly discuss the topic of facility readiness. To facilitate a marketing application, FDA really encourages early and enhanced communication for facility assessment at early CMC-focused meetings. This includes identifying any intended commercial facilities, including those facilities that may be used for commercial-scale manufacturing, testing, or packaging, as well as any facilities as part of referenced drug master files that may be submitted as part of the marketing application. Any non-commercial facilities that may generate data needed to support the marketing application. Cross-referenced applications such as drug master files should have letters of authorization submitted to the applications as well. Information on the commercial manufacturing process and comparison to the clinical manufacturing processes. The sponsors plan to ensure that commercial facilities are GMP compliant and ready for inspection. The proposed strategy for commercial launch, particularly if there's anything atypical. And I'll discuss some potential regulatory flexibilities in the next few slides related to commercial launch. The strategy for submitting facility information and the timing for that information, whether that should be submitted in an IND or in the NDA, and that should be really discussed early with FDA. And something that's more specific to BLAs is the submission of production schedules and manufacturing schedules. With this information, OPQ's Office of Pharmaceutical Manufacturing Assessment works with the Office of Regulatory Affairs to assess and make recommendations related to facilities. One area that is emphasized in this map is examples of regulatory flexibilities and approaches, and some general areas for consideration. I'm going to briefly discuss and list the general areas here, and then I'll kind of go into them in a little more detail over the next few slides. The basic areas and examples provided in the map of flexibilities are related to control strategies, process validation, analytical procedures, marketing of batches manufactured with the clinical manufacturing route instead of the intended commercial process, and finally, stability data that's needed for a marketing application and the studies to support marketing. One area of flexibility is related to control strategies. The amount of flexibility that may be warranted is based on the sponsor's experience and overall understanding of the product and manufacturing process. And all of the available data is considered when related to control strategies. Of course, the greater the understanding of these aspects, including a thorough understanding of the product and manufacturing process, analytical methods, and capabilities, really helps the agency consider any alternative approaches that may be needed for controls. The OPQ team typically will meet with sponsors and discuss strategies to establish commercial controls or specifications and acceptance criteria. Besides the overall manufacturing experience, the team will also consider the proposed justifications and assess the benefit-risk considerations for the product, taking into account the expedited clinical designation. Often, sponsors with expedited development programs may have limited manufacturing history, clinical experience, and in these cases, the OPQ team may consider additional steps as part of the considerations for the overall control strategy. And this may include asking for additional specification test or in-process testing due to the limited data. This may require some narrow acceptance criteria or process parameters, and there may be the option of setting wider limits until additional data are available. The use of comparability protocols or CMC-focused post-marketing commitments to revise specification test or acceptance criteria once more knowledge and experience is available is also an option. And finally, there may be times when analytical methods may need revisions, and these could be considered as part of a post-marketing commitment. There are a few alternate approaches that may be considered for process validation, particularly stage 2 validation, which is the process qualification of the commercial process to demonstrate reproducibility and must be completed prior to commercial distribution. There are different requirements for process validation for NDAs versus BLAs, and the focus here will be that any alternate approaches should really be discussed early with the OPQ team. As part of the stage 2 validation, based on the amount of data that is available, there may be flexibilities in the submission strategy, and certain studies may be submitted post-approval. Decoupling of the drug substance and drug product process validation is also a potential flexibility. Because both drug substance and drug product processes need validation, it may be possible to perform the PPQ activities in parallel to the drug product. As long as the drug substance used in the drug product PPQ activities are considered comparable and representative of the commercial drug substance process. Another potential flexibility that may be discussed with the agency is concurrent validation and release approaches. OPQ may consider accepting a plan for commercial release of the stage 2 validation batches before all of the validation activities have actually been fully completed. Typically, to accept a plan like this, the review team would need to see the data supports that the process is in a state of control, and that the process is capable of consistently delivering quality product, meeting the specifications and quality attributes. The equipment and testing methods should be qualified and validated, and there should be a commitment from the sponsor to submit stability data for the concurrent validation post-approval. And finally, the overall submission strategy for data and the protocol to be submitted should be discussed with the OPQ review team. I mostly covered the flexibilities related to analytical procedures in the context of control strategies, and I want to touch on flexibilities related to analytical methods and validation of those methods. For submission of NDAs or BLAs, the regulatory requirements should be consistent with those outlined in 21 CFR 314 and 601, respectively. In general, the analytical methods used to support the control strategy should be suitable for their intended purpose. There may be situations, though, during the review cycle where there may be changes that are needed and to the analytical methods, and these changes may be implemented as post-approval commitments, as long as the control strategy is acceptable for approval. It's important for sponsors to know that these types of confirmatory changes may be considered as a PMC if the assessment team finds that to be an acceptable approach. Another potential alternate approach is marketing or launching with clinical batches. Sponsors may consider this in situations where they intend to scale up the clinical manufacturing process for commercial manufacturing, and the scale-up activities may delay the commercial launch of their product. In these situations, commercial distribution with clinical batches may be considered, and the following information would need to be submitted to support the strategy. Identification of the manufacturing facilities that were used, that are intended to be used for commercial distribution. Any material for commercial manufacturing has to meet FDA standards, including GMP regulations. Facility readiness for inspection. Reminders that the stage 2 validation requirements, and those reminders are kind of communicated to the sponsor from the OPQ review team. And scale-up activities can be facilitated by either comparability protocols or PMCs related to those activities for scale-up. Another area that I would like to briefly touch on is commercial launch with stockpiled clinical batches. In these situations, OPQ would need additional information on the facility that manufactured the clinical batches. If the material was manufactured using a process that is representative of the commercial or commercial process and intended commercial processes for future batches, the quality of the batches should be reflective of the commercial control strategy and meet any updated specifications as part of the application. And of course, the batches should be distributed with approved labeling. Flexibilities related to stability data and stability studies are often considered and evaluated by the OPQ review teams. The OPQ assessment team considers all available data and information, including product and process understanding, supportive stability data, and any relevant prior knowledge. If an alternate approach is being proposed for submission of stability data, the FDA encourages sponsors to discuss the strategy with the OPQ team early. Specifically, the aim would be to seek agreement on the stability data and overall strategy to establish a shelf life or retest period prior to submission of a marketing application. FDA would also recommend that sponsors also clarify the data and information that will be submitted and the timing of that data, whether it should be submitted at filing or can be submitted during the review cycle, for example. Examples of regulatory flexibility or stability data or studies include submission of less than the recommended amount of data for primary stability batches, additional stability data being submitted during the review cycle for an NDA or BLA review, stability data from batches that may differ in size from the recommendations outlined in ICH Q&A. So, for example, batches that are less than the pilot scale. And then submission of supportive data may be considered and may be helpful to establish a retest or shelf life that is longer than would be possible if relying only on the primary stability data in batches alone. In these cases, the OPQ team would assess comparability between the supportive stability data to ensure that they are representative of the commercial process. This map also addresses the use of predictive stability models and focuses on use of mathematical modeling of stability data to establish a retest date or stability or shelf life, specifically for NDAs. Predictive stability modeling may be used to establish comparability between primary and supportive stability batches as well, and may be used as part of the justification for using supportive stability data. The evaluation of any proposed stability model will really be evaluated based on the capability of the model to capture all relevant stability factors, for example, temperature, light, and humidity, the validation of the data demonstrating that the model's kinetic assumptions are appropriate, applicability of the model to the container closure system, and then information on whether product stability indicating critical quality attributes were considered when establishing the model. And then information on situations where that model would not really be appropriate. I would like to summarize this portion of my talk. MAP 5015.13 outlines the quality assessment considerations and regulatory flexibilities for products and expedited clinical programs. FDA really encourages early engagement to address any CMC challenges for these types of products. And finally, the goal here is really to provide patients with earlier access to new drugs and biologics. This brings us to challenge question number 1. The MAP quality assessment for products and expedited programs describes regulatory flexibilities for all of the following except A, process validation approaches, B, control strategies, C, stability studies, or D, cGMP requirements. The answer is D. The MAP covers flexibilities for process validation approaches, control strategies, and stability studies, but does not include any flexibilities when it comes to cGMP requirements. Next, I would like to briefly introduce the CMC development and readiness pilot and provide a very quick CDER perspective for this pilot. The background for this pilot is that often development of CDER-regulated drugs with unmet medical needs have an accelerated clinical timeline. And marketing applications for products in expedited programs still need to meet FDA's approval standards. However, products may face challenges expediting certain CMC activities that are required for submission in a marketing application. Therefore, the goals of this pilot are really to facilitate CMC development in accordance with the timeline of clinical development. This would include additional interactions with FDA during development, use of science and risk-based regulatory approaches, streamlining CMC development, and again, the goal here is earlier patient access to products. And I think one of the other goals overall for this pilot is to learn how to do this better and really understand how we could expedite CMC development when there is expedited clinical development. This pilot is really part of the PDUFA VII commitment letter and the associated Federal Register notice published last fall. The pilot will accept between 8 to 10 proposals every year for a 4-year period. 6 of those will be CBER-led products, and 3 applications per year will be accepted for CDER-led products. The Federal Register notice outlines that the sponsors who apply and are selected will have some additional interactions with FDA, including product-specific CMC advice during development, 2 additional CMC-focused Type B meetings, limited additional CMC-focused discussions, and the hope is that these interactions will lead to a mutual understanding of the approaches to CMC information that should be submitted for a marketing application. I'll go over some of the basic eligibility criteria for this pilot. This sponsor should have an active commercial IND in electronic common technical document format. The application should generally be submitted before the end of phase 2, with some exceptions, such as if the development is following an innovative trial design or intended to treat a rare disease. The thought here is that it would be most beneficial to participate in the pilot so that there is sufficient time to discuss CMC development and address any CMC challenges. The sponsor should demonstrate a commitment to pursue CMC development aligned with an expedited clinical development timeline. And for applications under consideration for CDER, the application should be intended for approval as an NDA or BLA. The program should be under an expedited clinical timeframe with anticipated benefits for earlier patient access, such as breakthrough or fast track designation. Sponsors who do not meet these criteria may still apply, even if they don't have expedited designation, and the eligibility will be determined by FDA, typically in consultation with our Office of New Drugs. The published FRN also describes the full request to participate process, which includes providing a description of the CMC development program, the projected timeline for product development and how that aligns with clinical development, information on CMC activities that are anticipated to yield a complete CMC data package, and information that would be needed for the marketing application. Any identified CMC challenges that would require FDA input, and then proposed timing for the additional type B meetings if the applicant does have that information and is selected for the pilot, of course. The request to participate should really include a CMC development status and plans. That includes the available information on product characterization and critical quality attributes, information on the current status of drug substance and drug product manufacturing processes and control strategies. Particularly, it'd be helpful to know if bioassays are used for your product and information on those bioassay development. Any plans for commercial manufacturing or control strategies, if those are known. Information on the proposed facilities and inspection history for for commercial launch, if available. And then drug substance and drug product assessment plans for stability. And finally, information on process validation plans as well. Now, of course, FDA acknowledges that it's unlikely early in development that all of this information will be available, but for the request to participate, what we're really looking for is as much information as possible about the sponsor's current thinking and their thoughts for moving towards commercialization and any specific challenges that may be seen. The pilot applications will be reviewed in a quarterly manner, and FDA will aim to notify sponsors of their selection to the pilot within 180 days of receipt. The pilot will generally accept an average of 9 applications per year, as I noted earlier, and 3 of those will be within CDER. The overall criteria for selection will be based on whether or not facilitating earlier patient access would be clinically beneficial, the overall novelty of the product, the complexity of the product and the manufacturing process or technology, the sponsor's overall manufacturing experience, and overall experience with the specific product type, class, or manufacturing process that will be used. And finally, I think it's important to note that the FDA is aiming to achieve a balance and diversity in the product types, sponsors, and therapeutic indications for this pilot. I would like to summarize this portion of the talk. At this stage, the CMC development and readiness pilot really aims to facilitate CMC development for products with expedited clinical timelines. The emphasis is really to increase communication between FDA and sponsors for these types of programs. The pilot started in April of 2023, and applications will be accepted throughout the year, and sponsors are really invited to apply for this pilot. The overall goal is to provide patients with earlier access to new drugs and biologics. And I would also like to draw your attention to a CBER perspective that will be presented at the latter end of this week, June 8th through 9th, on the same pilot, on the CDRP pilot, and there will be additional details presented in that presentation. So this brings me to challenge question number 2. How many CDER CDRP applications will be selected per year under the pilot? 10, 6, 3, or all of them? The correct answer is 3. CDER will take 3 applications per year and on average, and CBER will accept 6 applications per year on average. And with that, I would like to provide you with some resources. These are resources for the CMC development and readiness pilot. The website provides information on the Federal Register notice and additional information on how to apply. And shown here are resources for the quality assessment for products and expedited programs, including a link to the map, as well as some other guidance documents that are referenced as part of the map. With that, I'm happy to take any questions, and I would like to thank you.
Thank you for those excellent presentations. We are now ready to roll right into our panel session. So if you have not had a chance to enter your questions into the Q&A chat pod, please do so now. We will again answer as many questions as time allows. It looks like there are some questions rolling in. Let's see, let's start with Ms. Weil. We have a question for you here. For PMR release requests, is it preferred to have a meeting to discuss the status and release of the PMR first, or should the sponsor provide FDA the rationale for release of a PMR directly to an application and labeled as described in the presentation?
Hi, yes, this is Kathy. That's a very good question, and we're coming across that. And I think it depends on the timeline that you're looking for a response. So sometimes a meeting may be preferable, and you might get a written response before you would get a response related to the release request timeline. So, but if you submitted a release request, it goes on a 60-day clock under the new process. So it just really depends on what your timing is, what your preferred timing is for a response, because those timelines are a bit different.
Thank you. I've got a few more questions for you. So could you talk a little bit about FDA's expectations for CMC-specific PMCs and PMRs? Are these typically published on the FDA website or included in the approval letters?
So I'm not the CMC PMC expert. That would be Prisma, Dr. Patel. So what I can answer for you, okay, CMC first stands for Chemistry Manufacturing Controls. And there are actually some CMC PMRs, post-marketing requirements. If they're safety related, they fall usually under the 50503 PMR authority. And so if a CMC study happens to be required under 50503, it would be published on FDA's PMR PMC website, and the status would be displayed every quarter. We update that website at on or around the end of January, April, July, and October. And of course, they would appear in the approval letters And you, under our current process, should see them in as anticipated PMRs if we have that information available by the time, either 8 weeks for standard or 6 weeks for priority before the PDUFA goal date. I can tell you CMC PMCs are not published on FDA's website. They are considered non-reportable. Reportable PMCs under what we call 506B PMCs are considered those that fall under clinical safety study or trials, clinical efficacy, clinical pharmacology, or non-clinical toxicology studies and trials. So that's what the difference is between a non-reportable PMC and a reportable PMC. So I can address those two. And from my experience, and Dr. Patel, please feel free to chime in, I have seen these CMC PMCs as part of the approval letter, and have had the privilege of working with the division and CDER and OND and OPQ on some of those PMCs.
Thank you. Let's see, we've got one more question for you here. What information can we expect to see about anticipated PMRs in the communication?
So as far as, I think I lost you a little bit there. I think you asked what is expected to see in the anticipated PMR communication letter. Was that the question?
Yes, sorry about that. What information can we expect? What information can we expect to see about anticipated PMRs in the communication letter?
So as per the PDUFA VII commitment letter, the agency agreed to add available information about the purpose of the study and trial, the study trial design elements, study trial type and population. So those are things we think we will know by the time that letter goes out and is issued. However, please remember this letter will not include a schedule of milestones because it's too soon. It won't include a PMR PMC set number. Those will all be discussed in what we call an engagement period between the time that anticipated PMR letter is issued and the time the PDUFA goal is met and the approval letter is issued. Thank you.
Thanks. We have a few questions coming in for Dr. Carrie Jo Lee. Dr. Lee, is there a priority for a disease that has a severe phenotype or one that affects a larger number of cases?
Yeah, thank you for that question. We talked a lot about expedited programs today, and I think what the person is asking is how one qualifies for that, and really it's about whether or not it's a serious condition, right? So it's a condition associated with morbidity that really has the potential to interrupt day-to-day functioning, which is why so many of our rare diseases, irrespective of how rare they are on the spectrum, qualify for the expedited programs. And those are, again, fast track, breakthrough, priority review, and then there's a separate approval pathway. There's an additional approval pathway for the accelerated approvals. And again, this is really about whether or not these are serious conditions, often whether or not there's unmet need. It could be rare or not rare.
Thank you. We have another question for you. To overcome small populations for rare diseases, do you allow more global reach of patient population?
So that's another great question. Rare disease drug development is absolutely global. And we work very hard at the FDA in partnership with our international regulatory partners. We have an international rare diseases cluster which is between the FDA, the European Medicines Association or Agency, and Health Canada. So we meet routinely to discuss shared indications and scientific considerations related to protocols and marketing applications in order to promote global collaboration in rare diseases. And it is not uncommon to have patients enrolled in multinational trials.
Thanks. One more question for you before we move on to Dr. Patel. If a drug's mechanism of action is similar between a rare disease and a non-rare disease, it may be possible for the drug developer to utilize animal efficacy data of this mechanism of action to support an ODD application for the rare disease indication.
Yep, I lost you a little bit at the end, but yeah, I think what the asker is getting towards, or at least the way that I'm taking it, is if you have a really good animal model, can you utilize that in support of for efficacy in a rare disease marketing application? Now, this would, in general, be in the setting of one adequate and well-controlled clinical trial, plus confirmatory evidence, and that confirmatory evidence can be a number of things. And whether or not an animal model could additionally be utilized to support confirmatory evidence for an application is really going to depend on the strength of that model. And this is why translational science, whether it's an animal model or in vitro work, is so critical in rare disease drug applications. So it's going to depend on the adequacy of that animal model for the rare disease in question. It's going to depend on whether or not you have defined pathophysiology for that rare disease, so that you are confident that the therapeutic and the target that it engages does in fact have a clinically meaningful benefit on the outcome for that disease. So there's a number of factors that would go into play on the strength of the use of an animal model for a rare disease indication. And definitely in those cases, I would say to come early to the FDA and describe your thinking so that we can advise on the strengths of that model for the use in which you are intending.
Thank you, Dr. Lee. Let's move on to Dr. Patel. First question, what are the criteria to participate in the pilot program?
Thanks for that question. So the criteria for the CDRP pilot are really clearly outlined in the FRN, and I think I provided the link to the website there. But I just want to kind of briefly go through it. A few of the basic criteria include that the IND has to be an eCTD format, which is electronic common technical document. Typically, we want to see something that's as early as feasible, so before the end of phase 2, so that we actually have time to interact with sponsors and provide helpful advice as development is proceeding. One of the other eligibility criteria is that there's a commitment to actually pursue CMC development in alignment with the expedited clinical development. And then one of the other criteria to participate is that you either have an expedited clinical timeframe, so that there would be anticipated clinical benefit for earlier patient access, so something like either breakthrough or fast track designation. However, there, FDA is encouraging sponsors that may meet this criteria but not have one of the designations to that they can still apply, and the eligibility will be determined by FDA once we actually see the whole application in hand. So those are just kind of the highlights of the requirements and criteria to participate as part of the pilot. But the FRN does clearly also provide some specific CBER versus CDER eligibility criteria. And I do encourage anybody that can to attend the CBER talk later this week, because there's more time dedicated to the pilot discussion in that talk. Thank you.
Thanks, Dr. Patel. Another question for you. If this is an ultra-orphan drug and the clinical batch is sufficient for at least a year of commercial supply, is this sufficient justification to perform process validation post-approval?
So thank you for that question. And I guess I would just want to clarify that for an marketing under 505B, so for an NDA, process validation is not a requirement at the time of marketing. So an application can be submitted without having that process validation done. And
that would be conducted after approval of an application. And I think I do want to point out, though, in a situation like this, it is helpful to discuss with the agency if there's any plans for flexibility, especially in a situation of an ultra orphan drug where you may have limited batches or you're anticipating limited manufacturing of batches, that you discuss with the agency any flexibilities on the requirements at the time of marketing, or any flexibilities in how you're launching your commercial manufacturing, your commercial launch plans, essentially. So, for example, if you were planning to launch with clinical batches versus the commercial process itself. Thank you.
Thanks. And I believe this question is for you. Are PSB and PPQ batches needed for expedited programs, BLA and NDA?
Thank you. Yes, that does look like it is for me as well. So just to clarify again, the PPQ batches, which is essentially what we call the stage 2 validation of the manufacturing process, is not required for an NDA, for the submission of an NDA. However, it is generally a requirement for BLAs. So BLAs, it is a requirement for the BLA submission that those stage 2 PPQ batches and information be submitted in the marketing application. So that is the difference between BLAs and NDAs. And again, that is also outlined a little more clearly in the map. Thanks for that question.
Thanks for those responses. Let's circle back to Kathy Weil again. So first question. What if I don't receive a letter communicating anticipated PMRs from the agency?
Thank you for that question. If you don't receive a letter, please keep in mind that per the PDUFA VII commitments, that these anticipated letters only apply to NME NDAs and original BLAs, and they don't apply to resubmitted applications. And also remember that the agency, because this is our first year in this new process, we have to meet the goal 60% of the time. So we may not be able to meet it all the time, but we're working very diligently to meet it as much and above 60% of the time for the first year. If you want one and you haven't gotten one, please feel free to reach out to your regulatory project manager, your RPM in the division, and you can always ask about it at that time. Thank you.
Thanks. Another question for you. What if we request release of a PMR in our annual status report or with another submission?
Yes, thank you for that question. I think that question came up a little bit earlier about putting it, requesting a meeting versus, asking for it as a specific release request. We may, if it comes in as part of the annual status report, or that it's mentioned, that you're going to request a release, or as part of even another submission other than, say, a meeting request, the agency might reach out to you and request that you submit a release request in a separate submission so that we can accurately track the progress of that request in our system using our notification goals and make sure that, that request is handled appropriately as per our new PDUFA VII commitments.
Thank you. Let's do one more question for you. We have here, if we receive a request for additional information about a release request, what information should we provide?
Yes, thank you again. So the letter that requests the additional information should explain exactly what the agency needs to better understand that request for release. If it does not and you need further clarification, please reach out to your regulatory project manager for the division before you respond to the letter. If you happen to respond to that letter before, having a clear idea of what you want to provide, we have the opportunity to send another additional request for information that will add to that and explain further what we're looking for. So thank you for that.
Thanks. Let's move back to Dr. Lee. Dr. Lee, would stimulated C-peptide be a qualifying endpoint in the REdI pilot program for indications in preventing or stopping progression of autoimmune type 1 diabetes?
Yeah, so in terms of our REdI proposal eligibility, what you're going to want to remember is that the first the first gate is the sponsor has an active pre-IND or IND for a rare disease. Now, there are some exceptions to that. Namely, if you don't yet have an active development program but are initiating a natural history study where the proposed endpoint is intended to be studied, which would be applicable to a rare disease, or if you have a common disease that you are studying that includes an innovative or novel endpoint that you can then justify as applicable to a rare disease. That's sort of the first type of eligibility criteria that one has to meet to submit a proposal for REdI. And the second group of eligibility criteria is around the proposed endpoint. So is the endpoint novel? And what we mean by that is if it has never been used before to support a drug approval, or if it's been substantially modified from previous use to support drug approval. So those are the questions to sort of ask. We have a web page up with this and a lot more information, as well as the meeting two half-day meetings that we're having coming up both tomorrow and Thursday. So I hope that people make use of those, and it'll be a lot of good information to share.
Thank you. Let's take one more question for you. What is the current status of the Leader 3D program? Have Leader 3D training events initiated, or are training currently available?
Yep, that's a great question, and I'm happy to share on this as well. So Leader 3D, we are currently in the assessment phase, so we've received a lot of feedback from external stakeholders, and now we are reviewing that feedback and determining what are the priorities that our stakeholders really want in order to inform drug development for rare disease products. We're also looking at, what do we currently have available based on their priorities that we could enhance versus what do we need to develop. And so that's the phase that we are in, and what I would really encourage people to do so that you are informed when we start to come out with materials related to Leader 3D is that you can sign up for the Cedar Arc newsletter, which was in my slides, and I believe my slides are going to be available shortly if they're not already. But I would really encourage people to sign up for that newsletter because we will always put information related to education and engagement for rare disease stakeholders in there. And there's also the Cedar Arc webpage at which there is information on the Leader 3D program.
Thank you, Dr. Lee. Let's move on to Dr. Patel again. Dr. Patel, could you comment on the applicability and use of predictive stability models for monoclonal antibodies or fusion protein products to set shelf life and the agency's view on them?
Thanks for this question. This is a really good question. So currently, FDA doesn't generally recommend predictive stability modeling for biologics, including monoclonal antibodies or fusion proteins at this time. The idea is really that the degradation of these types of biologics is unlikely to be amenable to mathematical modeling. With that being said, I think that if there's some sort of sufficient justification or data to support it, that the FDA would certainly want to see that data as part of the justification if an applicant is interested in using or is interested in applying these sorts of models. Thanks.
Thanks. Next question for you. How many years is it anticipated that the pilot program will run?
Yeah, thank you for that question. So this is really outlined in the PDUFA VII commitment letter. So the pilot program at this stage is anticipated to run for 4 years from 2023 to 2027, and an FRN is planned to be published annually in case there are any minor changes that need to be made to the pilot as we learn, as there are lessons learned annually from the pilot itself. And to kind of get to someone else's question, the idea is that CBER will take about 6 applications a year, and CDER will take about 3. There is some flexibility in that as well. Again, it'll probably just kind of depend on what we see from year to year as we run this pilot. This pilot just initiated in April of this year, so we'll kind of take those lessons learned and apply them to next year. Thank you.
Thanks. And one more for you. Is it still possible to submit the CDER CDRP after 3 projects are accepted? And how do the sponsors know if there's
Yeah, so good question. Yes, so we will be reviewing the applications on about a quarterly basis. So even if 3 applications have been accepted into the pilot, that doesn't mean that a 4th may not be, right? So it really just, it's a case-by-case sort of basis at this stage. But applicants will be notified in about 180 days whether or not they've been selected into the pilot. And if for some reason an application was not selected, that doesn't mean that that same applicant couldn't come back again for another cycle. So just to kind of, there's a lot of flexibility here, and I think we wanted to convey that since the application process is currently open, but the applications will be reviewed in a rolling manner. Thank you for that.
Thanks. Let's move on to, we have a question that came in for Ms. Kathy Weil. Can we submit more than 1 request for reconsideration if our release request is denied after requesting reconsideration?
Yes, great question. We consider the request for reconsideration decision sort of the end of that whole released request process. So at this point, that kind of ends the back and forth period about that PMR, and it could be a PMC, but most of the time it's usually going to be a PMR. So thank you.
Okay, thanks. We have some more questions for Dr. Carrie Jo Lee. Dr. Lee, let's see, here's a question. Difference between the REdI pilot program and the drug development tool, or DDT, qualification programs.
Right. So the REdI pilot is really a series of focused meetings specifically on developing a novel endpoint for a rare disease. So if you would like some very focused, promised up to 4 interactions with the FDA, the relevant review division, and additional experts across, CDER or CBER, depending on where the product is, that's sort of the meeting pathway that we would really encourage you to apply to. The drug development tool qualification program is different, and the REdI program, if you complete that, it doesn't result in qualification of an endpoint. The drug development program really should be for sponsors who want to qualify their COA, their biomarker, or their animal model for use under the animal rule.
Thank you, Dr. Lee. Another question for you. When would FDA recommend interacting with the FDA through the REdI pilot program instead of interacting with the FDA review division through the IND?
Sure. Well, the REdI pilot program is in addition to whatever interactions are currently going on, and the review divisions will be involved. in the REdI interactions. So if you as a sponsor, rare disease drug development, as I said in my presentation, is very challenging. There's a lot of things to work out for a rare disease drug development program. But if you're a sponsor who wants focused, dedicated time spent solely on developing a novel endpoint, which can often be very challenging, that's really when you want to submit to the REdI program rather than in addition to your usual meeting interactions with the review division.
Thank you. We have one more question that just came in for you. How many proposals will be accepted into the REdI program?
Yeah, so it's a pilot program. So pilot programs tend to start smaller, and we will be accepting proposals on a quarterly basis. We are going to accept one proposal for the phase that is going to be opening on July 1st. So submitted between July 1st and September 30th, there will be one proposal, and it's on the fiscal year. So each new year starts in October. And then for the subsequent years, so fiscal years 24 to 27, we will be accepting one proposal per quarter with a maximum of 3 proposals per fiscal year.
Thank you. We have some questions that came in for Dr. Patel. Dr. Patel, if scale-up will delay commercial launch, is it possible to commercially distribute clinical supplies with approved FDA commercial label? Does the clinical supply batch need to be prior to
So thank you for this question. I think to kind of summarize, yes, there is a possibility of launching, so your initial commercial batches being manufactured using clinical supplies. And the process validation can happen after an application, I'm thinking NDA right now, after an NDA is actually approved. But in order to launch with clinical batches, again, this would be in a situation where if you would delay commercial launch if you didn't launch with your clinical batches. If the patient need is there, then that is something that the FDA would consider. But in order to do that, there would be a number of things that you would have to provide as part of your justification. In addition, your manufacturing facilities that manufacture the clinical batches will need to be assessed for acceptability as far as the facility statuses goes, and could be open to inspection if needed, to assure that those were manufactured under GMP and appropriate GMP regulations. And so that would be like the major thing that I think we would need to look at when it comes to the clinical supply that's being used for the commercial batches. But once the expectation is that the process validation stage 2 requirements would still be required after that NDA has been approved. I hope I answered that question. Thank you.
Thanks. Another question. Sponsor apply CDRP again if the request was not accepted the first time?
Yeah, thank you for that question. Yes, I think that the idea of the pilot is we will be accepting applications on a rolling basis, and as a program changes, it is encouraged that if an applicant feels like they may want to reapply, maybe something's changed in their program, in their development program, that is highly encouraged at this stage. And we will be reviewing those on a quarterly basis and kind of assessing where the CMC development is and what challenges exist in the development program as well. So absolutely, that is something that the FDA would consider. Thank you.
Thanks. This next question is addressed to both Kathy Weil and to you, Dr. Patel. If a CMC, PMC, PMR is not on the approval letter or FDA website, how does FDA track completion? Are these still considered optional and not a commitment or requirement?
Thank you. I'll go ahead and take at least the first part of that question, but if I might take an opportunity to go back to the release reconsideration question, I just would like to remind the audience that those requests for reconsideration go up through senior leadership for discussion and review. So that I forgot to add to the prior response. As for this question, if it's a CMC related PMR and a PMC, both of those should be in the approval letter. So I just haven't had an experience where we've agreed upon a post-marketing commitment in writing or required a PMR that's not in the approval letter. As a reminder, all PMRs and 506B PMCs will be available on FDA's website with each quarterly update. And for both of these, all CMC, PMCs, and PMRs are tracked in our database of record. So we monitor their progress using that schedule of milestones that's usually that's provided in the approval letter. Where it says, are these considered optional and not a commitment or a requirement? They're usually, anything that's in the approval letter is either agreed upon as a post-marketing commitment, agreed upon in writing in the approval letter, or required in the approval letter. If it's not in the approval letter, it's not an we don't consider that necessarily optional. So that may be different. In OPQ's office with PMCs, but I'll let Dr. Patel require or provide a response to that part. Thank you.
Yeah, thanks, Kathy. I mean, I think to just add a little bit to what you already said, there are CMC PMRs often, sometimes. Most often, CMC PMCs are communicated to applicants. We usually work with applicants similar to a PMR in order to outline the requirements and milestones for a PMC, and that's how those PMCs are tracked according to those to the the interim reports and the final reports, and kind of when those deadlines are. Those are also should be submitted, should be provided as part of the approval letter, that there is a PMC associated, a CMC PMC associated with an approval.
Thank you both for those responses. Dr. Carrie Jo Lee, we have one question for you here. What exactly is the difference between CEDAR-ARC and REdI?
That's a good one. So CEDAR-ARC, the Accelerating Rare Disease Cures Program, is an umbrella program, as I explained in my presentation, and it's really sort of the umbrella that sees all rare disease initiatives and drug development that is going on across the center in order to really provide strategic oversight on what we need to do in order to really engage with our stakeholders internally as well as externally to strengthen rare disease drug development. And the REdI program is a PDUFA VII mandated pilot program. So it is a rare disease initiative to which CEDAR is taking part in, and it will certainly be followed very closely by CEDAR-ARC. It is a PDUFA commitment that the rare diseases team is implementing as part of our PDUFA requirements in terms of being able to provide rare disease support to the center. And CEDAR-ARC would take learnings from REdI and ensure that they are being translated to all parts of rare disease across the center, as well as beyond to external stakeholders that we can.
Thank you. And I think we are out of time for questions. So we are going to go into a short break. I just wanted to thank again the presenters and panelists for responding to the questions and for your excellent presentations, and to the audience for asking such great questions. So let's take a short break, and we will reconvene at 2:50 Eastern Time. Thank you.
Welcome back from the break. We're on the home stretch now. Our first presentation and our final session is on the best practices for human drug product recalls. Our presenter, Doris Chen, is a consumer safety officer in the Incidents, Recall, and Shortages Branch, Division of Supply Chain Security, Office of Drug Security, Integrity, and Response, and the Office of Compliance. Next, we'll present a rough guide to biologics manufacturing. Our presenters include Dr. Joel Welch, who's the Associate Director for Science and Biosimilar Strategy and Chair for Emerging Technology Team in the Office of Technology Products and OPQ. And finally, in this presentation, we welcome Dr. Chris Downey, who's the Director in the Division of Biotech Manufacturing within the Office of Pharmaceutical Manufacturing Assessment in OPQ. Please join me to welcome our first presenter, Doris Chen.
Thank you for that introduction, and good afternoon. Again, my name is Doris Chen, and my presentation will be best practices for drug product recalls. In the event your firm ever needs to conduct a voluntary recall, you'll definitely want to keep a copy of these slides to refer to later. Additionally, I have an extensive references and resources section, which you will want to print out so that you can refer to the most up-to-date guidances, recall coordinator contacts, and other pertinent information. Let's discuss our learning objectives. At the completion of this presentation, you will know when to conduct a human drug recall, who to contact in FDA to report it, how to implement your recall, and lastly, how to evaluate if your recall was effective. Let me start by sharing with you what the Center for Drug Evaluation and Research, or CDER's Office of Compliance's mission is, and that is to shield patients from poor quality, unsafe, and ineffective drugs through proactive compliance strategies and risk-based enforcement action. That's a lot of words, but it means that here in the Office of Compliance, we want to protect the public from violative drugs by encouraging the firm to do the right thing, by taking a market action to remove their violative drug product, or by using some of our compliance or enforcement tools, like seizure of products, warning letters, or if the product was imported from outside the U.S., an import alert that prevents new product from coming into the U.S. market. I work in the Office of Drug Security, Integrity, and Response. Our office has many component branches. I work specifically in the Incidents, Recalls, and Shortages branch on the Recalls and Shortages team. Our goal is to facilitate proactive, risk-based regulatory compliance actions based on science and the law in a timely and efficient manner in order to protect the drug supply chain integrity. We aim to provide high-quality services and to promote excellence and collaboration in our partnerships with all stakeholders. Our incidents team coordinates response to incidents and informants that threaten the safety and quality of the nation's drug supply involving counterfeits, unapproved drugs, and manufacturing quality issues, and collaborates closely with other agency resources to resolve these public health threats. Our Recalls and Shortages team classifies recalls of all human drug products regulated by CDER. We process recall recommendations through CDER recall referrals, which we'll touch on later. We coordinate potential and initiated recall activities by engaging with all relevant agency units, including field divisions in the Office of Regulatory Affairs. We obtain technical evaluations on compounding-related issues from the Office of Compounding Quality and Compliance, adulteration issues from the Office of Manufacturing Quality, and misbranding violations from the Office of Unapproved Drugs and Labeling Compliance. Our team acts as the point of contact for and supports the drug shortages staff, by coordinating all CDER Office of Compliance program actions for management of human drug shortages. We conduct compliance shortage evaluations related to firms that may be impacted by recommendations of enforcement actions, such as warning letters or import alerts. We coordinate the Office of Compliance evaluation of shortage proposals from industry sent to the drug shortages staff. We obtain and evaluate CDER health hazard evaluations, which are obtained for classification of recalls or for potential regulatory actions. How do you know if your firm needs to conduct a recall? A recall can be initiated at various levels of the drug supply chain. It could include manufacturers, contract manufacturers, distributors, own-label distributors, or wholesalers, just to name a few. A recall can also include excipients, active pharmaceutical ingredients or components. A recall does not have to only be a finished drug product. Let's start by defining what is a recall. Referring to 21 CFR 7.3G, a recall is a firm's removal or correction of a marketed product that the Food and Drug Administration considers to be in violation of the laws it administers and against which the agency would initiate a legal action, such as a seizure. A recall does not include a market withdrawal or a stock recovery, so a recall is for a violative product that has been distributed outside the firm's control. A market withdrawal is a firm's removal or correction of a distributed product which involves a minor violation that would not be the subject of a legal action by the FDA, or which involves no violation. What do you consider before you conduct a recall? You're not sure a recall is necessary. Your product is approaching expiry and is not expected to meet specification at expiry. Should you recall? If you have a product you believe is violative, be transparent. Reach out to your ORA Farm point of contact and discuss the situation. ORA Farm can provide guidance if your market action fits the definition of a recall. You're concerned about a drug shortage. If you believe a market action would cause or exacerbate a shortage of a medically necessary product, reach out with your proposal to CDER's drug shortage staff before initiating a recall. The drug shortages staff will review your proposal and determine if this is a viable option to keep an otherwise violative product on the market, because the benefit of having the product on the market outweighs the risks. As I mentioned before, our recalls and shortages team will coordinate with offices within CDER's Office of Compliance and the drug shortages staff to evaluate the situation, taking into account mitigating factors that will allow the safe use of a product that is otherwise violative. Shortage notifications and updates may be reported to FDA at drugshortages@fda.hhs.gov. We also have guidance for notifying FDA of a permanent discontinuation or interruption in manufacturing under Section 506C of the Food, Drug, and Cosmetic Act. So if you're aware of issues that could lead to permanent discontinuation of a product or could temporarily cease manufacturing or release, you must notify the FDA prior to those actions. So whether you're in a situation where you're considering a recall or not, you want to be prepared. Make sure that you establish and maintain recall SOPs in accordance with the FDA guidance and regulations. You can find many of these resources on my references and resource pages so that you know what to do when you're ready to recall. Identify and train your staff on those procedures. Establish recall communication plans so that you know how you will notify your consignees that you're recalling. Know your FDA recall tools and contacts. What do you think has been the #1 reason for recalls in the past 3 years? Is it A, failed dissolution specifications, B, lack of sterility assurance, C, subpotent drug, D, failed impurities degradation specifications, or E, current good manufacturing practices deviations? How did everyone do? The #1 reason for recalls over the past 3 fiscal years has been E, CGMP deviations. Now let's look at the data for fiscal year 2020, 2021, and 2022. The top reasons for human drug recalls have been a combination of CGMP deviations, failed impurities, degradation specifications, a lack of sterility assurance, subpotent drug, and failed dissolution specifications, with the #1 reason being CGMP deviations. As appropriate, your firm should implement procedures to identify indicators that there may be a problem with a distributed product that suggests it's in violation of the Food, Drug, and Cosmetic Act and other laws administered by FDA. Examples of indicators may include an internal report of a product specification deviation or out of specification testing results for a product, consumer complaints about a product. This could include reports of adverse events, reports of disease, injury, or death associated with product use, or inspectional observations related to a product made by a regulatory authority and indicating noncompliance with applicable product regulations. What do you think is the most common form of initiation for human drug recalls? Is it A, firm initiated, B, FDA recommended, C, FDA requested, or D, FDA mandated? How did everyone do? The answer is A, firm initiated. Let's talk about the different ways recalls can be initiated. Firm initiated are ones where the firm on its own may decide to voluntarily recall and report their action to FDA. FDA recommended are ones where ORA Farm will coordinate a call with CDER and the firm to recommend a firm voluntarily recall due to information supporting product adulteration and or misbranding charges. I mentioned earlier that our team processes the recall referral forms that we receive from other offices within CDER's Office of Compliance, such as the Office of Manufacturing Quality, Office of Unapproved Drugs and Labeling Compliance, and the Office of Compounding Quality and Compliance. The recall is still considered a voluntary decision by the firm. FDA requested recall is ordinarily reserved for urgent situations where mandatory recall authority does not exist or is not appropriate. Most often these are Class 1 situations where there's a reasonable probability of a serious adverse event or death associated with use of the violative product, and the firm has not yet recalled on its own. The FDA requested recall is a letter from the Associate Commissioner for Regulatory Affairs to the firm requesting they initiate a recall. The recall is still a voluntary decision by the firm. Mandatory recalls are for controlled drug substances only at this time, where there's a reasonable probability of serious adverse health consequences. Now let's talk about what to consider when reporting your recall to FDA. This can include, who should I contact at FDA? What if FDA contacts me to recommend a recall? What information should I provide? Who do you contact at FDA to initiate your recall? The Office of Regulatory Affairs recall coordinators are your primary points of contact for recalls. I've also included a link for the FDA ORA recall coordinators in the References and Resources section. The USA and its territories are divided into 4 divisions, and you will work with the division based on your geographical location to discuss a potential market action or to implement your recall. If your firm is located outside the U.S. and you distribute within the USA, your U.S. agent will act as your representative and facilitate your recall on your behalf. Your U.S. agent will contact the ORA recall coordinator based on their geographical location. The ORA farm recall coordinators can provide information on initiating and conducting your recall and provide guidance through all the steps up to terminating your recall. What should you do if FDA contacts you first? If FDA contacts you, this would be considered an FDA recommended recall. Cedar Recalls typically coordinates with ORA Farm to recommend a voluntary recall based on findings supporting product adulteration and/or misbranding charges. During this call, we will communicate to a firm the recommended products, issues, charges, and recall initiation guidance. This is followed up with written guidance from the ORA Farm Recall Coordinator, and we recommend a 24-hour response from the firm in writing with the firm's decision. When you have made the decision to voluntarily recall your product or products, what information do you provide to FDA? This information is summarized in something called Attachment B. You can refer to the link for the Regulatory Procedures Manual, Chapter 7, that provides FDA guidance on what information you would include, such as product information, strength, package size, UPC or NDC codes, lot information, copies of labeling, firm information for the recalling firm, manufacturing firm, and the responsible firm who caused the recall, the reason for your recall and your health hazard evaluation or assessment, volume of recalled product, its distribution and consignee list, and your proposed recall strategy, including copies of recall letters and press release if one was issued. What things do you consider when you're ready to implement your recall? How do you determine the appropriate scope and depth? What level of recall communication is appropriate? In order to answer those questions, we first need to understand the 3 recall classifications, which are determined by the Recalls and Shortages team. I want you to understand the classifications, however, FDA determines the classification and not the firm. Class 1 is a situation where there is a reasonable probability that use of or exposure to a violative product will cause serious adverse health consequences or death. Class 2 is a situation where use of or exposure to a violative product may cause temporary or medically reversible adverse health consequences, or where the probability of serious adverse health consequences is remote. Class 3 is a situation in which use of or exposure to a violative product is not likely to cause adverse health consequences. Your recall strategy is a plan-specific course of action to be taken in conducting a specific recall, which addresses the depth of the recall, need for public warnings, and extent of effectiveness checks for the recall. Let's talk about the depth of a recall. Depending on the product's degree of health hazard, its likelihood for occurrence, and extent of distribution, the recall strategy will specify the level in the distribution chain to which the recall is to extend to. In CEDAR, we typically will recommend wholesale, retail pharmacy, or consumer user level depths. Scope is the recalled lots that are or may be affected. FDA will want to know your bracketing rationale for limiting your scope to the recalled lots identified. FDA will also want to know your bracketing rationale in the event you made the decision to expand the scope of the recall to include additional lots. Upon review of your scope response, among other information, there may be circumstances where FDA recommends an expansion beyond what you're recalling. Your firm's recall communications are based on the product defect risk and is determined as part of your recall strategy. See the resources and references slide for links to the Regulatory Procedures Manual, where you can find model recall letters and response forms, as well as a model press release should you wish to include press as part of your recall strategy. Press is recommended for high-risk recalls, which would include potential Class 1 recalls or recalls to a vulnerable population, such as over-the-counter infant or children pain reliever products. The firm's recall press release is posted on FDA.gov's website. After drafting your press release using the press release template, you would work with your ORA Farm Recall Coordinator, who will send it for review and let you know the agency's comments and edits before you post it. Let's talk about some FDA recall communications and what they are. A CEDAR alert warns consumers not to use a certain product. It is generally issued when the firm has not yet taken a market action or the firm's communication is inadequate and CEDAR needs to clarify. A CEDAR immediate public notification is technically not a recall communication in itself. However, it is generally initiated by health fraud when FDA analysis has found a tainted dietary supplement and wants to warn consumers not to use them. This is different than the recall CEDAR alert because the manufacturer or distributors are unknown or often difficult to find. An FDA press release is an agency-level communication to get the attention of the media and helps amplify the agency's message to patients. This is usually reserved for serious defects, for example, those that can result in death. The FDA enforcement report contains recalls that have been classified or have been posted as determined to fit the definition of a recall, but are not yet classified. FDA evaluates the effectiveness of a recall by evaluating a company's efforts to properly notify customers and remove the defective product from the market. If a recall is determined to be ineffective, FDA will request the company take additional actions. Effectiveness checks verify that all consignees at the recall level depth specified by your strategy have received notification about the recall and have taken appropriate action. It's FDA's expectation that you reach out to 100% of the consignees that you sold or distributed your recalled product to. In order to ensure your recall was effective, you will conduct effectiveness checks. Refer to the guidance for industry for model effectiveness check letters and response forms. Communicate regularly with your consignees to ensure they receive their recall letter and understand what to do. Follow up with consignees that have not been responsive. Before ORA Farm will terminate your recall, all reasonable efforts shall be made to remove the violative product from the market. Therefore, communicate with your ORA Farm Recall Coordinator contact to keep them apprised of the status. Let's talk about a few high-profile recalls we have seen in the news recently: contaminated ophthalmic products and hand sanitizers. As I discuss the case studies, what indicators do you observe? And if your firm manufactures sterile ophthalmic products or hand sanitizers, what best practices can you take back to your team? This is a screenshot of the CDC webpage. CDC alerted the FDA of an outbreak of a drug-resistant Pseudomonas aeruginosa strain associated with the use of an over-the-counter artificial tears product. CDC had tested products and found a commonality with one drug manufacturer. Based on concerns with contaminated products that already entered the market and manufacturing information provided by the firm, Cedar Recalls and ORA Farm contacted the firm and recommended a recall. The firm agreed to recall all distributed lots. The distributing firms, who are the recalling firm's consignees, were to conduct a sub-recall, which is an action taken to notify their own consignees of the recall, as long as the recalled product is unchanged, including the packaging. If you are a manufacturer of ophthalmic drug products, what best practices did you learn from this presentation? Under 21 CFR 200.50, all ophthalmic drug products must be sterile. Review your manufacturing process. Is it adequate for sterile production? Review your formulations and container closure systems. Is your product a single-use product without a preservative? Is your product a multi-dose product? Does it have appropriate controls to prevent microbial growth throughout its shelf life? Ensure your products are manufactured under CGMPs. If you are aware there's a problem, quarantine the products and stop distribution while you're deciding on an action, and keep the agency informed. Since the beginning of the pandemic, we have seen an increase in hand sanitizer products come on the market. Some imported into the USA because domestic resources could not keep up with the demand. Firms saw a need and possibly a monetary incentive for marketing hand sanitizers. The problem was that some of these products had the presence of methanol, 1-propanol, acetal, or acetaldehyde. Some were either subpotent or superpotent for ethanol. Some had microbial contamination. Some were packaged in food-type containers like the pouches they sell pureed fruit in for children, or bottles resembling water bottles. Some products were even labeled as being edible. As of May 1st, 2023, the agency recommended 104 voluntary recalls from firms who manufactured or distributed violative hand sanitizer products, and 150 violative hand sanitizer products have been recalled. The FDA used additional regulatory tools to protect the U.S. public that include firm-based and a country-based import alert. On the FDA.gov website, you can see a full list of hand sanitizers that the agency urges consumers not to use. If you are a manufacturer of hand sanitizers, what best practices did you learn from this presentation? Know your suppliers, like the API, components, supply chain. Are they reputable? Ensure your products are manufactured under CGMPs. If you're aware there's a problem, quarantine the products while you're deciding on an action. Keep the agency informed. If the agency is letting industry know there is a problem, look into the issue to see if it impacts your product. These best practices are also true for other types of drug products, not just hand sanitizers. Ready for our first challenge question? Which of the following ways of initiating a recall is not considered voluntary? Is it A, firm initiated, B, FDA recommended, C, FDA requested, or D, FDA mandated? The answer is D, FDA mandated. Firm initiated, FDA recommended, and FDA requested are still considered voluntary actions. Challenge question 2. If I have a product that I'm not sure if I should recall, I should A, contact my local ORA farm recall coordinator to obtain guidance. B, wait until I'm inspected and let the investigators ask me why I didn't take a market action. C, do nothing and hope for the best. D, wait until there are adverse events reported. I hope everybody got this. The answer is A, contact my local ORA farm recall coordinator to obtain guidance. Challenge question 3. Which of these scenarios is not considered a recall? A, distributed product that failed impurity specifications. B, product lot that obtained out of specification results for dissolution but is now expired. C, lots that were tested that meet specification but are supported by a stability lot that failed for assay. D, liquid product that was manufactured using water that was found to be contaminated, but finished product testing did not find contamination. The answer is B. A product may be OOS, but at the time you initiate the recall, it cannot be expired. You can recall product on the day it expires as long as you notify your consignees regarding the recall before the expiry date. Here are some closing thoughts. Conduct effective recalls in the interest of public health. Establish adequate recall procedures that support early and prompt execution of appropriate market action by well-trained staff. Know your FDA contacts and important tools such as FDA guidance and websites, Recalls Procedures Manual, and the Code of Federal Regulations, also known as the CFR. And talk to FDA early and transparently. Take these 4 major points back to your team and make sure your firm will be prepared in the event you ever need to recall. Thank you. The 4 resource slides I spoke about in my presentation are in the PDF file of this presentation that you can download with my slides from the website. These will be helpful for you to print out and refer to should you ever need to recall. I will be back shortly for our Q&A session. Thank you.
Good afternoon. It is a real pleasure to be with you today. My name is Joel Welch, and I am the Associate Director for Science and Biosimilar Strategy in the Office of Biotechnology Products and the Office of Pharmaceutical Quality in Cedar. My co-presenter today is Dr. Christopher Downey, who is the Director for the Division of Biotechnology Manufacturing in the Office of Pharmaceutical Manufacturing Assessment in the Office of Pharmaceutical Quality in Cedar. Before we begin our presentation, we first must begin with a foundational idea, which is what is the meaning of pharmaceutical quality? Everyone deserves confidence in their next dose of medicine, and pharmaceutical quality assures the availability, safety, and efficacy of every dose. This presentation will include a basic background on how biological products such as monoclonal antibodies and other therapeutic proteins are regulated by FDA CDER. It will discuss unique factors for chemistry, manufacturing, and controls for biological products, including both scientific and regulatory nuances. The presentation will also address how CDER approaches inspectional activities for these biological products. And finally, it will identify key themes, common, that lead to complete responses for marketing applications for these products. First, I will begin by discussing some of the unique factors of chemistry, manufacturing, and controls for biological products, including both their scientific and regulatory nuances for these programs. Before we begin, it's helpful to clarify which center regulates which biological product. The Center for Drug Evaluation and Research regulates monoclonal antibodies for in vivo use and most proteins intended for therapeutic use, for example, enzymes or cytokines. The Center for Biologics Evaluation and Research regulates other biological products. This would include cellular products, gene therapies, vaccines and vaccine-associated products, regardless of their structural composition, allergenics, blood and blood components, and finally human cell and tissue-based products. With regard to biological products regulated by CDER, described on the previous slide, this summary helps identify the type of application that would be submitted for a potential product to CDER. As you can see, the dividing line between new drug and biologic is 40 amino acids. For products 40 amino acids or fewer, they're regulated as new drug applications or subsequently abbreviated new drug applications. For products greater than 40 amino acids, a biological license application is submitted. Critically, this is true even for molecules for which the amino acid polymer is made by chemical synthesis or derived from a biological source material, including production through recombinant technology. This slide attempts to summarize steps associated with a typical manufacturing process for a recombinant protein therapeutic. It summarizes key activities grouped by their scientific function, and on the right, the guidances associated from ICH, the International Conference on Harmonization. You can see the guidances are grouped on the particular topic of the technology and manufacturing process portion they support. The process begins with the creation of an expression system using a desired gene and expression vector to create a cell, to populate the cell substrate, and with it create an expression system. From there, cell banking establishes how typical production will be performed. After cell banking, cell culture begins to generate protein, which is subsequently harvested and purified and becomes what we call the drug substance. Formulation and fill finish then is performed and results in a final drug product to be given to patients. Some guidances apply only to a subsection of these activities, while some, like Q7, apply to the whole drug substance. Finally, overarching guidances such as M4Q on dossier arrangement, and Q9, Q10, Q12, which describe quality risk management, pharmaceutical quality systems, and finally, technical and regulatory consideration for the life cycle, respectively, would apply to the entire process. Nevertheless, in the addition to the regulatory considerations, biological products will have some very specific, unique scientific considerations that need to be addressed as well. First, biological products are frequently highly complex in both their size and number of CQAs, critical quality attributes, relative to small molecule products. Secondly, biological products frequently have not only product-related impurities, but also product-related substances that may retain activity, which drives both the need for consideration of molecular characterization as well as that molecule's control. This control strategy may well be informed by methods and data generated not only at the intended commercial process and scale, but frequently generated in scale-down models, which are needed to evaluate some aspects of a manufacturing process that cannot be performed at scale, such as viral clearance. Understanding how a model was qualified and its relationship to commercial manufacturing is a key feature. This control strategy for critical quality attributes may well be informed by some techniques that may not fully resolve each quality attribute, such as the charge variant profile shown here on the bottom right of the slide. Understanding the totality of how an attribute is monitored and controlled is therefore critical. Finally, these critical quality attributes may not only be molecule-specific, but may well be indication-specific as well, such as an effector function for a monoclonal antibody depicted here on the bottom left, which might demonstrate antibody-dependent cellular cytotoxicity in only a subset of indications. Here we discuss the role of process validation and the expectations for biologics. The process validation is fundamentally the collection and evaluation of data which establishes scientific evidence that a process is capable of consistently delivering a quality product. It is a series of activities that occurs over the life cycle of the product and process. Process validation is divided into 3 stages. Stage 1 is process design, where experience helps define the manufacturing process. Stage 2 includes an evaluation of the capability of the process. And finally, Stage 3, continued process verification, demonstrate that the process remains in a state of control, the validated state, during the commercial manufacturing lifetime. Process validation stage 2, or what is sometimes referred to as PPQ, or process performance qualification, is a potential distinction between ANDAs and NDAs and biologic license applications. While PPQ must be completed prior to commercial distribution for all molecules, this data is expected to be submitted in the dossier for assessment at the time of BLA submission. Additionally, for BLAs, the facility should also be manufacturing the product for licensure is sought during the review as well. Biologic license applications have unique considerations and conditions that must be met as a part of their licensure. First, the facility in which the biological product is manufactured, processed, packed, or held must meet applicable standards. As mentioned previously, inspection plays a critical role for biological products, and this is a topic that will be discussed in greater detail by Dr. Downey. For this reason, additional considerations such as consent to inspection and a determination that the product complies with license conditions and meets applicable standards must also be performed. Critically, these licensing conditions apply equally whether or not you are submitting a 351A or 351K application. Given the unique consideration for BLAs that have just been discussed, several other key items should be considered when preparing a submission. First, it is important to make sure all facilities are registered with FDA at the time of inspection. Secondly, a preliminary manufacturing schedule should be provided in the BLA submission to facilitate the planning of pre-licensure inspections during the review cycle. For this reason, manufacturing facilities should be in operation and manufacturing the product on review during the inspection. Finally, as the FDA expects that a license holder has knowledge of and control over the manufacturing process of the biological product for which it seeks licensure, Type 2 DMFs for drug substance intermediate, and drug product are not typically permissible for BLAs for biological components. Finally, key data and information should be included to ensure sufficient information is provided to describe both routine operation and data commitments that are ongoing as part of a biologics license application. Provided here is a list that is not comprehensive but gives some of these critical examples. These include data to support shipping validation and information on the reuse of key materials such as membranes and chromatographic purification resins. In addition, routine monitoring for things such as reference standards and cell bank should be provided with specifics on how they will be monitored. Finally, other elements, while not required, are also potential nice-to-have submission elements. These might include qualification protocols for things such as new reference standards or new cell banks. This brings us to challenge question number 1. When is Stage 2 process validation data required for a new BLA submission? Answer A, never. Answer B, in all instances. Answer C, upon the request of the review team. Or answer D, only for combination products. The answer here is B, in all instances. And with that, I will thank you for your time and invite Dr. Christopher Downey to continue the presentation. Thank you.
Thanks, Joel. As Joel said, I'm Chris Downey. I'm the Director of the Division of Biotechnology Manufacturing in CDER's Office of Pharmaceutical Manufacturing Assessment. In this next section, I'll be discussing the pre-license inspection portion of CDER's Biologics License Application Assessments. Pre-license inspections, or PLIs, are conducted by CDER as part of our Biologics License Application Review process. These inspections are led by CDER, and the CDER team includes a team of experts in application assessment who are both experts in assessment and inspection. These are distinct from surveillance, post-approval, or for-cause inspections, which are led by the Office of Regulatory Affairs. PLIs, assess the facility and manufacturing process for the subject product of the BLA, and they observe the BLA product during its manufacture. Because of this, the applicant provides a manufacturing schedule at the time of submission and works with the CDER inspection team to plan the inspection to meet this requirement. Applicants are expected to have knowledge and control of all stages of the manufacturing process. And I want to highlight that this includes manufacturing and testing activities that are conducted at contract manufacturing organizations or CMOs. The facility assessment is a key part of the application for CDER assessments, and an acceptable outcome of the facility inspection is required for application approval. CDER pre-license inspections have a number of objectives. These inspections assess the readiness for commercial manufacturing of the biologic that's the subject of the application, and this includes not only an assessment of cGMPs, but also an examination of product-specific procedures and controls, equipment, facilities, cleaning, contamination and cross-contamination prevention procedures, personnel training, things like that. Conformance of what is actually performed in the facility with the information that's provided in the application, as well as the procedures and controls in place to ensure data integrity. In other words, that the data that we see in the BLA is reliable and was collected in a way that's transparent and well documented, are also examined. The inspection provides a holistic assessment of whether the firm's quality system, and by extension the applicant's, has sufficient knowledge and control of the manufacturing process and controls to assure the final quality of the product. Decisions about biologic facility inspections are risk-based, which means that all facilities in the application are assessed for whether an inspection is needed and what inspectional tools CDER might use as part of the application review. This assessment, as I said, is risk-based and considers the previous inspection history of each facility, which includes whether it has been inspected at all, or if it has been inspected, whether the observations or other information in the inspection reports suggests that there is risk that we need to assess. The inspection history assessment also considers information shared by trusted regulatory agencies, which might be shared, for example, through mutual recognitions agreements. We also consider whether the facility has experience with a similar manufacturing process to the one that's the subject of the BLA. We also look at whether the process uses new or novel technologies, or if the technologies are new to the facility or applicant. And we also assess if there are specific risks identified from the application review itself. FDA has a wide variety of inspectional assessment tools available to it. The backbone of CDER's inspection program is information gained by FDA inspections, both those conducted by CDER and, of course, those conducted by the Office of Regulatory Affairs. And we use the information we gain from those inspections in risk-based ways. But we have additional tools available to us as alternatives to inspections, or to augment inspections. The first such tool is records requests made under Section 04A4 of the Food, Drug, and Cosmetic Act. This document review information can be used in advance of or even in lieu of an inspection. The second tool is what's called a remote interactive evaluation, or RIE, in which interactive technology can be used to live stream or view pre-recorded videos of things like facilities, operations, data, or other information that we use in our assessment, or to interview staff at the facility. I want to highlight that providing documents that are requested through the 704A4 records request is mandatory, whereas participation in a remote interactive evaluation is voluntary. However, a facility declining to participate in a remote evaluation does not eliminate our need to conduct our assessment, and so FDA will need to use other tools, which might include an inspection in those cases. If we've selected a remote evaluation as a way to expedite the process, having to do an inspection instead may be a slower process to complete than a remote evaluation. And as we noted in the previous slide, we also use information gained from foreign partners under mutual recognition agreements or other confidentiality agreements to share information. And I want to note that the availability of a previous inspection report from a say, an MRA partner does not necessarily replace the need for an inspection. Just because they had an inspection with a successful outcome doesn't automatically mean that we wouldn't need to inspect. But we do use that information as part of our risk assessment, and the more information we have, the better decision we can make about what kind of tools we can use. So further on discussion of using remote regulatory tools as alternatives to BLA inspections, the use of remote tools such as record requests or remote evaluations to replace or shorten inspections is a risk-based decision, and that decision is made by the CDER review team. The team considers all available information, some of which might not be known to the facility or the applicant, and we consider risk factors such as the previous facility inspection history, process risks, microbiology concerns, product concerns, things like that. We also consider whether the use of remote tools can adequately support or mitigate the need for an inspection, and in so doing, potentially save the facility and the applicant and the agency resources. Of course, we also consider the impact of travel restrictions. For example, during the COVID-19 pandemic, we used remote interactive evaluation tools in cases where we weren't able to travel. Because of all these factors, FDA does not consider requests to use remote tools from facility or applicants. We instead make a decision as a team as to whether and what remote tools may be used, and then once a decision is made, we will contact the facility to request either things like documents from a 704A4 or records request, or to request use of a remote interactive evaluation. So this brings us to our second challenge question. Can a BLA be approved with outstanding facility deficiencies? A, yes, if the facility commits to correcting them. B, yes, if the facility requests a follow-up remote regulatory evaluation. C, no, satisfactory facility evaluations are required for an approval. Or D, yes, but only for 355A or non-biosimilar applications. And the answer is C. Successful facility evaluations are a requirement for approval. All right, so moving into our next section, I'll discuss an assessment that Joel and I did of recent complete response actions and the natures of the deficiencies that were identified in the complete response letters. So we surveyed a predetermined subset of complete response actions for CDER BLAs over roughly the last 3 years, and we assessed the deficiencies identified. We looked at a total of 32 complete response actions. Each letter might identify more than one facility, and a single BLA may have had more than one complete response letter. So you'll see in the subsequent slides that there are more than 32 deficiencies. I would also note that the 32 BLAs also included 15 biosimilar applications. So this first chart sorts CR deficiencies into 4 broad categories and identifies the number of times a deficiency in each category occurred. The 4 categories were 1, clinical or related issues, 2, issues related to the facilities where the manufacturing occurs, 3, issues in the chemistry, manufacturing, and control sections of the BLA dossier itself, and 4, issues in the data that would support the biosimilarity of a biosimilar to its reference product. And we found that the most common deficiencies were facility inspection-related issues. Non-facility issues in the CMC dossier, along with clinical information, was the next most common. Notably, the lack of demonstration of biosimilar to the reference product was not the most common deficiency for biosimilars. Facility or other CMC issues were. So unpacking the facility issues a little further, we categorized the types of facility issues into 11 subcategories. These subcategories are not necessarily mutually exclusive, and I used a little bit of judgment in sorting them, so a deficiency might fall into more than one category. So those caveats noted, the most common issues were related to cleaning procedures and controls, environmental monitoring, aseptic procedures, and bioburden control, as well as contamination and cross-contamination controls. A theme that occurred the most often was concerns about quality oversight, which is things like Did the firm conduct thorough investigations of deviations, and did they take sufficient corrective actions? Did they adhere to their written procedures? Did they have good documentation practices? Did they assure training of their staff? It's important to understand that issues that were sufficient enough to warrant a complete response action are often not resolved until there is a follow-up inspection that can verify the effectiveness and the implementation of corrective actions. So it really is a big delay to have to make corrections to the facility. So to ensure that new products are brought to the market as quickly as possible, I think this information tells us how critical it is to development programs that applicants ensure that their facilities and their quality systems are ready for commercial manufacture, commercial production, commercial level GMP standards before submitting a BLA. So next we examine the deficiencies in the CMC sections of a BLA dossier other than those related to biosimilarity. Once again, these categories are not necessarily mutually exclusive, so there's a little bit of judgment call in these sortings. As you can see, microbial control of both drug substance and drug product was a frequent concern, as were other manufacturing controls and validation. Specifications and issues related to stability testing and stability programs were also quite common, as were the specification and stability controls regarding the reference standard qualification and requalification. So reference standards come up as well. We also saw that control of cell banks and other such critical inputs was a recurring source of issues. So the key conclusions for this CR evaluation of this survey of CR deficiencies were that facility issues were the most common reason for complete responses, and CMC dossier issues other than the facilities and clinical deficiencies were the next most common. For biosimilars, facility and CMC deficiencies are more common than issues in demonstrating the biosimilarity of the reference product. We covered how CMC issues can span a variety of topics. This includes manufacturing and process control, microbiology, being the largest categories. Specifications, analytical methods, stability, including those for controlling reference standards, are frequently CR issues, as are issues in cell banking. Inspection issues span a variety of topics, including environmental monitoring, equipment cleaning, aseptic procedures. But I think even more importantly, insufficient oversight by the quality system was the most common theme that our inspectors brought to us. And this underscores that site selection and readiness for commercial manufacture are paramount to biologics development. So our third challenge question is, for BLAs and CEDAR which receive a complete response, what are some potential causes? A, CRs may occur only for clinical reasons. B, CRs may occur for manufacturing facility reasons. C, clinical, product quality, and manufacturing facility reasons are all common causes, or D, none of the above. And the answer is C, all sorts of issues, including manufacturing, quality, and facilities, are common causes. And this slide provides some handy hyperlinks to some of the things that are related to some of the things we talked about and can provide additional information. And finally, in summary, I hope we've demonstrated for you that biologic products have unique scientific and regulatory considerations, and that inspection plays a critical role in CEDAR's assessment of BLAs, and that we use a variety of tools as part of our inspectional assessment. And finally, the successful development of biologics requires CMC development along with clinical development, and this CMC development includes ensuring the readiness of manufacturing facilities for commercial production. And with that, we'd be happy to discuss any questions with you, and thank you so much for coming today.
Thank you all for the great presentations. As we move into our final Q&A panel, if you haven't had a chance to submit your questions in the Q&A chat pod, please do so now. Lance, there's questions as time allows. To Doris Chin and Ms. Chin, is it the location of the agent or the initial importer which dictates where foreign manufacturers shall report to?
Hi, great question. The foreign manufacturer, the geographical location of the U.S. agent determines which ORA farm division they should contact. Thank you.
Thank you for responding to that question. We have a few more questions for Ms. Chen, and here's the next question. When our raw material supplier makes a recall, what should we do as an impacted industry?
Another great question. So when a raw material supplier, such as an API, active pharmaceutical ingredient supplier, or a excipient supplier, conducts a recall, if you have repackaged the API, if you're a distributor that sells, these API ingredients and you've repackaged it, you should conduct a downstream recall on your own and send notifications to your consignees. If you use those, either the API or an excipient in a finished product, your finished product is now adulterated because you made it with recalled products. So you would contact your ORA farm recall coordinator and conduct your own recall. Thank you.
Thank you for responding to that question. We've got a couple more questions from Ms. Chen, and here's the next question. Is the recalls and shortages team the same as the drug shortage team?
Hi, great question. No. So the recalls and shortages team is in the Office of Compliance, and we coordinate with our colleagues in the Office of Compliance to evaluate and make regulatory decisions to allow a defective product to remain on the market if the benefit outweighs the risks. The drug shortages team is in the Office of the Center Director, and they work with other groups such as the Office of New Drugs, Office of Pharmaceutical Quality, Office of Biotechnology Products, and Division of Microbiology Assessment. And they evaluate the safety and clinical impact of the defective product to make a recommendation of benefit over risk. Thanks.
Thank you for responding to that question. We have one more question from Ms. Chen in this round, and here's the question. I have a defective product that currently is on the drug shortage list, but I don't think that this will cause a disruption in market supply. Who should I contact to discuss if I can move forward with my recall?
Hi, so that is a great question. If you don't believe your market action is going to cause a disruption in supply, you would contact your ORA farm recall coordinator before you initiate your recall and provide information like quantity of product distributed, the dates of distribution for your proposed recalled lots. FDA is also going to want to know your planned amounts and dates for replacement of your recalled products. So ORA Farm will send this information to my team, who will verify that your market action doesn't exacerbate a current shortage or create a shortage. And we want to do this before you initiate your recall. The drug shortage team will evaluate your market action and its impact across all suppliers of the product. So please wait for ORA Farm to relay that our team has contacted drug shortages before you move forward with your recall. If you believe your product will cause or exacerbate a shortage, you would reach out with your proposal to the drug shortage team, and that information is located on my slides. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we have got a group of questions that came in for Dr. Joel Welch. And here's the first question for Dr. Welch. What if you have a trimer of proteins in varying length from 35 to 55 amino acids? What division would you work with, C-DIR or C-BIR?
So thank you for that question. It's a very good one. I think you can appreciate from our presentation, we tried to create and provide some general frameworks for how these products are regulated and classified. Certainly when there are unique complexities around a particular product and where it should be and how it should be regulated, the easiest and recommended path would be to request a designation. A request for designation submission is described on the FDA website, and that would be the most appropriate mechanism to figure out where your product would reside and how it would be regulated. So we would recommend you reach out that way to get a firm response for a specific case. Thank you for the question.
Thank you for responding to that question. We have a few more questions for Dr. Welch, and here's the next question. How many PPQ batches are considered appropriate for biologics, 2 or 3?
So thank you for that question. In the CGMPs and FDA policy, there is not a specified minimum number of batches to validate a manufacturing process. Rather, FDA recognizes and acknowledges that any manufacturing process or even a change to a process can't be necessarily boiled down to a specific number of batches. Rather, a minimum number of conformance batches is typically needed to validate a process, but we expect sponsors to have a sound rationale for its choice for what that looks like, and to approach the agency with science-based approaches for how they would intend to complete process validation. If there are specific questions about a validation program, that's always a good topic for a potential meeting with the agency under the appropriate user fee program. Thank you for the question.
Thank you for responding to that question. We have one more question for Dr. Welch in this round, and here's the question. Are type 2 DMFs of biological manufacturing platform, DS intermediate, DS and DS permissible for biologics IND?
Great question. Thank you for that. In general, it is potentially permissible to use a type 2 DMF in those cases under an IND. The best resource to understand a little bit more of those limits would be the 2019 FDA proposed rule on type 2 DMFs for biological products. It's on the Federal Register website, and it would provide more specificity, including limits around DMFs, but in general, it is permissible to potentially use them under an IND. Thank you for the question.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a group of questions that came in for Dr. Christopher Downey. And here's the first question for Dr. Downey. Would FDA agree with an applicant timing its BLA submission such that a validation protocol is submitted in the BLA and executed validation files and the validation report are complete and reviewable during a pre-license inspection?
Thank you for the interesting question. In general, the short answer would be no. The application is expected to be complete at the time of submission, and that would include the process validation is complete and the report is available in the submission. That is reviewed as part of the BLA review. It's not reviewed as part of the inspection. So the inspection is more facility specific than that. So the inspectors aren't reviewing application materials as part of their inspection generally. They're reviewing the facility practices and GMPs and how that facility is performing the stuff described in the application. In rare cases with the breakthrough product or an expedited program, we might discuss with an applicant if they have fewer than 3 PPQ batches, the potential of submitting a concurrent validation protocol where they would submit partial validation data from an initial batch and then additional validation post-approval. But that's sort of not the norm. It would be something that would need to be worked out in advance with the FDA. Thank you.
Thank you for responding to that question. We have a few more questions for Dr. Downey. And here's the next question. Could you elaborate on one of the inspection deficiencies, conformance to application?
Yes, thank you for following up on that one as well. I think I alluded to it in my previous answer. The conformance application means is what the manufacturing and testing activities being performed at the facility, do those match what is described in the application? I know from my own experience, for example, I've been on an inspection where there were manufacturing steps being performed at the facility that weren't described in the application. And so that can be a potential concern. And one of our objectives of an inspection is making sure that the process we are licensing matches the process is actually performed on site at the facility. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Downey. Here's the next question. How soon would a follow-up inspection be?
Thanks. There's not a set time. A pre-license inspection is tied to the timing of that is tied to the submission of a BLA application and the manufacturing schedule of the facility itself. So if an inspection generated observations that ultimately led to the facility not being approvable, whether there was an additional inspection needed that decision would be made during the review of the resubmitted BLA, and again might be dependent on the manufacturing schedule of the operations performed for that application that's submitted by the applicant. So it really depends on the applicant. That said, I think it's important to highlight that the corrective actions should be in place and the facility should be ready to be re-inspected. It doesn't do any good to immediately resubmit an application that has received a complete response if the facility isn't completed with its corrective actions or have data to support that the corrective actions are effective, because if FDA goes back and the issues are still there, then there'll be the same observations again, and ultimately the application could again not be approvable, and now essentially the time has been wasted by the applicant submitting too soon. So it's somewhat dependent on resolving the timeline of resolving the problems and the strategy of the applicant in terms of how they determine that and resubmit their application. Thank you.
Thank you for responding to that question. We've got just barely over a minute left. I'm going to try to squeeze in one more question for Dr. Downey. And here's the question. Can you comment on the FDA view on reliance on mutual recognition agreements and applicability to inspections?
Yes, so we utilize mutual recognition agreement partner inspections, and we evaluate their reports, and that goes into our risk assessment in terms of what type of inspectional assessment is needed. You have to bear in mind that the partner may not have covered the same products on their previous inspections. It may have only been a GMP inspection. It may not have the same products or objectives as a FDA pre-license inspection, so it may not by itself eliminate the need for an inspection, but we do rely on the information available to determine risk for the facility, and if there's a lot known about the product already from that inspection, that can help us determine the facility's low risk and make decisions in that way. Thank you.
Well, that's all the time we have for questions in this panel, and we want to give a huge thank you to all of our presenters and panelists for fantastic presentations and answering numerous questions that came in during all the Q&A panels. We have the pleasure of welcoming back for closing remarks our Small Business and Industry Assistance Director, Captain Brenda Stoddard. Welcome, Captain Stoddard.
Thank you very much, Captain Ford. Well, everyone, sadly, we have come to the conclusion of a CEDAR Drugs Track REdI 2023. And I truly hope that you have enjoyed the 2 days and have found many, if not most, of the presentations to be helpful to you, worthy of saving as references and sharing with your colleagues. We have covered a lot of territory in the past 48 hours, and I am truly impressed and reassured with its relevance in that so many of you attended from literally all over the globe, and so many of you stayed with us until now, the very end. I thank every one of you, also our presenters and panelists, for their exceptional presentations and very expansive Q&A sessions. And of course, our technical staff for handling and troubleshooting all the logistics behind the scenes. My thanks also to our SBIA's Captain Ray Ford and Lieutenant Commander Ray Newell for moderating over the 2 days. Within the overall context of PDUFA VII, PDUFA III, and AMUFA, we provided the most current intelligence on many CEDAR programs. We addressed the various user fee meetings, bioinformatics and electronic submission practicalities. We discussed digital health technologies from the review and IT perspectives. We took a look at a couple of our pilot programs at user-related risk analysis and human factor protocol reviews. We provided valuable information on decentralized clinical trials and post-marketing commitments. We gave you very timely insight in our rare disease endpoint advancement pilot program. And then we wrapped up with CMCs and its development and readiness pilot program, recalls, and biologic manufacture and inspections. It is my firm belief that if you walk away today with validation of your current processes, or perhaps you realize that you are not doing things as the FDA recommends, and now you know exactly what to do. If you receive new and applicable information, you got responses and clarification on your questions, then SBIA would have achieved its purpose. And to emphasize the one recurring theme in all our presentations and Q&A sessions, and that is to contact CEDAR early and often. This approach is in everyone's best interest. As a reminder here, and I'll try to do this quickly, I know everyone wants to leave at the end of a 2-day conference. This 5-day event is offering a total of 32.25 contact hours to physicians, pharmacists, and nurses. To be eligible for this, you would have had to self-identify as a healthcare professional during the registration process and attend via Adobe. No healthcare CEs can be given for those who attended via YouTube without the benefit of registration or for post-event viewing. The deadline date for the CE submission is June the 23rd. And then we have our general event evaluation and survey, which is the mechanism by which you will receive the certificate of attendance, which will support continuing education credits for RAPS, SOCRA, SQA, and ACRP. When an attendee completes the survey, they can then download a certificate of attendance, which may be used in support of those CEs. The certificate must be downloaded immediately after completing the survey, otherwise the opportunity is lost. And I do want to emphasize that the certificate of attendance and the healthcare CE submission are two separate processes. You know, a person can have them both, but they're two separate processes. In order to simplify things, each night SBIA will send one email to the attendees of that individual event day. The email will have information on both the CE claim code and instructions, and also the survey link. Each track will have its own survey link. The deadline for both processes, the healthcare professional and the certificate of attendance, is June the 23rd. No exceptions. So I entreat you to complete your paperwork upon receipt of our email at the end of each day. That way you don't get distracted and you don't miss the deadline. All the sessions are being recorded. It will take us about a week or so to process the recordings, but you will soon be able to access them from SBIA's webpage. And please bookmark that webpage, fda.gov/CEDAR-SBIA. When you go to look for this specific event, you can go to our shortened URL, which is fda.gov/REdI2023. Again, here is our contact information along with our important websites. Now, if you submitted a question during these 2 days and we were unable to respond at that time, or if you have a question later on after these 2 days, please feel free to contact us via our email here, cedarsbia@fda.hhs.gov. If your question refers to a specific presentation and speaker, please put that information in your email subject line, as that will assist us to more easily triage your inquiry. Remember to subscribe to our email service and follow us on LinkedIn to stay abreast with CEDAR's communications and SBIA's upcoming events. For those of you who are attending our Devices and Biologics track, please learn as much as you can and enjoy the dynamic presentations and Q&A sessions we offer to you for the rest of the week. Our Devices track will begin tomorrow at 8:30 a.m. Eastern Time. Meanwhile, farewell and thank you.